existing the OTC drugs, and OTC drugs are used without a prescription, and you can buy over the counter and take the medication. That will be giving a lot of side effects to the customers and using the people who are using, and we have to be very careful in that. You can see 114 billion US dollars means that's the highest use of OTC medication. And this information is given over 200 markets in 150 countries. That $114 billion was calculated. When we talk about the statistics in market value, it depicts the top 10 biotech and pharmaceutical companies worldwide based on a market capitalization of 2021 as of September of 2021. And Johnson & Johnson has a market value of 454 billion US dollars. So it is ruling the worldwide market value. And when we talk about, because the development is required and COVID pandemic, we need a lot of research and development has happened in 2020. The research and development spending in the pharmaceutical industry is totally near about 200 billion US dollars. 200 billion US dollars is very high amount for the research and development. That's why we have come up with several vaccines and medications to treat the pandemic also. And total R&D expenditure was $137 billion in 2012. But when we come to 2020, it was $200 billion, a tremendous increase in the research process, okay? And this research is, the amount is compounded over preclinical, clinical stages, and marketing also, the medication. When we're talking about which novel drugs are releasing in the statistics, when we see that, into 2020, there is a 53 novel drugs were approved by Center for Drug Evaluation and Research. Even though it's a pandemic and everything was shut down worldwide and everything was like, closed, but pharmaceutical industry, which has come out with 53 novel drugs during this pandemic time. The number of new medical products entering the pharmaceutical market each year varies heavily, but for example, when you take the statistic into
Ma'am, there is a network issue. Sunita ma'am. Ma'am, we cannot hear you, ma'am. Sorry about the technical glitch faced from the conference co-chair side. Maybe we'll connect back to her in a few minutes. So meanwhile, I would like to invite our conference chief patron, CEO and founder of the TechnoRail Group, Mr. Rudra Banu Satpathy. Good morning, sir.
Good morning.
Welcome, sir. I would like to welcome you to address our presenters who have joined here for the 9th World Conference on Pharmaceutical Science.
Very good morning here. Thank you very much, all the speakers, co-chairs, everyone, for their kind and valuable support towards World Conference on Pharmaceutical Sciences and Drug Discovery, scheduled at Dubai in hybrid mode. So it has been a great experience towards this conference, and this journey was like we began working on this conference since last. We have been working. My team has been working on this since last 9 months, and we have been connected with many organizing committees. So my special thanks to all the organizing committee members for their kind support and valuable addition to the value addition to the conference, as well as we had more than 140 informative presentations. We had scheduled the conference keeping in mind of COVID pandemic, so that we can mobilize the things like our vision and our mission directs us to work in a particular direction of congregating the research ideas, congregating the innovative thought processes and research works of all professionals all across the globe, because this is a high time that everyone should unite, integrate, and work together to contribute towards scientific advancement. So we took up this conference as a responsible organization, as a responsible scientific community and academic fraternity, by connecting all universities and connecting all companies and corporates and industries. A forum needs to be developed on which the scrutinized, shortlisted, and selected articles must be presented, and that should be globalized towards the innovation and the implementation and trials of the same should be done. So on this perspective, we scheduled this conference, and we got an overwhelming response. We received numerous articles from nook and corner of the globe, but unfortunately, we have to select few articles on basis of the quality, because we focus and we concentrate that the content being produced by our organization in this forum needs a lot of refurbishment and a lot of selection, so that we can deliver quality content to the academics and to the globe. In this regard, I express my hearty gratitude to all 14 keynote speakers for their valuable time, my honorable conference chairs and co-chairs for their support and their tremendous effort towards this, all scientific community members and scientific reviewers, as well as academic session chairs, as well as everyone. I mean, to my all crew members and everyone, all my supporting team, the team of Shravani, team of Jazila, who have done tremendous work on this, the pharmaceutical unit of BioLeaks group has done a tremendous job. And there were a few technical slags, I believe, and we apologize for the inconvenience, and we will try to fix them in our upcoming conferences. And because our commitment was that we will create a environment where there will be more like physical and virtual interactions, and ideas of physical attendance will come to virtual participants, and virtual participants' thought process should reach everywhere. So this was done. And in upcoming days, we are rapidly collaborating with many universities, academic institutions. I welcome everyone to be part of our organization, because we have individual memberships and premium membership also for individuals and organizations and universities. So universities and pharmaceutical colleges all across the globe could be a partner, academic partner of the organization. And we believe that together will definitely bring the difference in upcoming days. Thank you very much for your valuable support. Thank you.
Thank you, sir. Thank you so much for your kind words. Thank you so much. So next, I would like to invite our conference chief guest. Unfortunately, he was unable to join us live, but he has sent across his video message. So I would like to welcome our chief guest, Asif Mahmood, the Chief Safety Officer from VP Global Clinical Safety and Pharmacovigilance at Medicago, Durham, North Carolina, from United States of America. invite So meanwhile, our conference chief guest connects, we will invite our other keynote speakers of the day. That is, first will be Professor Dr. Awad Mansoor, Professor, Pharma Tech International from Greater Chicago from United States of America. Then I would like to welcome and introduce our second keynote speaker of the day who will be joining us soon, Dr. Dan Lim, Vice President, Advent Health University from United States of America. Dr. Gunawan Indranto, Professor, Universitas Surabaya from Indonesia. Mr. Santanu Simhapatra, the Principal Scientist from Biocon Research Center, India. Keynote speaker, Gail Jaron, co-founder and CEO of refinedsourcing.com from Paris. And our last keynote speaker for the day will be Shri Ashish Kumar, Vice President from Zydus Group from Ahmedabad area, India. And I would also like to welcome and introduce to you our session chairs for the day. Dr. Jayavardhan Patel, Dean from Department of Pharmacy, Sankal Chand Patel University from India. Professor Dr. Nehla E. Ismail, the Council Member from Malaysian Academy of Pharmacy, Malaysia. Nida Bawarsad, Assistant Professor of Department of Pharmaceutics, School of Pharmacy from Ahvaz Jundishpur University of Medical from Iran. And our session chair, Dr. Bonta Venkata Lokesh, Senior Lecturer from University of Malaya from Malaysia. Dr. Firoj A. Tamboli, Head and Department of Pharmacognosy, Bharati Vidyapeeth College of Pharmacy from Maharashtra, India. And Dr. Azimova Bahtigul, the Senior Lecturer from Tashkent Pharmaceutical Institute from Uzbekistan. Thank you all for joining. Welcome to the 9th World Conference on Pharmaceutical Science and Drug Manufacturing. So now we'll quickly move on to our keynote presentation for the day. So I would like to welcome Dr. Gunawan Indranto, the professor from Universitas Surabaya from Indonesia. Welcome, sir.
Hello, welcome. Good morning. Can you hear
sir.
my voice?
Yes, I can hear you. How are you? Thank you for joining.
How are you?
Good.
Okay. Should I present my presentation now? Or?
You may share your screen, sir.
Sorry?
You may share your screen and begin your keynote session.
Okay, I will share my presentation.
Sure.
But please enable, still disable.
1 minute. Yes, you may try now.
Can you see this slide?
Yes, it's visible, sir. You may begin.
Okay. Okay. Good morning, ladies and gentlemen. It is a great honor that I will invited in this seminar. Today, I will talk about recent development on chemical validation method for drug analysis. This is based on current USP 2021 and related publication. In quality control laboratory, as you see, the result of analysis can be affected by some many factors, including the qualification of the analyst, the sampling method, the stability of the target compound in sample, using the method. The method should be validated method, and to prove that the validated method still valid during application, we need to apply quality control sample. Quality control sample should be analyzed together with the sample. If the result of quality control sample are okay, the analyst can have conclusion whether the product is accepted, okay, or reject. But if the quality control sample is not okay, it means that the whole analysis should be discarded. In this presentation, I will concentrate to discuss how to make a validation or verification of the method. For performing validation of the method, we should know first the goal, the analytical target profile that describe the objective and the acceptance criteria of the method. Method can be selected from the current pharmacopoeia, that is the recommendation, or can be selected also in publication. And then we can select the method depend on the equipment or region that we have. After that, the method is selected, we should try first the stability, and the SST should be tested before to evaluate the validation parameter. If the validation parameter performed at the R&D laboratory, we should do a method transfer from R&D to quality control. At last, of course, the validation report and the SOP should be written and developed. According to the USP, the current USP, the validation of analytical procedure is the process by which it's established by the laboratory studies that the performance characteristic meet the requirement for the intended analytical application that described by the ATP. By USP chapter 1226, user of analytical method, mostly chromatography, which described in USP and described in monograph, are not required to validate the procedure, but merely verify the SST of the method under actual condition. Verification process of compendial test procedure is the assessment whether the procedure can be used for intended application under actual condition of use for a specified drug substance and or drug matrix. If the user found that the SST is not comply the monograph, they can make a condition. They can make the change the condition, but it should be according to a chapter of chromatography 621. This is the slide of what we mean master golden rule of validation method. This means the analytical procedure has to be validated as a whole, including sample treatment prior to analysis. The analytical procedure has to be validated, covering the full range of analytical concentration specified in the method scope, and the analytical procedure has to be validated for each kind of matrix where it be applied. So it means if we do the validation for capsule, it's not valid for syrup. And if we do validation for blood, it can be used for urine and so on. That is what they call the Masad golden rule of validation. Selectivity according to the EuroChemCat 2014 is mean the extent to which the method can be used to determine particular analytes in the mixture or matrix without interference from the other component or similar behavior. But specificity according to current USP defined as the ability to assess unequivocally of the analyte in the presence of component such as impurity, degradation product, and matrix component. Lack of specificity of individual analytical procedure may be compensated by other supporting analytical procedure. In USP, it can be done by specing the drug substance or product with appropriate level of impurity or excipient and demonstrating that the assay result is unaffected by the presence of this external material. But if the impurity or degradation product are unavailable, specificity may be demonstrated by comparing the test result with the sample that contain the impurity. Or we can incubate the sample in stress condition, something like this. This is our work. We do not have any impurity. That's too expensive. That's why we incubated this sample in hydrogen peroxide, in hydrochloric acid, and sodium hydroxide. And after some times, we find that some degradation product is formed, C. So in this case, we should prove that the peak A and B still pure. Then it can be proved using Day2 detector or mass spectrum, something like this. We should prove that if we can measure in the some point of the peak, if the UV spectrum is identical, it means that the sample is pure. If it's not, it means the sample is not pure. The peak is not pure. But Horizon SPTLC and TLC equipment have been completed with software which can perform this evaluation easily. We can just only click and then we can know whether the peak is pure or not. But the problem is, although the peak have identical UV spectrum, different compounds may have similar identical UV or V spectrum. That's why we should use another detector. Mass spectrum is better. This I will show you the application of the MSMS. Every organic compound has specific mass spectrum. Every organic compound has specific mass spectrum, something like infrared and photons. But if we have mass spectrum, something like this, in this case, we can select 210. In this case, the ion of 210, we can insert to second mass spectrum, and with the second mass spectrum, it will separate to fragment to daughter ion. In this case, ion 158 and 191. This is the fragment ion. Reaction 210 to 119 and 158 is called multiple reaction monitoring. But the reaction from 210 to 191 is called selected reaction monitoring. So this is show that every compound has specific molecule ion and has specific fragment. So if we know the molecule ion and the fragment, we can confirm the identity of the compound according to guidance of European Community. In this guardian of European Community, to confirm the identity of a peak if the identification point has 4 and the ratio of the two daughter ion within 4 plus minus 40%. For the example, something this is the precursor molecule ion and this is the daughter ion. That is specific for years of compound. For only liquid chromatography mass spectrum only, that's why it's need 4 specific fragment because the IP is 1. But for LC-MS-MS, it needs only 1 precursor and 2 daughter ion. Totally, the identification point is 4. But if we use liquid chromatography, high resolution mass spectrum, it just need 1 precursor and 1 daughter ion to have 4. That is for identity every peak using mass spectrum that should fulfill this regulation of the European Community number 808. This is the example. This is the example. If we do not have any specific detector like mass spectrum or Day2 detector, we have only ALSD or UV fix to prove the selectivity of certain compound, something like that. This is 3 in the sample. We should change the condition at least 3 times. If the retention time of standard ensample should be identical in every condition. That is, if we do not have any Day2 detector, we just have a non-specific detector like ASSD or RIT or FIT, GCMS, something like that. This is the requirement from the Eurochem 215. This is described here. The chromatography could be repeated using a column of different polarity. We can use column of different polarity. We can use mobile files and other. So we should prove that the retention type is always identical. That's the method of selectivity if we do not have any specific detector, some kind at Day2 and mass spectrum. No, and for linearity, the linearity of analytical procedure is the ability to elicit test results that are directly or by well-done mathematical transformation proportional to the concentration of analyte in the sample within a given range. This is, we should try to find correlation between concentration and response detector. For that, whether we know whether the linearity is okay or not, the USP describe the correlation coefficient, I-intercept, slope of the regression line, and residual sigma square. That is the requirement that is different from use of equipment. ICH recommend that the establishment of the linearity, it needs minimum 5 level of concentration. But Eurochem 2014 need 6 to 10 concentration. This is different guidance. It seems like different regulation. This is important. This is described in Eurochem 2014. Described that it cannot be performed by dilution of stock solution. That's described in this Eurochem 2014. So it means that for the calibration standard, calibration spot of standard should be independent of use of other spot. Should be independent. Independent, I mean it cannot be made by dilution of one stock solution. It should be diluted at least from two or more stock solution. It is nice that the calibration, if we do calibration, should be applied using uniform volume of solution, but different concentration. It is not nice if we use different volume of solution, example 1 micro, 2 micro, but with the same concentration. That is the parameter of the linearity. There are many parameters as described in this slide. AOSC in 2013, Appendix K, state that use of correlation coefficient as the test of linear test is incorrect. This is also new OSC. This is 2019. This is the same thing. That use R as a test for linear test is incorrect. That's why we need, if we just only R, that is not nice. We should calculate it with many one of two of this parameter. This is for the example from ISO, from a Mendel test. This is some parameter to prove the linearity. Indonesian standard SNE 70.035 using back calculation not more than 5%. I will show later. This is the calculation of SNE. This is the formula for calculating phase X0. ISO did not use R, but they used phase X0 for express the linearity. And for the others, they show back calculation. This is used also for bioanalysis. The back calculation, the difference is not more 5% according to SNE. If you use FDA, it's about 15%. This is back calculation, how to express the linearity if we use back calculation method for the testing of the linearity. This is show that I've told you R square almost 1. The error is also 1%. R square 0.987, the error is 4%. This is show why we cannot use R square only. We need other parameter to express the linearity. No, I will process to accuracy and precision according to USP. This is the definition. It's written. I copy from the USP to this slide. This is the definition of accuracy. The accuracy of analytical procedure is the closeness of the test result obtained by the procedure and the true value. How can we determine the accuracy? According to the USP, for determination, this is 3 methods. The first, by application of the analytical procedure to analytic down purity. This must be with using some reference standard. This is for the example, if we want to try to find accuracy of such standard metal by AES, we can buy the standard reference standard. Or by comparison of the result of the procedure with those of second well-characterized procedure. So it means we compare our proposed method with validated method. And the third, accuracy can be determined by application of the analytical procedure to synthetic mixture or laboratory made mixture that we have make a certain preparation with certain concentration, and then we analyze using our proposed method. The assessment of accuracy can be accomplished in variety. Jadi to know whether our accurate or not, USP describe here evaluating the recovery. And number 2, evaluating the linearity of the relationship between estimate and actual concentration. The slope should be around 1. This is the requirement of the USP chapter 1225. For determination of the accuracy, using at least minimum 9 determination over a minimum 3 concentration level. So it's been 3 times 3 level, 3 replication. This is the accuracy according to USP, current USP chapter 1225. Definition of accuracy according this chapter. The precision of analytical procedure is the degree of agreement among individual test results when the procedure is applied repeatedly to multiply sampling of homogeneous sample. I have only copied from the USB and I move to this slide. The precision of analytic procedure usually expressed by RST, standard deviation or coefficient variation. There is three level of precision. This is repeatability if we done in the same laboratory, in the same analysis, in the same equipment. But if we do with other analysis, other analysis or different analysis or different days, it called within laboratory. It called intermediate precision or ruggedness. But if we do it a different laboratory, it called reproducibility. This is the determination according to USB. Assay in this context are independent analysis of sample that have been through the complete analytical procedure from sample preparation to final. This is the definition, and this is the level of precision. This is system precision. This is repeatability. If we do it at the same laboratory with the same analysis, with the same equipment, that's called repeatability. But if we do with different operator, with different time, with different, it's called intermediate precision. But if we do collaborative trial, it's mean doing at different laboratory, it's called reproducibility. Or if we can do for the long term, maybe months or years. What we call with system precision is system precision of the equipment. For the example, if we use auto sampler, we should know that our auto sample precise. I mean, if we can inject 1 micro, should be 1 micro, not 2 micro. That's why we should check the system precision of our system. This is the method calculation of intermediate precision according to Ermer. According to Ermer, this is the intermediate precision. This is the term of repeatability. This is run. This is run. This is called variant run or emitting run. And this can be calculated using Excel, sample like that. After we can get this variant runs, we can get the intermediate precision using ANOVA. This is the example. Jadi, this is the between treatment. This is between treatment. This is repeatability. This is between treatment. This is something like that, between treatment. And then from that, we can calculate P as P square. And from SP square, we can calculate intermediate precision. Repeatability is the square root of the between variability. This is intermediate precision, and the units in USB is described, but the equation is not described. This is the determination of accuracy according USB 40. This is, I've called you this one, the percent recovery is mean we analyze a certain sample that have certain concentration, and we should calculate the percent recovery. How many percent the difference between the actual and the estimate or the predicted. And the number two, evaluation the linearity of the relationship between estimated and actual, something like that. This is the formula for calculation. In this last presentation, I will give you some link to download our self-made software that can all calculated all parameter. So the acceptance criteria, slope is not significantly different from 0, and intercept is not different from 0. Slope is not different from 1. Sorry, this is the requirement from the USB 2025. So the acceptance criteria for accuracy percent recovery is described by the ATP based on the specification ring is described by the current pharmacopoeia. Years of drug in the monograph describe the specification range, maybe from 95 to 105, something like that. In USP, there are other USP 1210. There are other methods to evaluate the accuracy and precision. This is the two separate evaluation and combined evaluation. This is separate evaluation, accuracy and precision separate. It's been accuracy and precision evaluated separately. In this method, they do not describe the percent recovery, but accuracy described as confidence interval of bias. Confidence of bias, that is the equation, and confidence interval of SD, that is the equation. The interval of bias must be included in the specification range of the ATP. That's why in ATP, it should be described what is the minimum and maximum bias, and what is the maximum confidence range of the standard deviation. That is the example of the calculation I copy from the USP. This is the data, and this is the calculation for confidence of bias and confidence of standard deviation. And USP that describe combined evaluation of accuracy and precision. It's been accuracy and precision evaluated combined together, not separate. For this method, they have using two methods. That's using prediction interval and tolerance interval. This is the equation. This is the mean, and this is the prediction interval. This is the mean. This is the prediction tolerance interval. This is the summary, the calculation. For the tolerance interval, you can find this link, and you can calculate the tolerance interval without manual calculation. This is summary of the evaluation of accuracy and precision according to USP. This means if this is upper limit, this is lower limit, every result of analysis should be included in this range. Not the mean, but all. This is not okay, this is okay. I mean the data, not the mean include in this specification rate, but the whole data. So mean every result should be included in this range. This is what they want, this with prediction interval and tolerance interval that we can calculate with prediction or tolerance interval. So it is better if we describe our analysis mean plus minus tolerance interval or prediction interval instead of using SD, instead of using standard deviation. Now I continue to detection limit and quantitation limit according to USP. Definition of the detection limit is the lowest amount of analysis of a sample that can be detected, but not necessarily quantified. Determination of detection limit by the ratio of the noise signal to noise 2 to 1 or 3 to 1. But this is the definition of quantification limit. This is characteristic of quantitative assay for low-level compound in sample matrix, such impurity in bulk drug substances, and so on. It is the lowest amount of analysis that can be determined with acceptable precision and accuracy. Some can they use 10 to 1, but it is not nice. The detection limit is important if we want to determine impurity, degradation product, cleaning validation, heavy metal, toxic metabolite, pesticide, bioanalytical method, residual solvent. For this kind of analysis, we should determine the detection limit and quantification limit. This is the best method for determine detection limit or limit of detection according to USP chapter 1210. That is, if in this method, we should develop a linear regression of our material with low concentration, with very low concentration. Then we can calculate the confidence of this regression line, and this is the limit of detection according to this USP and this equation to calculate detection limit according to USP. That is the data, that is the example. For limit of quantification, the value of this segment replaced with 10. According to FDA 25, we also need to determine sample detection limit and sample quantification limit. It means if we work with different sample, different metric, for the example, urine, herbal, or other different, this is difference detection limit or quantification limit. Although the calculation of the detection limit according to USP to the standard is identical. This is how can we do that? Something like that. We should seven times, spiking 7 times our sample matrix with the estimate LOQ, and then calculate the standard deviation and the limit of detection sample and limit quantification sample can be calculated with this formula, with this equation. This is according VICS 49 of FDA 2515. You can download within this link if you want to know detail. No, as I told you before, I want to give brief example for proving that the use validated method. We know that for the analysis we always use validated method, but we should to prove our validated method is still valid during our application. This is according EuroChemCAT 2014-2016 and FDA Oral Laboratory Manual 2019. So in this case, as I described before, if we analyze sample, we should also analyze quality control sample. Quality control sample, a sample, stable sample that already usually made by R&D. So if the result of quality sample is okay, is mean our analysis is correct. But if the analysis of quality control sample is not correct, so our method is wrong. Usually, if we have 20 sample for analysis, at least we should have 1 quality control sample, or about 5%, something like that. If we use many sample, hundreds of sample, if we use auto sampler, if we mean 100 sample, we should 5% from 100 sample, we should have 5 quality control. And quality control, we should determine together with the sample. And the result of the quality sample is depend, it can be something like this, 2 SD or 3 SD as we wish. And if the result of the quality control sample outside this limitation, it means that our method need to be revalidated. That is what I explained before regarding a quality control sample. If we do analysis in quality control laboratory, we analyze our product or sample together with quality control sample. If we do not analyze quality control sample, we cannot sure that our method still valid or not. Now I want to show you, as I told you before, the process, the method of validation could be developed by R&D laboratory, or the method of validation can be established with other institution. So for applying this method, should be transfer from the R&D to quality control. For this transfer, there is two methods. This is comparative testing, comparison of proposed method and validation method. Co-validation is mean the staff of quality control should be work together with R&D to develop the validation or revalidation. This is the easiest one. My experience is using co-validation. So if the R&D develop some certain method of analysis, the staff of quality control together with the staff of validation R&D do the validation. If something like that, we do not need use transfer of method comparative testing as required by USP 2024 and 2024, something like this. For comparison procedure, comparison is mean that the sample should be analyzed from the R&D and should be analyzed from the quality control, and then should be compared with using comparison, and this is the equation that should be less than D. D is the maximum permitted difference between new and old method. In the USP, there is a mathematical calculation. It was recommended that number of replication is at least 18 to get power of analysis 80%, and to get power 90%, the replication should be 23. That is, if you want to know why something like that, you can refer to this USP. How to calculate is described in detail. This is the formula, a copy. This is the formula. You can use this via Excel, and this is example of calculation. This is the mean of new method or proposed method. This is the mean of old method or validated method, something like this. This is the variant, and then using this formula, this equation, we can calculate something like that. This is if we use homogeneous sample, comparative for homogeneous sample, homogeneous material from R&D and quality control. If the sample is not homogeneous variation, the formula, the equation is a little bit different. This is the equation to calculate D, and this is the complete formula, and this is the calculation. This is same but different formula because the sample is not identical. The sample is different batch or different material, something like this. This is should be used scenario 2. This is all described by USP 2020 or 2021. This is the same. This is the calculation. I think I've already listened the newest development according to USP, and this is the conclusion. The reliability of any result of chemical analysis are dependent on some factor. The quality, the qualification and motivation of the analyst. Second, validity of all used equipment and software. The availability of authentic chemical standard, and at last validity of all used method. All methods should be first validated using the newest available guidance, because if we use, for example, not USP 44 but USP 32 is quite different. We should use the newest current pharmacopoeia. as I have described before, many methods were described for determining the accuracy and precision. So from USP, we have 2025, we have 2010, that is about 3-4 method. I do not know also what is the best method for determining the accuracy. USP not describe the best, but USP just describe the method. The validity of the reportable result of quality control laboratory should be confirmed using quality control sample. That's called internal quality control. If the quality control sample yielded out of specification, or result of analysis must be rejected. Maybe we need a revalidation. I recommended to report the analysis as mean plus minus confidence interval, prediction interval, or tolerance interval instead of mean plus minus RSD or variant. Because according to USP, the uncertainty of the method is covered if we use calculated using GI, PI, and DI. That is the software that I want to show you. You can download, free download is here. All of the calculation that I described in this presentation can be calculated in using this software, except the transfer of method. Except the transfer of method. If you want to use all of VX tool, anything, you can calculate using this software. And let's thank you for your nice attention. Have a great day. If you have a problem, maybe we can have a discussion. One can sorry because my English is very bad. I hope you can understand what I have presented today. Thank you, ladies and gentlemen. Good night.
Thank you, sir. Thank you so much. And no problem about the language. We were able to understand each and everything that you have conveyed. Thank you so much, sir. It was an amazing presentation, in fact. So participants, if you have any questions, you may ask us now, or you may put in the chat box also. Open to the questions.
Prof, good morning, Prof.
Good morning, sir.
Yeah, I would like to ask one question. Prof, actually, you mentioned that USP guidelines, for the statistical purpose for the method validation. So would you find any difference between ICH guidelines and USP guidelines? Usually we follow ICH guidelines, right, for method validation?
You mean ICH guidelines and USP guidelines?
Yes, ICH guidelines, yeah.
ICH guideline is published in, if I'm correct, if I'm not wrong, in 2005. But USP, some of the ICH guideline is used by the USP, as I show you.
Yeah, usually when we do for the publication purpose and method validation, we sincerely follow the ICH guidelines. So the ICH guideline chapters are updated, but the guidelines overall is updated. Is latest is 2005. So I think USP might have updated, might be updating, probably in advanced.
ICH guideline is okay, but it's I think it's some out of date. For the example, as I told you before, as I told in this presentation, it is better using combined evaluation between accuracy and precision. That's only described by USP, but not in ICH. USP is used ICH in chapter 2025, but USP describe many accuracy methods. For the example, in 2010, as I described.
Yeah. But in your presentation, you also mentioned about the number of samples. Usually, actually, means I have published a few papers in various journals, analytical work. Usually, I use only maximum 3 samples, n is equal to 3.
Yeah, it depends. It's up to you, but USP recommended at least for accuracy 3 level of concentration. 3 replication, so it's meant 9 for 1 day. So for the intermediate precision, we need 3 times 3 times 3, so it means at least 27 sample.
Okay. Okay, thank you, Prof. Thank you very much for your update. Thank
But
you.
it depends on us. This is only guardian of the
Because I'm afraid actually when we increase the number of samples, we are compromising the statistics. Actually, if your method is more specific and accurate, even n is equal to 3, you get the TI or CI or the percent RSD will be very less. The difference,
Yes.
you know, so usually we n is equal to 3 is ideal. But as you mentioned, no, different concentration when you use the 333, like, you know, number of samples you can increase, and probably you can enhance the statistics, you know, further to make it more specific. Yeah, thank you, Prof.
Okay, thank you.
Thank you so much, sir. Thank you, keynote speaker, Dr. Gunavan, and thank you to our session chair, Dr. Lokesh. So with this, we'll move on to our chief guest, Mr. Asif Mahmood. Yes, Dr. Asif Mahmood is the Chief Safety Officer of VP Global Clinical Safety and Pharmacovigilance at Medicago, Durham, North Carolina, United States. And he has vast and diverse leadership experience as a health service professional with significant accomplishments in all aspects of pharmacovigilance, clinical development, medical affairs, primary health care, and management of international health programs. Vast experience of dealing with various therapeutic areas, including devices and combination products, biosimilars, oncology, rare diseases, and many more, as well as managing PV quality, compliance, and operations. He is a self-directed professional with excellent communication, interpersonal, and problem-solving skills to build consciences and promote network. So I request our chief guest to deliver the welcome speech. Unfortunately, he has not been able to join us. So we'll be playing his welcome message, video message.
Welcome all of you. Good day. I would like to first of all welcome all of you to the 9th World Conference on Pharmaceutical Sciences and Drug Manufacturing 2021. I understand that participants have come or virtually attending from various different countries, and I hope that you will enjoy this as well. I apologize, I am unable to download directly on this Zoom meeting because it is too late night like here and in the online US. To the distinguished panel of speakers representing various organizations and institutions, I express my gratitude for your contribution in making this conference a reality. I strongly advocate the fostering of close relationship between the pharmaceutical industry, academia, and the regulators. I hope that all participants will take this opportunity to establish networking and build bridges rather than barriers in order to establish a common goal. Medicines and vaccines have become a very important and powerful tool, now more than ever in improving the health status of populations and in the long term for reducing the healthcare costs. As a safety physician, I am very aware of the fact that an ever-increasing number of diseases are successfully managed today, especially with the possibility of more effective, more reliable, and more robust preventive and curative treatment options. A study conducted by the Boss and team and published in the Pharmaceutical Research and Manufacturing of America journal showed that the evolution of clinical practice guidelines over the last 25 years have emphasized an increased role of drug therapies in healthcare. According to the study, factors which have contributed to this trends are the rising burden of disease, an earlier role for the use of vaccine and medicines. Many treatment regimens now consist of combination therapies for a single disease, condition and are often prescribed for extended maintenance use rather than for repeat acute care. Another factor which has led to the great use of medicines is the increasing number of chronic diseases involving the growing geriatric population. This is illustrated by increased prevalence of chronic diseases like cancers and previously underdiagnosed conditions such as Alzheimer's disease. Meeting the need for more effective drugs with better safety profile is a challenge to scientists and the research-based pharmaceutical industry. Scientists are working hard to tackle complex diseases with new approaches. Over the past 10 years, expanding knowledge of the human genome, insight into how diseases occur at genetic and molecular level, and the impact of cutting-edge new technologies have begun to change how medicines were discovered. New possibilities are opening up. Treatments based on the individual genetic makeup show greater promise to improve healthcare. Physicians today, in many cases, do not have the ability to predict whether an individual patient will respond to a specific treatment or not. Similarly, they may not know at the outset of treatment the likelihood of the individual patients developing any adverse reactions. Of course, there are instances when they do know. Personalized medicine would help physicians tailor the use of medicines for individual patients. All of us are aware of the fact that developing a new drug is a long and costly affair, while chances of successfully marketing a new drug is quite low. The estimates of the cost of developing a new drug are ever increasing every year. Pharmaceutical research companies are said to have contributed billions for the new drug research and development and ongoing, with billions being invested by Pharmaceutical Research and Manufacturing of America, which is number one in this state. Today, there is rapid growth in the research environment in the emerging economies such as China and India as well. There is also an increasing shift of R&D sites from Europe to Asia because of lucrative incentives such as tax reliefs and pioneer status. Pharmaceutical research and development contribute a major part of the research process to move new science from the laboratory to the bedside. Through industry efforts, many new vaccines, drugs, and devices have been developed and marketed. These drugs have led to striking decline in mortality from heart disease and strokes. And COVID-19 vaccines recently marketed by many different countries is a very good example of this. I am happy to note that this conference a wide range of subjects and be supervised. With that, I would like to wish you all a very fruitful and successful. Thank you very much.
Yes, thank you to our chief guest, Asif Mahmood. So next, we will be moving on to the technical sessions. Meanwhile, our keynote speakers join. So we'll be beginning now the pharmacology and toxicology sessions alone. The remaining sessions will be conducted in parallel rooms, which will be breaking out the rooms later. As of now, we'll be starting with the pharmacology and toxicology session. And the session chairs evaluating this particular session, I would like to welcome Professor Dr. Nehla E. Ismail, who is the council member from the Malaysian Academy of Pharmacy from Malaysia, and Dr. Bunta Venkata Subramania Lokesh, senior lecturer from University of Malaya from Malaysia. Hello, sir. How are you? Yes, so with both the session chairs' permission, we will be beginning the session 1, pharmacology and toxicology. I would like to know the availability of K. Annie Vijeta for her presentation. K. Annie Vijeta.
Yes, ma'am. I'm ready with my presentation.
Yes, welcome K. Annie Vijeta. So K. Annie Vijeta is from Jawaharlal Nehru Technology University from Hyderabad, India, and she will be discussing about the formulation development optimization and in vitro, ex vivo and in vivo characterization of self-nano-emulsifying drug delivery systems in ibrutinib. So you may begin.
Yes, ma'am.
Yes, for all the presenters, the allocated time is 10 minutes. Please make sure you complete your presentation within the allocated time. So you may begin.
1 second, ma'am. Share option is
Yes.
given to me, ma'am.
Yes.
Okay, ma'am. Is my PPT visible, ma'am?
Not yet. You have started sharing. Yes, it's visible now. You can put on slide mode and begin.
Good morning, everyone. I'm K. Annie Vijeta from Center for Pharmaceutical Sciences, JNTUH, India. And I'm doing my PhD under the guidance of Dr. M. Sunita Reddy, ma'am, Associate Professor, Center for Pharmaceutical Sciences, JNTUH. Before starting my presentation, I want to thank BioLeaks and APR for giving this opportunity to present the work in 9th World Conference on Pharmaceutical Science and Drug Manufacturing. Coming to my work, this is part of my PhD work. I have done formulation development, optimization, and in vitro, ex vivo, and in vivo characterization of self-nano-emulsifying drug delivery systems of ibrutinib. These are the contents of my work. So, I have chosen oral route of drug delivery, which is the mostly used method of administration, and I have preferred this. And most of the drugs which are released into the market and which are discovered newly are facing problems with solubility. Solubility is the major problem, which decreases the bioavailability of the drug. So the drugs which are to be manufactured and which are discovered, which belongs to any of the class biopharmaceutical classes. Class 1 is having high solubility and high permeability. Class 2 is having low solubility and high permeability. Class 3 is having high solubility and low permeability. Class 4 with low solubility as well as low permeability. The drug which I have chosen is belongs to class 2, which is having low solubility and high permeability. So to enhance the solubility of the drugs, I have chosen lipid-based drug delivery systems like self-emulsifying drug delivery system. Okay, these are nothing but these are pre-concentrates of emulsions. When it enters into the biological fluid, it gets converted to an emulsion. And it is an isotropic mixture of oils, surfactants, and co-surfactants. So the difference between sets, METs, and SNETs is just depends on the particle size or globule size. Okay, SNETs, the size of the globule is less than 100 nanometers, and for SMETs it is 100 to 250 nanometers, and for sets it is higher than 250. This is the literature review. I have started with collecting from literature review of Poughton, who has started the work on self-emulsifying drug delivery systems. And the aim and objective of my work is to formulate and evaluate self-emulsifying drug delivery of ibrutinib and to enhance the solubility and bioavailability. And my objectives are to develop and validate a HPLC method for ibrutinib and to perform solubility studies of drug in various excipients like oil, surfactants, and co-surfactants, and to determine drug excipient compatibility studies, and to find the nano-emulsion region by using pseudo-ternary phase diagrams, and to optimize the oil and S-mix combination by design of experiments by choosing central composite design, and to prepare set formulation and to perform evaluation studies like in vivo, ex vivo, in vitro evaluations, and these are the in vitro evaluation tests which are mentioned below. This is the drug profile of ibrutinib, and the marketed name of ibrutinib is Imbruvica. It is an anti-neoplastic drug, and the log P value is 3.97. These are the method development trials. So this is the scan spectrum of ibrutinib by HPLC, which is at 286.4. And at this 286.4, I have trialed various optimization conditions. And first I started with acetonitrile and water in 50:50 ratio. And later I increased the concentration of acetonitrile. And eighth one is taken as the optimized one, which has been accepted with a good peak, and the retention time was found to be 2.5. And this is the calibration curve of ibrutinib. I have followed a flow rate of 1 ml per minute, and the mobile phase is acetonitrile is to 0.1 normal orthophosphoric acid, which is the trial 8th of the last slide. And the diluent which I have used is acetonitrile, and the detector wavelength is 286 nanometers. And the column which I have used is C18 reverse phase column, Sunfa C18 5 microns one. And I have used HPLC water system, and the injection volume is 10 microliters, and the runtime maintained is 5 minutes. And at this conditions, I have constructed this, and the R-square value was found to be 0.996. And these are the chromatographic peaks of blank and ibrutinib. The retention time at 2.5 minutes. And validation parameters. These are the validation parameters for the developed method. All the validation parameters were passed the limits, and this is the concentration range which I have maintained, and this is the slope of the calculated one for the calibration curve, and these are the system suitability parameters where retention time is 2.5 and tiling factor was 0.42, which is acceptable, and theoretical plates and percent RST of the peak area all were in the acceptable limit. And these are my precision intraday and interday precision values, which should be below 2. The percent RST should be below 2, and it is below, all the values were below 2, which are acceptable. And limit of detection is 0.276 micrograms per ml, and limit of quantification is 0.837 micrograms per ml. And these are my accuracy limits. And after developing a suitable method and validation, I have went through solubility studies of ibrutinib to prepare self-emulsifying drug delivery systems. I need an oil, surfactant, and co-surfactant in which it is the maximum amount of drug is solubilized. I have taken those excipients, and here the first one, these are the various oils in which ibrutinib is solubilized, and the amount of ibrutinib was found to be highest in capriol 90, which is chosen for the formulation one, and in surfactant colifer EL was found to be highest, and in co-surfactant transcutol HP was found to be highest. So for the further study, to make the formulation, I have chosen capriol 90 as oil, in which 56.4 mg per ml of drug was solubilized, and in capriol colifer EL 39.5 mg per ml of drug was solubilized, and transcutol HP 65.7 mg per ml of drug was solubilized. Later, after taking this, I have gone for compatibility studies. So the 3 excipients where the solubility was highest, I have taken those capriol 90. The first, this is the drug one. Drug FTI spectra, this is the capriol 90, colifer EL, and transcutol HP, and this is the combination one, and the drug was solubilized in, the drug was compatible with all the 3 excipients. And later, after compatibility studies, I have done pseudo-ternary phase diagrams to check where nano emulsion is forming. So I have taken 1:2. Here I have just kept 3:1 and 4:1, but I have done with 1:1 and 2:1 also. But in those regions, nano emulsion was not formed. And coming to 3:1.
Excuse me, Ms. Vijeta, sorry to interrupt. You have another 2 minutes to conclude.
Okay, ma'am.
Yeah.
So in 3:1, 3.7 to 1.9 ratio, the nano emulsion was formed. And next slide, like here 4:1 ratio of S mix. Here also 3.7 to 1.9, the nano emulsion region was found. But I have taken 3.1 because the surfactant concentration which I choose was lesser one. So I've taken that for the further trials, and I've constructed design of experiment by using central composite design. And here by central composite design, 15 runs were run by taking block 1. And here run 2, 11 and 14 was found to be optimized because the response for this was good. That is droplet size and self-emulsification time was good. And these 3 were formulated by taking the ratios from the design of experiments, and later evaluation tests were done. For all the 3, the evaluation tests were passed.
Ms. Vijita, is there any problem? Hello.
Hello, Ms. Vijeta, are you there? Probably disconnected.
Ms. Vijeta? Okay. So with this, we will conclude her presentation then. Okay, thank you, Vijita, for her presentation. I think unfortunately she has disconnected, and we will not be able to give her the feedback, sir. So we have been joined here now by our keynote speaker. I would like to invite Shri Ashish Kumar, the Vice President of Zydus Group from Ahmedabad area, India, who have joined us here. Welcome, sir. Good morning. Thank you for joining.
Good morning. Good morning. Is it fine if I put my presentation?
Yes, sure, sir.
Okay.
Looking forward to it. Yes.
Are you able to see the screen?
Yes, sir. We are able to see the screen.
Okay. So good morning, everyone. I'm Ashish, I'm Vice President with the Zydus Group.
So, sorry to interrupt. So would you mind putting on slide mode?
Is it not on slide mode?
No, sir.
Okay. Is it there now?
Not yet.
It's here, it is showing that way.
Okay,
Sorry.
sir. No problem. You may continue. Maybe there's some internet issue. No problem. You may begin.
But are the slides changing? Can
Yes,
you...
the slides are visible.
Okay. And they're changing?
No, it's in the first slide itself. That's improving access to cancer.
Okay, they're not changing. Sorry.
No problem.
Can you see the disclosure or you can't see?
Yes, we saw the disclosure.
Okay, great. So then the slides are moving. So I'll start the presentation.
Okay, sure.
Yeah. So good morning, everyone. I am Ashish, and I take care of the oncology and ophthalmology business for Zydus based out of Ahmedabad. And the topic today, which I'll be speaking on, is improving access to cancer treatment, the role of biosimilars. These are my disclosures. The presentation doesn't intend to promote any brand or drug. And also, it is based on the national and international published information across the globe. So the reason for speaking on this topic is the huge impact this class of drugs have created on the cancer care across the world. And before we set into the impact of biosimilars, I will throw some light and share some information, the primary information on the biologics, what are biologics and where are they used and why biosimilars were developed to copy the biologics. So when we talk about the biologics, and my presentation would, apart from biologics, biosimilars, would also cover up what are the pathways across the world for the approval of biosimilars and the potential role of and impact of biosimilars and the challenges which various stakeholders across the globe see in accepting these biosimilars. So starting from what are biologics? So as many of us would know, a biologic drug is a product which is produced from living organism or contain the components of living organisms. And they include a wide variety of products which are derived from either humans, animals, microorganisms by using the biotechnology. And biologic drugs have had a wide facets. They have been used as vaccines, blood components, cells, genes, tissues, recombinant proteins. So that's the wide facets where the biologic drugs have found its usage. And they may contain proteins that control the action of other proteins and cellular processes, genes that control production of vital proteins, modified human hormones, or cells that produce substances that suppress or activate the components of the immune system. So that is why these drugs become important, because they are directly dealing with our immune modification, suppressing or enhancing the immune systems. And they're also sometimes referred to as biologic response modifier because they change the manner of operations of natural biologic intracellular and the cellular action. So that's the action of biologics. And why biologics become so important in cancer, though, as I said, the wide facet of biologics have been used across therapy areas, but cancer becomes extremely important area for biologic drugs is that cancer, as we know, is a result of the malfunction of the immune system, where our own cells start to divide in an unproliferated way. And these biologic therapies can definitely repair, stimulate, or enhance the immune responses. Traditionally, we have seen that in the cancer treatments, chemotherapic drugs have been used, but then there also have been challenges we have faced with chemotherapeutic drug because of the side effects, because chemotherapeutic drugs are not targeted. Chemotherapy drugs, while acts on the fast dividing cells, we also have our normal cells like blood, hair, which are also fast dividing cells. So they tend to produce a lot of side effects apart from their cancer killing actions. So that is why biologics become even more important, because they are more targeted, they are specific, and they can also have synergistic effect with the chemotherapy drugs. And as I shared, that these drugs also can be immune modifiers, which can help on the outcomes of the cancer treatment. So these class of drugs become extremely important and a very special class of drugs to treat the cancer. Now, how can the synergy of biologics with the chemotherapy, which has been the age-old treatment for cancer, be there? Now, chemotherapy, as I said, would act upon all the fast-dividing cells. They're not specific, but biologics, as we know, are specific to the targets, to the cancer cells. So the new way or the new future is, can we combine the biologics with chemotherapy so that when the drugs enter the body, the biologics, rather than just being the target cancer killing cell, also become the carriers of these chemotherapeutic drugs to the target site, so that the side effects which the chemotherapy drugs are doing to various normal cell can be minimized or reduced to a very low level. Because if the biologics become carrier of chemotherapy drugs and take them to the site of cancer, there would be a huge synergy. While biologics will also act on the immune system on the specific target, the chemotherapy drugs would also act on the tumor cells. So that's the synergy building up slowly, And that's how the biologics and chemotherapy become the cornerstone for the all future developments of the cancer treatment. Now, which are different types of biologic agents which are presently used in the cancer treatment? So broadly, they can be divided into 4 different types of biologic agents in the cancer treatment. They can either be cytokines. Cytokines, as we know, are proteins that enhance or suppress the immune system by either attacking the foreign infected or damaged cells or signaling the other cells to attack. The most common or the most used or probably the drugs which have amongst the biologics found most impacts have been MABs, which are monoclonal antibodies. And why they have found so much usage in the cancer treatment is because they genetically engineer the antibodies, and then these are directed against the antigens on the surface of tumor cells. So if we know that what is the cause of the cancer, suppose we know that it's a diffuse large B-cell lymphoma and it is CD20 directed, we can create a monoclonal antibody, which is rituximab, which will go directly to this and probably target these cells and would not have much of the other side effects. We also have interferons, which we know are proteins made and released in the response to the presence of pathogens, which include virus, bacteria, and tumor cells. And they allow communication between cells to trigger the immune system to destroy the invading cells. We also have an important class of biologics which are called growth factors. And these are important because so many chemotherapeutic agents are being used, which can, as I said, also act on the normal dividing cells, which includes our blood. So when the chemotherapy is being given or certain other agents are being given and our blood components also get impacted, we also need boosters, which are these growth factors, to increase our blood counts. So these kind of biologics are also very, very important as a supportive care, especially in the treatment of cancer. But then comes the most important agenda, and that's the cost of biologics for the cancer therapy. If we look at the cost of simple drugs versus the biologics, there is a huge difference. Why? Because of the complexity of the development process of the biologics, extremely sensitive and difficult manufacturing process. As we are dealing with the live organism, we have to be extremely careful. Even the preclinical studies are very, very important in these class of drugs. And a single biologics to develop across the world can cost, on an average, more than $1 billion drug. And then I am talking today from India, and I must tell you that if we have over 1,000 companies in India, only 10 to 15 companies have turnover more than $1 billion. So we are talking a development of a drug taking more than $1 billion, and then the revenues of so many companies in a country like India is not $1 billion. So that's the reason why the cost of biologics also become very, very high when they get into the market to serve the patients. So that's why it becomes extremely prohibitive. And the reason is the development cost, which is more than $1 billion on an average. And that is what is happening across the world. We have been hearing about the 80-20 rule, that the 20% of the drugs would have profound impact on the 80% of the population, but that's not the case with the biologics. Biologics, rather, we say that has a 42 rule. And what is the 42 rule? The 40% of the spend across the world has been happening on the biologics in the last decade or so. This is the data from one of the reports in 2017. But when the 40% of the drug spend is on the biologics, only 2% have access to the biologic drugs. So, and that again is because of the high cost involved in these drugs. So there has to be something done, that when you're spending 40% of your spend on the biologics development, why only 2% have the access to it? So that was the area which was actually concerning the whole world 2 decades back. And over a period of time, we have found a solution to that, which I will discuss in the subsequent slide. But then this is the comparison between the biologics and the small molecule spend on the drug development. And if you see from 2013 to 2017, and in these 5 years, if you see, the drug spend on the biologics have been increasing year on year from $76 billion to $120 billion in just 5 years, while for the smaller molecules used in the other treatment, the cost has been more or less same across these 5 years. So that is what is a worry factor, that if the drug development cost of biologics is going to be high, as we found and develop newer drugs, we also have to find a way to make them more accessible. So what is the reason behind the huge gap in the cost between developing a biologic drug and developing a simple drug, or probably a general drug, which are not derived from a living organisms? And the reason has been multifold. And if we have to name few, which are the most prominent reasons behind this wide gap, then the biggest reason is the complexity of the biologic science, because unlike the general drug, we are here dealing with the live organism. And biologics, as we know, is a extremely difficult area due to its complex structure. We are talking about lakhs of amino acid sequence here, the protein structures, and then they coming together and targeting a particular cell, so inside the human body. So that's why the preclinical work also becomes extremely complex, while it is extremely easier for to produce the smaller molecules. Even when the innovator drugs becomes off patent, we see that the smaller molecules, just on the bioequivalence studies, can be approved in various countries across the globe and can be used as a replacement of the smaller molecules. But that's not the case with the biologics. Because of this complexity of the structure, even the biosimilars, which in the easy terms are called as generics of biologics, have to go through a complete process of development as a new drug is being developed. So that's the huge complexity we are facing with the biologics, which is behind the reason as a gap in the huge cost. And there are very less players if we consider the biologics development or the biosimilar development across the world, which again is the reason for the higher cost. More are the players, and as we know, the economy drives the better competition to decrease the price. So that again is a huge gap in terms of number of players who are into the biologics development. If we look at the, if we look at the approvals which are happening for biologics versus the smaller molecule, unfortunately, while I said that the 40% of the spend, and today when we talk in 2021, almost 53% of the word spend is going on the biologics. But then if we look at the cost, the average production cost, a smaller molecule might cost $5 to probably develop. This is a report from a Boston consulting group. While to develop a biologic, it might cost us $60. So that's the difference, $55 difference you can see for developing a single, for producing a drug, not developing a drug. And if we look at, but despite this so much of spend, if we look at number of drugs getting approved across the world, especially on the FDA approvals, we see that it is not that despite a spend of 50%, that's half of the total spend across the world on biologics, the number of biologics getting approved is not 50%. It is actually only 20-25% of the total drugs approved across the world. So while it is complex, it is difficult, the number of biologics also getting approved is also limited because of the acceptance of various regulators across the world. So these are other gaps across the world which also need to be addressed over a period of time. So coming to the topic which is of importance today, that while this cost of biologics have been so high, and it has been addressed by the development of biosimilars, but then to see the impact of biosimilars on the access of treatment, we must also understand what are biosimilars. So biosimilar is a term which is becoming more common in the last 2 decades across the globe, rather 2.5 decades, I should say. And the FDA initially had started approving biosimilars as products and biologic products, which are large and generally complex molecules. They also are made from living organisms, and they are similar to the biologic products, which are reference product based on which they are made in their safety and efficacy. So they are closer to the biologic products, that's the reference product, on the purity, molecular structure, and also the bioactivity. So there are various regulatory pathways where you have to prove that your product is a closer product to the biologic product, which is a reference product. And that is how the biosimilars started getting approved across the world. And through them is where the access to the treatment, and today we're talking about the cancer treatment, started becoming more and more accessible. So if we have to say a difference between a biologic and a biosimilar, then the guidelines say that the biosimilar which is approved or gets approval has no clinical meaningful difference from a reference product, whether it's the pharmacokinetic, the immunogenicity assessment, or the clinical studies which prove that they are similar on the safety and efficacy. Biosimilar on the on a difference level to reference product doesn't have much difference. Though it should not be called like the generic product, exact replica of the reference product. That's why the term was coined as biosimilar or highly similar to a reference product. And these are approved after a rigorous evaluation and the testing procedure by the organization who developed these products. And while FDA have its own way of approval, if we talk about India or Europe biosimilar guidelines, they also have their own ways of approving a biosimilar against a reference product. So when we say biosimilars are similar to the biologics, is there a difference also on that? So the answer to that is there might not be a meaningful clinical difference on the product. There might be, but what we are probably concerned about is the safety and efficacy, and the biosimilars are only approved when the safety and efficacy of biosimilars are found to be similar to the biologics. Otherwise, the biosimilars don't get the approval. And this is a cartoon which probably suggests the pathway of getting a biosimilar approved. And now we have so many organizations across the world which are developing biosimilars, but the process more or less remains same, which you see in this cartoon. First of all, the organizations the pharmaceutical companies which develop the biosimilars have to do a reverse engineering of the biologic product for which they want to develop the biosimilar, and through various techniques like mass spectrometry, the amino acid sequence and the protein structures, and various chemical modifications are found out. And these are then probably used in the cellular factories where the cell cultures are developed to probably develop the similar kind of amino acid sequence and the protein structures. Since again, we are dealing with live organisms here and it's about biotechnology, the processes at each step are being validated and the developers have to be extremely cautious of the development process. Once the cell lines are developed, the structures are developed, there are various assays which are used to probably tell whether the biosimilars have the similar sequence as the biologics. Once those assays are developed and those biosimilar structures are being tested, both the primary and the secondary structure and even the tertiary structure, then it comes out to probably put this drug which would target a specific antibody in the or the immune response in the human body. Then the next step is to find a stable formulation, because as we know that each of the biologics would have its own patent, and when these biosimilars are developed and they develop the similar cell lines which have the same sequence, the next step is to probably develop the stable formulations. And once the stable formulations for these biologics are being prepared, Then comes what we do for all the other drug development, which is the clinical confirmation through the phase 1 to phase 3 studies, the phase 1 trial to show the drug is safe, and then the phase 3 trial to show that it has an efficacy similar to that of the reference product. And once that is done, the phase 3 is done, the biosimilars are being submitted to the regulators for the review. And based on the regulators' guidelines across various countries, based on the safety, efficacy, and the preclinical and the clinical data, the regulator choose to approve or probably reject the biosimilar in their countries. And as I said, regulators have their various path of accepting the data, but what remains same across the world is that the analytical studies should prove the similar structure and function of biosimilars. The animal studies should have the assessment of toxicity, and then the PK/PD and the immunogenicity has to be also done before we showcase significant phase 3 data on the similar safety and efficacy. So these components remain same across the globe in the various biosimilar guidelines for approving the biosimilar. Now, coming to the development, what's the difference between developing a reference biologic and the biosimilars? The only major difference I would say is that the biosimilar starts a journey from the reverse engineering, while for a reference biologic product, you have to first find out a product which would be effective and will have a therapeutic effect on the body. So that's a lot of research which a biologic drug has to do, and biosimilar only starts by copying that biologic. So that is why the drug development probably is 60-70% less than developing. When we talked about a $1.2 billion for a biologic, it might come down to $300 to $400 million or even less for a biosimilar product. US also coined a term which is beyond biosimilar, and there are very few or hardly any which have come under that landscape, and it is called as interchangeable product. So when on the biologics we say interchangeable, then also apart from various studies which are done to prove that you are biosimilar, there is also a study which is done on the study population, which are patients, and there the interchangeability between the reference product, the biologic and biosimilar is also studies, and more than 3 switches are done to assure that the biologics and biosimilars can be interchanged in the clinical practice. And once you do that, then you can also apply that your biosimilar is interchangeable to the reference product. So that is something where most of the biosimilars have not reached today, but probably that's the future in development of biosimilars. And when we talk about the number of biosimilars developed across the world, India today takes the lead. This is the data from 2019, but if I talk about December 21, in India, over 90 biosimilars have been approved. EU has close to 70 biosimilars approved today. And then US was late in approving biosimilars because US approved the first biosimilar in 2015, but now US is gearing up for a lot of biosimilars launches in the coming years. And if we look at the spend, you see oncology spending as a specialty, most on the biologics for the drug development. And the reason again is oncology needs, the cancer care needs the targeted drugs rather than non-targeted drugs in the cancer treatment. So what's the impact? So while the biosimilars are being developed so much across the world, and we saw that so much of spend is also going on developing biosimilar, but what is the impact? Is it really improving the access of patients or the health regulators across the world, or not so? So the biosimilars, as we know, are priced at least 20 to 30% lower than the reference product. And they have resulted in high cost savings because of the lower R&D costs as compared to biologics across the world. While the lower price biosimilar also tend to have a downward effect on the pricing of the reference biologics because of their availability. Even the reference biologics, once the biosimilar comes in, tends to reduce pricing. but we also, there are a lot of studies or probably data collected across the world that what has been the impact of biosimilar availability in different parts of the world. This is one data which I am showing you from India for a drug called trastuzumab. Till 2015, trastuzumab is a drug which is used, as we know, for HER2 positive breast cancer patients. And the data of trastuzumab, which is now a gold standard in the HER2 positive drug, is so promising that most patients should get, or all patients should, HER2 positive patients should get access to trastuzumab, if not the higher anti-HER2 agents. Until 2015, out of when the biosimilar was approved in India of trastuzumab, out of 100, only 25 patients had access to trastuzumab in India. But if we talk about 2020, with the access of biosimilars in India, almost half of the patients today get access to trastuzumab, and this is improving every year as the competition is increasing and the price of biosimilars is coming down. So that is what happens to the society, that is what happens to the patients. The health regulators pay less, and then the patients get the drug which they deserve. So that's what the biosimilars tend to do, and the increase in the penetration. This is an article from one of the reference which studied the economic impact of biosimilars in oncology and hematology, again studying trastuzumab, as I said earlier, and then also rituximab was studied in this. And it was found that the biosimilars of rituximab and trastuzumab are cost-effective treatments for both the DLBCL for rituximab and the HER2 positive breast cancer for trastuzumab, saving huge amount of money if the biosimilars are used in place of the biologics. A study from France, which probably studied the economic analysis if they switch the pegfilgrastim with the pegfilgrastim biosimilars, and it was found that over 5 years, almost 28 million euros can be saved if they use pegfilgrastim, which is just a drug for supportive care for treating the chemotherapy-induced neutropenia. And just on replacing a biologic drug with biosimilars, that's the impact the economy can have, almost saving 30 million euros over a period of 5 years. So, and the conclusion were drawn that the introduction of pegfilgrastim biosimilar will help to reduce cost and alleviate some of the financial pressure on the French healthcare system. So that is what the biosimilars can do, not just improve penetration to the patient, but also alleviate the financial pressure on the various healthcare system across the world. One more study done in Italy, which studied that what is the cost of biologics on the healthcare system, and what would happen if we replace that with the biosimilars. And it was found that if the healthcare cost of biologics can be replaced by biosimilars, in 2017, they projected that almost 32 billion euros just by replacing biosimilar can be saved by the Italian healthcare system or the national health system of Italy. So, that is what the biosimilars can have an impact. Last, to represent the impact of biosimilars on the access, a study done in India on the DLBCL patients. And the study clearly showcased that once the rituximab biosimilar was introduced, rituximab again is one of the gold standard treatment for DLBCL, which is a curative cancer, where patients come with huge masses, and after few cycles of R-CHOP, R is rituximab here, tend to have very positive outcomes, and most of the patients are disease-free after 6 to 8 months of receiving the R-CHOP regimen. And before rituximab biosimilar was available in India, there were hardly patients, less than 10% of the patients who had access to rituximab. But once the biosimilar started coming into India through the various pharmaceutical company, almost 90% of the patients see the impact. 9 out of 10 patients had the access to the rituximab. Coming to the last part of my presentation, that what are the while we have seen the positive side of the biosimilar impact, that it improves the access significantly, so many patients get access, the healthcare burden comes down. But what are the challenges which are there related to the biosimilar adoption and use? Now, the various challenges in nutshell, which remains to use biosimilar is, as of now, different regulators have different approval process for biosimilars. Some tend to be more stringent, some tend to be less stringent, but more or less now the biosimilar development guidelines across the regulators are finding a coherence, and we hope that over a period of time we will have more robust approval process, which is definitely there in many countries today. Also, the healthcare professionals, there is a confusion on interchangeability, whether they can interchange the biosimilars with the reference biologics. So the acceptance at times also is less from the healthcare professional, and also on the patient front, which probably there's a huge difference in the cost, and the patients also find themselves on a confusing side, that whether to pay a hefty amount for a reference biologic or probably to choose if they are out of pocket kind of a scenario, like various developing nations where the patients pay out of the pockets. These are different challenges which have the impact on the uptake of biosimilars. But then it is being clearly showcased in the last 1 decade that once the biosimilar starts to come in, there is a very, very healthy impact on the ecosystem, which is the society, because society tends to pay less. Society saves money, the healthcare system saves money, the overall cost of the drug comes down, and there is a huge impact on the overall treatment outcomes also for a particular disease where the biosimilars get into the market. So on the sustainability of the biosimilar sustainability improves definitely the patient access and the physician prescription choice of safe and high quality biological medicine. But when we look at the framework, we also have to be mindful that while patient access is important and the healthcare budgets are important, so is the importance of the development of quality, safe and efficacious biosimilars as well. So this framework has to be sustainable only when there is an improvement, not just on the cost and the probably outcomes, but also on developing the quality biosimilars. So that is probably a nutshell of what I presented till now, that while there are incentives on having more patients on the drug by developing biosimilars, the access gets significantly improved with the level of competition which goes up when more biosimilars come in. But in no way the safety and the quality of the biosimilars, that is what the regulators have to ensure, gets compromised at any time. So coming to the summary of my presentation, it is now clear that over the last few years, biosimilars have generated great interest worldwide as effective alternatives to biologics. Biosimilars have improved significant access to costly biologics across the globe and brought down the healthcare burden significantly. The term biogenerics is also used occasionally, but should be avoided because it may imply that biosimilars are integral to the original compounds, as in the case of generic version of small molecular drugs. And although the purpose of biosimilar is to provide accessible treatment option, however, the results may vary because of the different development process and the different approval process. And the switching between the innovator and biosimilar still remains an area to be addressed in various countries. And now it's the responsibility of the regulators to train their HCPs and patients to increase the adoption rate of biosimilars to help meet its purpose to provide affordable medicine. And I will end my presentation by saying a great quote from Sarah Gideon, that every single one of us deserves access to quality, affordable healthcare medicine. And while biosimilars have showcased that, the regulators, HCPs, patients, physicians across the world have to come together to ensure that the right processes and the right medicines keep coming out, by which are definitely improving the outcomes of the patient. So with that, thank you so much for this opportunity, and over to you.
Thank you so much, sir, for your amazing keynote session. Thank you so much. You have given us a lot of information on biosimilars, so that was really informative, and thank you so much for joining us at the 9th World Conference on Pharmaceutical Science and Drug Manufacturing. So any participants have any questions, you may put it in the chat box or you may interact with the keynote speaker. Anything that you would like to ask regarding the session that he has provided us?
Good afternoon, Mr. Ashish. Mr. Ashish, I just listened to your talk. It was a nice talk, there is a saying, it is easy to say than, achieve difficulty, so I mean to say when biosimilars are coming into market and achieve, I see a layered model between business model and also the what do you call development, because development is deprived because of the business model, do you think, or development is deprived because of the less number of products being approved by the regulatory? Because is there any contradiction between the regulatory and also the business model, or how do you think? What do you say actually on this?
I would say, sorry. Can I respond to that?
Yeah, please.
So I would definitely say that it may not be contradicted, but then the development process in itself for biologics is so complicated, and there is so much of unknown which comes in the journey, because you start with a cell line from which you probably keep developing the drug for years and years. And even if you change a cell line, it is being considered as a new product being developed. And you have to deal with various aseptic conditions as well. So I think, yes, there is a caution. Unlike the smaller molecules, that once you have developed the quality parameters of the same, you would be able to reproduce it same batch by batch, years on years, as many years you want to produce the drug. But biologics, when you deal with, even there are batch to batch variations, right? And how much to accept, how much to reject, I think the word is dealing with a lot of newness in the development of biologics. It's a learning phase for us, I would say. And that's why the regulators are also very cautious on approving the or accepting the data of the biologics, which is in the right space. And once we learn more, once I think few decades from now, when we probably talk, we would know much more about the biologics and the biosimilar, and the regulators would also be in a more confident space. WHO very recently drafted a policy on the biologics and biosimilars, where even they are saying that maybe the phase 3 study might not be that important when the biosimilars gets approved, but maybe the preclinical data and the primary secondary structures and the PKPD data is more important. So I think the word is learning and probably evolving. And with every passing year and a decade from now, we would be in a better condition, and you would probably not see any contradiction between the regulators and developers.
Yeah, absolutely. I mean the variation is very actually vastly changing, statistically, if you say, you're right, actually, because when you produce the quality products, but accessibility of available quality products to the patients would have been improved, right? Why the regulatory people are retarding in the situation where to release? Because you mentioned in your presentation, it has been improved to 50% or something like that. Mostly, I mean, why can't it be 100%? Because I can say, my mother also expired recently with cancer, because I can say that why it is happening in India, because if you see in USA, the products have been, of course, even the enormous cost of the maps and enormous cost of this treatment, undergoing. But however, as you said, it can be still improved with the quality products which are approved by the regulatory by biosimilars. And it's nice talk, actually. Nice meeting you as well, and it was a nice talk from you. Thank you so much.
Thank you for the opportunity.
Thank you, sir. Thank you, both of you. So moving on to next keynote speaker. Before that, I would request everyone to enable your camera for a group photograph. Yes. So all ready for the big smile and photograph? Yes. So 1, 2, 3. Yes, thank you all. Thank you so much for your time. Yes, so next I would like to invite our next keynote speaker who has joined here. Ms. Gail Jaron, the co-founder and CEO of refineresourcing.com from Paris. Gail Jaron was a curious and entrepreneurial woman who appreciates challenges to cope with. And she has more than 16 years of experience in pharmaceutical industry and more than 10 years of experience in distribution and fine chemicals. Welcome, Ms. Jaron.
Good day, everyone. Can you hear me well?
Yes, yes. Thank you so much for joining us. We are eagerly looking forward to your keynote talk.
Right away. Let me share my screen. And please let me know if you can see it.
Yes, we can see it.
Yes. Thank you. First of all, thank you for inviting me here. I'm very much honored to be part of this very interesting and very technical event. I am, however, here today, not necessarily to go through studies or technical issues. The purpose of this presentation is really to bring light onto a still underestimated part of our pharmaceutical industry and see what can be improved. And I have chosen to talk about why should supplier compliance management dive into a new era. So it might sound like everyone knows very well what we are talking about. However, there's still very much room for improvement. So if we talk about innovation, if we talk about new ideas that come out to or come to the pharmaceutical industry, we can talk about blockchain, for instance, blockchain used in logistics to secure, blockchain used to secure also safety of the drugs that come to the market. We can also talk about Industry 4.0. Industry 4.0 is still at the very beginning in the industry here, but is a source of huge improvements in terms of deliverable efficiencies, time to market, and also, of course, used for monitoring what is happening. IoT, Internet of Things, another nice word, whereby we're just about to start it, with telemedicine, trying to find out new applications in the pharmaceutical world with those various objects that are connected. We have then big data, another word that any of us encounter on a daily basis. However, big data can't really exist without data, can it? Big data issues are coming up whenever we're talking about trends, trying to identify them, see how various trends are interrelated. So there is still a lot of room to be taken here in the pharmaceutical industry. We're very much at the very beginning of all of this. Artificial intelligence, I would say the same. We're trying to use, artificial intelligence to enhance our own capabilities. In surgery, that is pretty much becoming a common space, but there are, again, a lot of other areas in the pharmaceutical industry where this could be deployed. And finally, another nice word that we know is this, in production specifically, are these digital twins. So digital twins are reproducing digitally a factory that not necessarily exists today. This is very much used, for example, in the automotive industry, just to cut down the build, the time that is necessary to build up that site, to enhance the training of people, to enhance also the construction itself and the plans. So all this is very great. It really looks promising. It actually makes me personally excited about what all this could bring to the pharmaceutical industry. But this cannot be happening if we don't have robust processes. And so, processes can be various. We're great at building up processes in the industry. But what is the coherence between what is now and what it is that everybody is talking about, in this recent era? So all those nice buzzwords I was talking about earlier. Having this, I think everybody would love that, right? We would be so far ahead. We would be a more of a proactive industry than a reactive industry. But reality looks much more like this in the daily life of everyone. And so how can how can we make both, so current and what the future is going towards, coherent? Can we leapfrog would be my next question. Well, the answer to me, and I'm open to debate, obviously, is pretty much unlikely, because to access this type of dream scenario, to be able to use all those technologies I was just presenting earlier, we need to have data. We need to have digitized data, and digitized data from the bottom of whatever the industry is building on, right? And this is currently still not the case. And that is why I would like to focus on suppliers and have a look on why is it so important to work at and to look at and to see how can we bring this to the next level in order to enhance the rest. So first of all, stakes are high. We're talking about sales, obviously. Sales are possible because we have production, and production is possible because we have supplies, which brings us to say, in a parallel way, patient safety is what is paramount over there. Product quality is dependent of the production. So we can produce and we can sell just because we have product quality. And if we have product quality, we achieve patient safety, right? And at the very bottom of that, we have the supplier risk assessment. Everyone has supplier risk assessments. Everyone does it. Yet it is still not there. It is not at the same level that it should be, specifically if we would like to move forward in the digital era, excuse me. And so how does this translate? Well, patient safety can be monitored through pharmacovigilance, product quality through product and batch release processes, right? And the supplier risk assessment is simply put only about ongoing supplier management and how do we deal with that within every of our companies. So you see, stakes are high. And as the pyramid is showing, pretty much relying on suppliers. And that is the reason why I would like currently to focus on those suppliers. Currently, this basis, and I probably say it provocatively, but it is fragile. Ongoing supplier management currently focuses on mostly risky suppliers, and everything which is also part of that supply chain, everything else that is not necessarily risky or critical is basically put aside. And that has a number of explanations. One of those are obviously also increasing regulatory constraints, which make it pretty much impossible in the way that it is dealt with today to cover all the supply chains. Meanwhile, every industrial company is asked and requested to ensure product safety, to ensure great pharmacovigilance, etc. So we're pretty much putting focus on the top of the pyramid, thinking it is going to cascade down. However, it cascades down very, very difficult. So you have now understood suppliers are critical, and what we want is to avoid this card pyramid at any cost. What we have to keep thinking or keep in mind is that supply chain is not necessarily just the supply chain of that drug. It concerns every single level or step in that supply chain. And therefore, what market authorization holders have as a supply chain, most of the cases starts and finishes at level 1, exception APIs, obviously. But then the suppliers of that market authorization holders have themselves suppliers, and therefore we're touching to a base which can be fragile. Probably some of our participants today have experienced this during the whole pandemic crisis as well, whereby we kind of very much see the limits of the organization as it is today. So if we want to kind of avoid that card pyramid, well, it looks like we should solve the way that we're doing or dealing with suppliers today. Innovation is not only a matter of technological innovation, which are the buzzwords I was talking at the very beginning of the presentation. Innovation comes also and very often from processes. And therefore, I would invite you today to try to think about what can be done differently in the processes that you have in place with those suppliers to solve this part of the problem. So again, our suppliers seem to be the starting point of our activities, but why is it so? Simply because they are providing us with goods and services that enable us to run the activity. Again, back to the pyramid earlier, if we don't have suppliers, we can't produce, and if we can't produce, we can't sell. If we can't sell, patients can't get their drugs. The compliance of those suppliers is measured against requirements, which can be imposed by regulators or even chosen. Most of the times it is a combination thereof. And supplier efficient management also means that whenever there is a problem, whatever nature of that problem can be, this has to be resolved in an efficient manner. And so that is the reason why we have to kind of start thinking, how can we do this or do things differently? So I propose to think about processes to tackle, which in a sense evolve around Annex 16 of the GMPs and more because you can actually go further, much further, and think further of those boundaries which are tackled by the Annex 16. I would not go into the detail of that Annex 16. However, I would like just to pick up the main processes which are interesting for this particular presentation from that annex. What are we talking about? We talk about supplier qualification. What is it that your company is currently doing to make supplier qualification efficient? Specifically when you need to open up to second sources of raw materials, for example, APIs or excipients or whatever. How do you deal with this in order to be able to, as quickly, as efficiently possible, to go through that process, which is complex, which is long, but if every step of that process are put next to each other, then what do you do to shrink that time? Second part of it would be the supplier ongoing assessment. This is a big chunk. This is probably the most imposing or most resource-taken part of a supplier management, because it goes and evolves around monitoring the quality of the product that is delivered, logistic performance. So is it delivered on time in full every time you have been ordering that? Around commercial aspects like price or commercial relationship, availability, responsiveness, etc. And quality management systems or technical and quality agreements everyone is putting in place, supplier questionnaires, and so forth. This is probably, as I said earlier, the biggest part where improvements improvements can be achieved. But obviously, supplier ongoing assessment is dependent on the risk assessment, but is also a lot evolving around how do you maintain that relationship with your suppliers, and how are you able to update the data and the documents and, well, broadly said, the information that you have to process from them. Again, not necessarily just looking at the most critical or the most risky ones. You today, if you want to build up and, you know, go to whatever I said, the big buzzwords earlier, you have to have solid data from the basis. So how do you do it to cover all of your supply chain? Annex 16 also says we have to deal with deviations and claims. There are multiple tools that exist, and we will go slightly more into detail later on. But this is also something that, you know, still in a number of companies today worldwide, this is dealt with on a more or less paper-based basis. So how can we do, what can we do to make this much more efficient? And lastly, but not, least, batch release. So supplier qualification, ongoing assessment, the monitoring of non-conformities or deviations, all this goes down or enables the batch release. Makes sense, right? So what are the currently what I call supportive tools? So what do people use in various companies? And I'm talking from huge international companies to down to the very small ones. There is still a lot of the red ones, manual processes, manual stuff, manual information, which by nature means loss of information, means sources of error. It means losing time afterwards to kind of try to avoid these. This simply can't be done like that on a 21st century. You would certainly agree with me. There are some tools that are digital currently and that are sometimes, not all the times, implemented. The ERP is there, that is true, but not every company is using a quality management software, system software. Not every company thinks of using a document management system, for example, or even an electronic batch record system. So there are a lot of tools out there currently that exist, whereby you should be improving these and progressively remove those red circles over there. So if we take again those 4 processes which are listed in the Annex 16, I would encourage to think about how all the 3 other processes could be tackled, and how can you digitize all that data so that you can leverage on that data to build up more data, to use artificial intelligence and so on later on. So there are 3: supplier qualification, supplier ongoing assessment, deviations and claims. How do you do and make it all work together? So there are a number of paths, I could say, that I can suggest. And this is, of course, very much dependent on the company that you are working for or working with. But these are the ways probably to think about that, so that everything is efficient, everything works like in a network, because a supply chain is a network, isn't it? So the start would be to harmonize supplier quality data. Harmonize meaning there are loads of different various information within the company that are treated. They may differ from site 1, facility 1 to facility 2. They may even differ from department 1. So for example, I don't know, R&D department to the purchasing departments to the QA department. So let's try to harmonize that data. What are we expecting from such and such category of suppliers? What are the types of documents that you will be collecting and treating and updating at the end of the day? How to make it consistent for all the sites or across the sites of your companies. So that's the step number one. Step number two would be to find a system that can centralize all this. It is not seldom to have companies that have, you know, 3 various sites with 2 different, for example, ERP systems. So this is just because of the legacy or history. In case of mergers or acquisitions, for example, that is typically the case. Yes, you buy another site, you inherit whatever it is over there. So centralize or find a way to centralize that data. It can be to spread the same ERP all over the place, all over the sites. That can be one thing, but this is very, very tedious and very heavy as we know it. But it also can be to try to find another tool, a digital tool or a software that is, so to say, agnostic, so that can communicate and interact with all of the others, without disturbing the activities too much of existing systems. And so also the operators. It can also be to reflect, as I was a little bit saying earlier, how do you interact with all of your supply chain? This is going to be, it was just saying recording in progress. So where was I? Yeah, so it was at the whole supply chain. How do we deal with all that? Again, it must cover regulators, specifically in Europe, are now increasingly hitting on that point that whoever has market authorization on that territory is supposed to know what is happening in its supply chain. And also, can you today say, okay, for this particular product that I'm selling, this is the supply chain I have? That might be a question that is asked by an inspector, for example. Fourth part would be start with low added value processes, related to the qualification and ongoing assessment, etc. That can be, for example, focusing on supplier documentation. How can we deal with that? That is what I call a low added value, because it has to be done. You can't really do anything without that. But yet still a lot of, actually proportionally to the added value, it takes a lot of resources internally. So that is one of the suggestions I can make over here. But there are others certainly to be considered within your companies. And also this updating has to be done, but everyone is losing just enormous time on that. So starting with added value processes, once we have harmonized the data, once we have centralized it, once we are able to apply it to every single part of that supply chain, and we have tackled with this very cumbersome low added value processes, we can now then deploy also to other activities of that Annex 16, right? That could be, for example, the regular assessment schemes, the questionnaires, the audits, the claims. So pretty much, as everyone likes it in those diagrams, a little nice circle whereby you do all this circle for every of those activities. So you would, again, start by questioning yourself. For example, for audits, okay, how do I harmonize my approach for audits? Do all of my auditors approach the audits in the same way? What type of information do we get out of it, etc., etc.? Centralize that, make it available to anyone in the company who has to have access. doing this will actually remove a lot of those Excel sheets and emails that you have seen earlier, make it traceable, which is important because you can't really have data, actionable data without traceability. And if you have data, you can have traceability. I mean, if you have digital data, you can have traceability. Don't misunderstand me. And so all this little circle, just to repeat, for all of those activities that relate to supplier management. I hope this makes sense. And if you do this, if you achieve to do this within your companies, you would get unprecedented knowledge as well about your suppliers, upon which you will be able to leverage to take informed decisions in production, but also in how do you want to drive your business forward. It could actually also have impact on your business models. Who knows? So one of my last slides would be, because I'm already running out of time, when those foundations are solidified, we go from a card house to actually a solid pyramid, to which we will be able to add much more easily all of those components, or any of those components that we were talking at the very beginning of the introduction. And there is absolutely no reason why QA could not take advantage of innovation, could not take advantage of digitized era, and hence foster further the digital transformation of your company. So my last slide, what path would you like to choose today? Is it the current way of doing and sticking to that, losing immense opportunities? Or would you reflect on a digitized supplier processes, and which ultimately improve also your patient safety and your competitive positioning? Thank you very much for having me here.
Yes, thank you so much.
Of course, if there are questions, please feel free.
Yes. Any questions? Thank you so much, Ms. Jaron, for your presentation. Any questions from the participant side? Okay. So thank you so much, Ms. Jaron, for joining at the 9th World Conference on Pharmaceutical Science and giving us an amazing keynote talk. So thank you so much. So I'm very sorry that the earlier picture, we couldn't capture it very clearly and some of them couldn't join. So I would request everyone to kindly enable your camera once more so that we can capture the picture once again. Just a moment. Yes, okay. So just a moment, 1, 2. Okay, thank you so much, everyone. Thank you. So next we'll be moving on to our technical sessions. So earlier we had started with technical session A, hall A. So all the presenters who are present for the hall A, you may remain in the same hall. There is no change. We have splitted the room to 2, hall B and hall C. So requesting all the participants to kindly move on to hall B and C, that is session 2 and 3. The remaining session A, that is pharmacology and toxicology participants, please remain in the same hall. Now, some of you were having doubts how to move on to the breakout rooms. Please click on your Zoom application where you see more option. Once you log into the Zoom application, on your right side, right corner, you will see more. On that, please click on breakout rooms. And you can see hall B and hall C being assigned. So you may just click on it and join. If you're finding any difficulty in joining the room, kindly put your Zoom name, whatever name you have logged into Zoom, and the required hall you want to enter, we will assign you if you're finding any difficulty. So once everybody moves in to your respective halls, we'll begin the sessions. Requesting the coordinators to please make sure all the participants in your particular session have joined and the session chairs have also joined. Ms. Anita and Abhishek John, please monitor the rooms. Ms. Camelia, can I know to which room you want to join? Which hall?
Good afternoon. Actually, I'm unable to, like, I'm not getting, like, in which pharmacology and toxicology I'll be attending or some other.
No, your presentation is in pharmacology and toxicology session, then you have to be in this room itself. You need not move anywhere.
Okay, thank you. Okay. Thank you very much.
Dr. Manjula, may I know you're having difficulty in moving to the rooms or the audio you're not able to hear?
I have like both. I couldn't hear you properly, and I'm also having difficulty moving to the room.
May I know which room your session is in? To which session you have to move?
That's what we didn't hear. Only that.
Just a moment. I'll just track your name. Manjula.
I'll connect with the internet.
Okay. Dr. Manjula, your presentation is in hall A itself. You need not move to any room. Okay, please be here itself.
Okay. Thank you.
Yeah. Preeta Bose, moving you to hall B. Yes, moved. Anybody else finding difficulty in moving to the rooms, please let us know. We will start the sessions immediately once everybody is into their rooms. Okay, so we are beginning with continuing with pharmacology and toxicology session, and the session chairs evaluating will be Professor Dr. Nahla Ismail and Dr. Bonta Venkata Subramania Lokesh. Welcome back, sir. So our next presenter is Aparanjita R from JNTUH, Hyderabad, India, who will be presenting on development and optimization of nebivolol hydrochloride loaded nanotransferisome patches for transdermal drug delivery. Yes, Aparanjita, your allocated time is 10 minutes. Please make sure you conclude within the time and you may begin. Your slides
Can you
are
hear
visible.
me?
Yes,
Can you
yes,
hear me?
loud and clear. Yes, yes.
Thank you.
Yeah.
Good evening to everybody. First of all, I thank the conference for giving me this opportunity to deliver my work in 9th World Conference on Pharmaceutical Science and Drug Manufacturing. My topic is on development and optimization of nebular hydrochloride loaded nanotransferosomal patches for transdermal drug delivery. Majority of diseases like asthma, diabetics, hypertension are due to lifestyle, and they need drug concentration in the body to maintain therapeutic level for prolonged periods. So to maintain this drug concentration and to overcome the oral drug delivery, there is a need for the alternate route of administration, that is skin has been showing the promising results for delivery of the drugs. When it comes to skin, the epidermis of the skin, stratum corneum, does not allow all the drugs to pass through it. Hence, there is a need for the nanocarriers like transferosomes, which are ultra deformable vesicles which can pass through the skin easily. Why transferosomes? Because they are biocompatible and biodegradable. They have high entrapment efficiency of the drug that made these transferosomes as best carriers for transdermal drug delivery systems. They can encapsulate the drug, and they can prevent the drug from metabolic degradation, and they can release the drug gradually and slowly. These are my materials and methods. I have used rotary evaporation and sonication method. In this method, the phospholipid and edge activated, that is the surfactant, along with the solvent mixture is kept in the rotary evaporator and hydrated with buffer solution and then sonicated. The preparation of the transdermal patches were done by solvent casting method. First, the polymer solution and nanocarrier suspension is mixed, and then plasticizer is added. Then it is solubilized and then kept for evaporation in a Teflon plate for 40 degrees at 40 degrees for 24 hours. First, I have formulated transferosomes by using drug, soya lecithin, and 3 types of edge activators I have used, that is 280, Labrosol, and SPAN 80. I have used in different ratios. From all the 6 formulations that I have made, you can see here the size of the formulations, that is T3 formulation have good size, acceptable size, and better entrapment efficiency. That's why I have gone for further study by using this optimized formulation. This is the fluorescent microscope pictures of nano formulations, and this is the zeta measurement, particle size measurement. And then after preparation of transferosomes, I have gone for the preparation of transdermal patches. First, I have prepared transdermal patches with the help of the drug, that is nebular hydrochloride. I have done the evaluation studies for the drug patches, transdermal patches. Then I have gone for preparation of another set of transdermal patches with nanocarriers. So I have again went for the evaluation of the transdermal patches with the nanocarriers evaluation. These are the evaluation parameters. Here we can see that all the parameters like weight variation, thickness, folding endurance, diameter, percentage drug release, moisture absorption of the prepared patches are under the considerable range. And here we can see for nebular hydrochloride patches, PN5 has shown good drug release, and for nanocarriers, TPN4 have shown good evaluation properties. Then after that, if I have went for a comparative study of the drug release of the 2 patches. The blue one indicates the drug patches which are normal patches, and the brown one indicates the transferosomal patches. Here we can see a significant increase in the level of the drug release when compared to the normal patches. Hence, these transferosomal patches were again taken for the skin permeation study using pig skin. In this study, I have gone for the cumulative amount of drug release for 24 hours and flux. I have measured the flux, permeation coefficient, enhancement ratio, and lag time by taking a control. Here we can see among all the formulation, TPN4 formulation, that is the formulation having 3.5% HPMC E5 and plasticizer 1.5% of 400 PEG 400 have shown better drug skin permeation properties when compared to all the other formulations. Then I have went for a model dependent kinetics studied for zero order release kinetics, model dependent release kinetics, where I have studied zero order, first order, Higuchi, Corsemeyer papers. Here I have seen that the formulation I have selected is following the zero order kinetics and following a non-Fickian diffusion. The drug release is non-Fickian diffusion. Then I have went for in vivo drug release studies by using rat models. Here I have taken control, that is drug solution, marketed formulation, that is also oral, and compared with my transferosomal formulation. Here we can see in the diagram, in the graph, that the transferosomal patch have shown significant, very good increase in this Cmax when compared to the marketed oral formulations. And here the other thing, I have went for the comparison of control with normal patch, that is with the only drug, and the transferosomal patch. Hence, with normal patch, and when you compare with normal patch, the transferosomal patch has still has given a good Cmax increase in the drug concentration, in the plasma concentration in the body. And then after doing a statistical analysis, it is very evident that the T1 formulation when compared to T4 formulation, that is T4 is the transferosomes. Here you can see control is T1, test 1, and marketed formulation 2, normal patches 3, and transferosomal patches 4. When you compare with T1 with 4 and T2 with 4 and T3 with 4, I have got a very good significance for my results. Here, when I have compared the parameters like Tmax and Cmax, the transferosomal patches when compared to control and marketed formulation and normal patch, they have shown very good significance, and which are in the acceptable range. The transferosomal patches have 1.75 folds drug release more, AUC more than the marketed formulation, and 1.4 folds higher than the normal patch. After that, I have went for a skin irritation study for 14 days. I have used guinea pigs for the skin irritation study. When after continuous apply of the transdermal patches on the skin, there is an obvious erythema which is very mild with my transferosomal patches. I have used a placebo like a surfactant and compared with my optimized patches, and this have shown when compared to the placebo, the optimized patches have shown very less and mild irritation. Then after that, the patches were subjected for stability studies. I have gone for accelerated stability studies for 6 months. There, I have evaluated for folding endurance, drug uniformity, drug release through the skin, and moisture absorption. When I have seen all the parameters after 6 months, the transferosomal patches have shown the acceptable range of results after the 6 months, which indicates that the patches are stable after 6 months. Then coming to the conclusion, the present study, the transferosomal nebulinol hydrochloride transdermal patches were prepared and evaluated. The nanocarriers were prepared by rotary evaporation method by using various edge activators. And it was found that the labrosol as an edge activator with soya lecithin is suitable for the preparation of nanocarriers. The prepared nanocarriers were evaluated for size, entrapment efficiency, and zeta, where the optimized formulation of labrosol and soya lecithin have shown 132 particle size and good zeta and with 89% of entrapment efficiency. After that, the optimized formulation was incorporated in the transferosomal patches by using different polymers of HPMC with PEG 400 as plasticizer. The prepared patches were evaluated in vitro skin permeation study and in vivo drug release. Here, the skin permeation studies after further optimization patch, it was seen that there is a good flux, permeation coefficient is very good, and better enhancement ratio, and very less lag, optimum lag time, and with
Excuse me,
good
Ms.
drug
Aparanjita,
release.
sorry to interrupt. You have another 2 minutes to conclude.
The optimized patches have shown to follow zero-order kinetics and also have followed non-frequent diffusion of drug diffusion. In vivo studies, we have compared with the drugs. We have compared transferosomal patches with drug solution, marketed formulation, and normal formulations. Here, we have concluded that there is a prolonged Cmax and Tmax indicating sustainable drug release and higher AUC indicating better availability of drug in the plasma. The related bioavailability of the transferosomal patches have been enhanced in comparison with the marketed formulation and normal patches. The transferosomal patches have not shown any significant skin irritation, and they have found to be have good stability. Hence, the nano formulation with transdermal drug delivery is recommended for going for a antihypertensive formulation. This is the bibliography. And thank you.
Thank you, Aparanjita, for your presentation. So, any questions for Aparanjita? Professor Nehla and Dr. Lokesh? So, can you please unmute?
Aparanjita, I want to ask one question. Actually, you mentioned sample time in your presentation. Yes. Sampling time, you mentioned days.
Yes, sir.
But you not mentioned anything here. Whenever we are presenting in conferences, certain things are very carefully presented. Like for example, here you mentioned sampling time, whether it is hours or days. You mentioned days.
Okay. Yes, sir. Days. I have mentioned days. Sorry.
One thing, and I wanted to ask you. Usually, this is a beta blocker. No, you choose a beta blocker drug.
Yes, sir.
And where do you apply these patches, basically?
Near abdomen.
Near
abdominal region.
So how do you support your study that when you are applying these patches on the abdomen, not on the left part of the heart, because this is related to
yes.
And how do you, because the skin permeation is different, different part of the body and different areas, so how do you support your study? Did you do the what do you call study that variation, study variation of the patches?
Sir, actually, the antihypertensive drugs are generally made to give the drug directly into the blood circulation, sir. So what I have planned for is clonidine was a patch which is already in the market for antihypertensive drugs, sir. Okay. The patch that they are using to apply is near the same region, sir. So when we are going for a antihypertensive drugs, we generally don't target something that is directed to heart. People are according to the literature, the drugs are directly targeted to the blood circulation, sir. If I am applying to that to the specific site, there the blood circulation is very good. So that was the one of the specific site. We can also go for the other sites, like at the back of ear pin also we can go, sir. So basically we target for a place of the on the body where there is a good blood circulation, sir.
Because actually skin is a multi-layered,
Yes.
multi-layered physiologically,
Yes,
so
sir.
when you're taking orally, when you take orally, it will be more effective than transdermal patch. So why the transdermal patch is needed actually sometimes, because
Yes.
when it is preferred for orally, the drug is preferred orally.
Yeah.
Then when you prepare a patch, so you need to see that the improvement better than the oral PK, the availability, the availability.
Yes.
So what are the other complications with the drug if you find out, like for example, in terms of solubility and all?
Yes, sir. This is a class 2 drug, sir. This has very less oral bioavailability and very less solubility. And also, this drug has high amount of first pass metabolism, sir, which made me to go for the another route of application.
Okay, what about the loading dose? Have you managed to reduce the loading dose?
Yes, sir.
How much percentage? Did you calculate that? From the oral to the
Yes.
patch, because the purpose of nano carrier patches and all to reduce the loading dose.
The loading dose, yes, sir.
So did you calculate how much percentage would you manage to get to reduce the loading dose?
Actually, I haven't gone for. I have just gone for a comparative.
No, it's just calculation only, because you see,
yes.
you mentioned some calculations here, like percentage of release, something like that.
Yes, yes.
So then based on that, what is the original dose and what is your patch loading dose? So you can just for example,
yes, yes.
My original dose, let us say 25 milligram. My patch dose is, let's say, 1 milligram. The percentage is drastically decreased, right? So
decreased, yes.
Actually,
yes,
so that
sir.
is very important.
Yes, yes.
When you prepare the novel drug delivery system, one of the outcome of novel drug delivery system and the objective is to reduce the loading dose.
Loading dose. Yes, here the loading dose. When I have seen, basically, the loading dose can be. If I am taking 10 mg of the drug orally, I can cut the dose up to 6 to 5 mg, sir. I can decrease it up to 6 to 5 mg.
6 to 5 mg is not much, because 50% is not much, because for that patch is not required. If it is actually to be 25-fold, 10-fold, 25-fold, 15-fold, usually nano delivery systems, usually their penetration is faster than the regular size of a tablet or any capsule or any pellets which are taken orally. So I trust, kindly look into these kind of factors when you design a particular project, on the novel delivery systems, especially nanoparticles. One of the aspect is the loading dose. So if you reduce the loading dose, you reduce the toxicity. You reduce the toxicity, and it is more effective, because since you are proven that your patch is giving more circulation, more bioavailability. That is also another achievement in your objective. So I trust it can be more improvement. Further go for a higher impact paper. Okay, thank you very much.
Thank you, sir.
Thank you. Okay.
Thank you, sir. Thank you, Aparanjita, for your presentation. So next, I would like to invite Divya Sripada from Kakatiya University from India. Divya's topic is docking-based strategy to design novel isolation derivatives as potent monoaminoous oxidase A inhibitors with prediction of their drug likeliness and ADMET properties. So Divya, do you want us to share your slide?
Yes, ma'am, please.
Okay, we'll do that. Just a moment.
Good afternoon, everyone. Thank you. First of all, I want to thank BioLeaks for giving me the opportunity to present my work. So my work is based on docking-based strategy to design novel 4,5-oxo-4,5-dihydro, 1-hydroimidazole, 1-aisle, N2-oxo-1,2-dihydro-3-indole, thymidine, besides the MAO inhibitor, prediction drug life as well as property. For the PhD in the Department of Pharmaceutical Chemistry at Kakatiya University, Warangal.
Voice is breaking. We are unable to hear you. And Divya, is the screen
Ma'am,
visible
now?
to you?
Yeah, ma'am, it's visible.
Okay, okay.
Ma'am, next slide, please.
Yeah. Are you able to see the slides moving? No?
No, ma'am.
Just a moment.
No.
How about now?
Yeah. Now it
Okay,
has.
okay. Then I will not put
Yeah,
on
it's
slide
moving.
mode. I think this will
Yeah,
be better. Okay. Yes, please
Okay.
continue.
The flow of contents include introduction, need for the study, materials and methods, results and discussion, and conclusion. Ma'am, next slide. So introduction include, so isotin is my basic nucleus, that is 1H indole 2,3 dione, which is also known as indene dione and indole quinone, which is a biologically active heterocyclic moiety. It has a nitrogen atom at first position and 2 carbonyl groups at second and third position, and it's a unique molecule which possesses both amide and ketocarbonyl groups. So apart from this, it has an active hydrogen atom attached to nitrogen or oxygen and an aromatic ring. It undergoes ketoenol tautomerism. So it exists in a tautomeric form. The 3 carbonyl group of isotin is strongly
Divya, are you online? Divya, your voice is breaking actually. Okay, please continue.
Okay, the C3 carbonyl group of isotin is strongly erythrophilic, and as a result, isotins are readily involved in condensation and addition reactions with carbanion type nucleophiles into 3 substituted oxynodes. Ma'am, next slide, please. So, my topic of interest is depression. So, depression is a neurological that affects hundreds of millions of people all over the world. So it's a disorder that has major impact on patients' well-being and on personal, social, and familial achievements. It is tremendous cost to society as this disorder causes several distress and disruption of life, and if left untreated, it can be fatal. So depression is a significant contributor to the global burden of disease and affects people in all communities across the world. So today, depression is estimated to affect 350 million people. The World Mental Health Survey conducted in 17 countries found that on average about 1 in 20 people reported having an episode of depression once in their lifetime. Next slide, please. So the mechanisms involved in depression. So electrical impulses or the information to the brain, they are transmitted to the synapse by a release of various neurotransmitters, which mainly include noradrenaline, serotonin, which is 5-hydroxytryptamine, dopamine, acetylcholine, histamine, GABA, substance P, and endogenous opioids. So all these aminergic systems consist of small groups of neurons which have projections encompassing throughout the brain and spinal cord, allowing them to globally modulate all the neuronal functions. So when these functions are disrupted in one or the more steps of this chemical transmission, it may be crucial mechanism which underlies the major cause, that is depression. Next slide, please. So what is the need for the study? So in the conventional drug discovery and development is highly expensive and is a time-taking procedure. So it involves many steps starting from synthesizing the molecules, then filtering the synthesized drug molecules and obtaining the lead molecule. So it is highly essential to overcome all these limitations of conventional drug discovery approaches with less expensive and effective synthetic approaches. So CAD, that is computer-aided drug design, is one such approach which assists in identifying potential drug molecules and it reduces the time and cost required for the drug delivery. So the application of computational tools for identifying a novel drug molecule or a candidate assists to lessen the number of experimental researches and for increasing the success rate. Next, ma'am. Next slide, please. Ma'am, next slide, please. So in the present study, what I have done is I've designed 10 isotype derivatives, 10 isotype which I have docked at the active sites of MAO-A enzyme. So this MAO-A enzyme is obtained from RCB protein data bank, which is a PDB-ID of 2BXR. It's a widely known enzyme which is target for depression, and the docking studies were carried out using Autodock software, Autodock 4.0 software. So after performing docking for all the 10 compounds, they are ranked according to their binding affinity values. So additionally, all the 10 compounds were also screened for their ADMET properties using Swiss ADME software. Next, ma'am. Slide, ma'am. Ma'am, next slide, please.
Ms. Divya?
Next slide. Yes, ma'am. Hello?
Yeah. That's materials and methods. Please continue.
Yeah. So when the materials have used, this procedure involves ligand preparation, the protein preparation. In ligand preparation, all the structures used were in chemistry software. Files are small format. They are converted from Babel software. File format is a format for the so it's open Babel software. The structures are like, and when it comes to the protection, the three-cell structure utilized for dogging was obtained from RCSB protein data and with ID2. So this file was saved and all chains attached to the enzyme molecule and the cofactors and water molecule. Hello? Hello, ma'am? Am I audible? Hello, ma'am, am I audible?
Yes, Divya, your voice is breaking actually. Shall we move to the next slide?
One second, ma'am. I'll complete this. Ma'am, I'll complete this. Yeah. The file was saved in the PDF format. All the extra chains, cofactors, water molecules are deleted. And in the second, polar hydrogens and missing atoms were added. And in the Coleman charges are also added to the enzyme. And the atoms were assigned as AD4 type. So that is residual charges are also spread all over to the enzyme. And the energy minimization, which is very much essential, was achieved using SPDB software. Ma'am, next slide, please. Yeah, these are the structures of the ligands which I have designed for docking purpose. Next slide, please. This table one gives us the information about molecular interactions present in the docked poses and the amino acids in first the ligand molecules. So these are the ligand molecules and derivatives and have their binding bond in hydrophobic conditions. To ensure that the design compounds are addressed.
Ms. Divya, you have 1 minute to conclude.
For the minute 50 times.
You have
Okay,
just
ma'am.
1 minute to
Next slide,
conclude.
please. Okay, ma'am, go to the conclusion. Yeah. Okay. So the depression is a neurological and potential is all that. Now in here were found to have antidepressivity. All the derivatives have designed and against mouse. So that were predicted using studies performed using the 2D visualizers where using Discovery Studio. Ma'am, next slide. Ma'am, next slide, ma'am.
Yes.
Yeah, the docking results active site. Binding energy is minus 4.8 per mole, so minus 0.37 kilocalorie per mole. So among the ends which I have designed, the compound nitrous derivative and derivative have best binding affinity. That is the minus 0.4 kilocalories and minus 13.7 kilocalories. Formed hydrophobic agents with MAO enzyme. So
I'm very sorry, Divya. We are not able to understand anything that you're saying. It's not clear at all. So it's time up. We'll just conclude because we are not able to follow your audio.
Okay, I'm sorry.
No problem. That's the internet problem. So session chairs, any questions or comments for her presentation? I think as we were not able to actually follow you, it will be difficult to comment
No
on the
comments, but
presentation.
whatever I see that actually, I would suggest one thing actually, you know, we are investing time here and sitting here, and right, the presenter should know in a particular environment where the internet is stable and sitting and present, because conference presentation is a responsibility. So I request Divya to be next time when you present, you know, kindly make sure that your internet is stable and you are adapted to the system, virtual system clearly, and present the presentation. Thank you very much. And I see some of the structures are not clear. Actually, I cannot ask any questions on that. Otherwise, I'm expert in talking. Actually, I would like to ask some questions, but I cannot ask anything because I could not see the satin structures which you have been consolidated for depression. Thank you. Huh?
Thank you, sir. Thank you, Divya. Yeah. So quickly moving on to our next presenter. Next, I would like to welcome Sezen Yilmaz Sariltan from Ankara University from Turkey. Sezen, can you hear us?
Please.
Yes. So Sezen will be discussing on the levels of interleukin-1 family cytokinins and oxidative stress in rheumatoid arthritis patients in terms of disease activity.
Hello, can you hear me?
Yes, yes. We can hear you. Yeah.
Can you see my presentation?
Yes, you can put this slide. Yes, you can put the presentation in slide mode and begin.
Yes. Is it okay? You can hear me, right? Okay. Dear participants, first of all, I'd like to thank you, the organizing committee, for giving me opportunity to present our study to this Congress. I'm Dr. Sezen Yilmaz Saraltan from Ankara University, Faculty of Pharmacy, Department of Pharmaceutical Toxicology. The subject of my presentation is the levels of interleukin-1 family cytokines and oxidative stress in rheumatoid arthritis patients in terms of disease activity. My presentation includes the following sections: introduction, methods, results, and last, the conclusion. Rheumatoid arthritis is a systemic autoimmune disease that causes chronic inflammation characterized by pain, swelling, and irreversible damage to various joints, mainly synovial joints. The most prominent features of this disease are cartilage damage, bone erosion.
Susan, your slides are not moving. Can you please look into it?
I'm moving now. What about you?
No, it's not moving for us.
What about now? Can you see my second?
Yes.
If it is possible, I can present my presentation like this, because if I use the full screen, I think there is problem about the system.
Okay,
is it
no
okay
problem.
for
This
you?
will be better.
Okay,
Yes,
I'm sorry. Let me start from the beginning. And this is my presentation includes the following sections: introduction, methods, results, and last the conclusion. Rheumatoid arthritis is a systemic autoimmune disease that causes chronic inflammation characterized by pain, swelling, irreversible damage to various joints, mainly synovial joints. The most prominent features of this disease are cartilage damage, bone erosion, and joint integrity deformations caused by synovial inflammation. Rheumatoid arthritis is the most common autoimmune disease in our country and also in the world. And this disease ranks third most common disease among patients aged 15 and over, according to Turkish Ministry of Health Statistics Yearbook. And also, this disease ranks third in the list of diseases diagnosed by the physician, according to the report published by the Turkish Statistical Institute. Approximately 0.24 to 1% of the population, of the world population, suffers from this disease. The formation and development mechanism of this disease, the rheumatoid arthritis, has not been clearly elucidated yet. Many factors contribute to the onset and progression of the disease. We can classify these factors as genetic factors and non-genetic factors. The non-genetic risk factors are smoking, nutrition, and female gender. Although rheumatoid arthritis is the most common systemic autoimmune disease, there is no specific biomarker that allows to the diagnosis of the disease yet. Moreover, there is no definitive and effective treatment of the disease yet. Also, it has been known for many years its etiopathogenesis is still unclear, revealing the fact that new biomarkers are needed in this pathology. Interleukin-1 family cytokines are key signaling molecules in inflammatory disease. These diseases are often the outcome of excessive oxidative stress, which occurs by increasing inflammation, regulating apoptotic cell death, and distributing immunity. The aim of this study is to assess the levels of interleukin-1 family cytokines and oxidative stress status in rheumatoid arthritis patients with different disease activities and using medications and not. The patient group was divided into active and remission groups in terms of disease activity. These patients are treated for rheumatoid arthritis. In addition, newly diagnosed patients who have not used any medication were included in our study. Plasma interleukin-1 beta, interleukin-18, and oxidative stress levels were measured by enzyme-linked immunosorbent assay. The levels were measured and compared in the patient and control groups. And also, as I mentioned before, we divide the patient group into 3 different groups, and we compared the levels in these groups. Our study included 5 to 6 patients and 30 controls. The mean age of the patient group was 43.71 age and year, and the control group was 38.17. 72.09% of the study group was female and 27.91% was male. During the selection of the volunteers in control and patient groups, it was noted that there was no significant age and gender differences between the patient and control groups. You can see the gender of the subjects and age of the subjects in figures here. The mean plasma interleukin-1 beta level of the patient group was higher than the control group as shown in the figure. And the differences between the groups was found statistically significant. We compared these groups using Mann-Whitney statistical test. As I mentioned before, we divide the patient group into 3 different groups. And the highest mean plasma interleukin-1 beta level among all groups was in active patients. The interleukin-1 beta level of the patients in the active period was significantly greater than the remission group. This is one of the most important points of our study. And plasma interleukin-1 levels of the patients in remission, active, and newly diagnosed groups were all significantly higher than the control group. We used a Kruskal-Wallis test to compare the results here. According to the results of ROC analysis, we also conduct ROC analysis. The area under the curve for interleukin-1 beta was shown on the screen here, and the result was statistically significant. And also the diagnostic value of the interleukin-1 beta was at the good level on differentiating control and patient groups. When we look at the interleukin-18 levels, the mean interleukin-18 level of the patient group was higher than the control group, as you can see from the figure here. And the differences between the groups was statistically significant. Here we also use Mann-Whitney test to compare the results. The highest mean plasma interleukin-18 levels among all groups was found in active patients. Plasma interleukin-18 levels of the patients in remission, active, and newly diagnosed groups were significantly greater than the control group when we checked the results. And similar to the results of interleukin-1 beta, active group has the highest level of interleukin-18. We also conducted ROC analysis for interleukin-18 as well. And as you can see from the screen, the area under the curve for interleukin-18 was shown on the screen, and the result was statistically significant. The diagnostic value of interleukin-18 was at the moderate level on differentiating control and patient groups. The mean plasma reactive oxygen species level of the patient group was almost 1.2 times higher than that of control group. The differences between the groups was statistically significant. Also here we use Mann-Whitney test to compare the result to determine the statistical differences level. And the highest mean reactive oxygen species level among all groups belong to the patients in the active period. Reactive oxygen species levels of the patients in the active period is higher than the control group. Similarly, remission and newly diagnosed groups plasma reactive oxygen species levels are statistically significantly higher than the control group. Here, we compare the results using Kruskal-Wallis test. And we also conducted ROC analysis for...
Sorry to interrupt, Ms. Susan. You have another 1 minute left for your conclusion.
Okay. And plasma reactive oxygen species levels, the results was statistically significant, as you can see from the screen. And the diagnostic value of the reactive oxygen species was at the moderate level in differentiated control and patient group. As a conclusion, to the best of our knowledge, our study is the first report to investigate and compare the levels of interleukin-1 family cytokines and oxidative stress in treated remission and active rheumatoid arthritis patients and not treated newly diagnosed ones. And interleukins and oxidative stress levels of the patient group were significantly higher than that of control group. The highest levels of examined parameters belong to the patients in the active period. The higher levels of these parameters in active period of patients suggest that an inflammation pathway involving these interleukin-1 family cytokines, and oxidative stress contribute to the progression of the disease. Although rheumatoid arthritis has been known for many years, etiopathogenesis is still unclear and indicating that new biomarkers are needed, as I mentioned before. And the parameters may be new biomarkers for rheumatoid arthritis, and new selective interleukin-1 beta, especially 1 beta inhibitor molecules, may contribute to the treatment of the disease. Because when we check the ROC analysis results, interleukin-1 beta is a good tool to differentiate control and patient groups. And lastly, I'd like to thank you, my colleagues, and Ankara University Research Fund to support this study. Thank you very much for listening to me. This is Ankara University Faculty of Pharmacy. This is my faculty. Maybe one day I'd like to see you in our congress in our faculty. Maybe you heard this before. We have a congress called ESOPs, and next year we will perform this again, and it will be nice to see you in our congress. Thank you for your listening.
Sure. Thank you for the invite, and thank you for your amazing presentation. Over to our session chair. So any questions and comments?
I think it was a good presentation, and I just want to ask one query. Could you manage in your study group obese and non-obese group? Did you try that?
Actually, not. We divide our patients as female and male, smoking and not smoking, but not obese and not obese patients. I think this is a good idea for us. Maybe for our next studies, we should do this. I think I'm writing this.
Yeah, I trust, because usually the primary target patients are usually obese group for arthritis, and arthritis start with the heavy bodiness, body. Those who are having high weight, like for example 80 and above, 90 and above, 100 and above, and all. So hopefully you can try that. You can probably pick up within your study. You can pick up if you find such high weight and obese patients, and probably you can correlate, intercorrelate your data within the data what you obtained actually.
Of course, because nutrition is an important factor for rheumatoid arthritis, but we haven't think that. That's why this kind of congress are really important for us, because this brainstorming. Thank you for your.
Thank you very much. Otherwise,
thank you very much so much.
Thank you so much for your presentation, and thank you, sir. So we will move on to our next presenter. Next, I would like to invite Sudha Devraj from AIMST University, Malaysia, who will be discussing on development of an optimized fraction of Strobilanthes crispus using response surface methodology for anti-cancer activity. Yes, Dr. Sudha, are you ready?
Yeah, am I audible?
Yes, you're audible, and you may begin. Your screen is visible. Yes.
Yes, thank you very much, Ms. Jazila. Good day to delegates and respective judges and organizers. So I'm Dr. Sudha from AIMS University. I'm a lecturer in the unit of pharmacology from AIMS University, and I'm here to present a part of my PhD project entitled Development of an Optimized Fraction of Strobilanthes crispus using response surface methodology for anti-cancer activity. Let's move on. Okay, so let me introduce my traditional herb that I've been using throughout my research project, which is about Strobilanthes crispus. Okay, interestingly, this plant is actually known as traditional medicine plant in Indonesia and Malaysia. It is commonly known as Pecah or Pecah culture. This is actually a Malay term for this plant. And this is actually a small shrub, belongs to a Acanthaceae family, because its maximum height that it can grow is about 0.5 to 1 meter. And there are many studies actually scientifically has proven that it has anti-cancer features in a different angle. Okay, and it also used as a remedy for diabetes, hypertension, kidney stones, and constipation. And actually, why this research has been carried out is because it is traditionally has been consumed by boiling in water like a tea, or actually some kind of disorders like disease, some diabetes and hypertension. And also there are claims that it has been used for cancer. So this provokes our interest to actually dig into this plant, as these people now are moving into natural products for prevention and treatment of many diseases instead of using single drug, because they believe that the use of natural products like nutraceuticals or supplements has lesser toxicity, as many components are actually working together in boosting our immune system, as well as to prevent and treat for the particular disease or therapeutic properties. So this is basically the introduction of my plant. Okay, so this is actually a picture taken of the plant near our location in Malaysia, Tasik Glubo Penang Island. Okay, so this is taken in one of the farm which has been where this plant has been cultivated. Okay, so basically the objective of my study is to optimize the extraction conditions of this Trabulanthus crispus extract using response surface methodology, and to develop an HPLC analytical method for quantification of the active fraction of this plant using the bioactive markers. Okay, so methodology basically I've compiled here in a flowchart. It started with a preliminary ultrasonic extraction method followed by a single factor experiment. And from the single factor experiment, the parameters has been narrowed down, which has been subjected for response surface modeling using box Benken design. And eventually it has been subjected for cytotoxic evaluation, which is LDH assay, and also for standardization of the optimized extract, which is HPLC method. Okay, so the preliminary extraction method was where the leaves of the S. crispus, the plant is freeze-dried and powdered, and subjected for ultrasound-assisted extraction. Okay, where this plant with the respective solvent is placed in a water bath and with some and sonified, ultrasonified, okay, to actually extract out the metals, the active ingredients from the extract. So the plant to solvent ratio has been used is 1 to 10. And the solvents, we started with hexane and followed by DCM. Because why hexane? Because we want to remove all the fatty acids, I mean the waxes and fatty materials, and followed by DCM to extract the real components. And following that, the temperature used is around 20 to 25 degrees Celsius, and the duration of the sonication is 20 minutes. And also the overnight, followed by overnight soaking with both hexane and DCM to ensure that more ingredients, active ingredients are extracted out. Okay, for the single factor experiment, this single factor experiment is done to determine the most significant parameters for the response surface methodology analysis. Okay, so what are the parameters has been optimized here is the drying method, where we use 3 types of drying methods: freeze dry, air dry, and oven dry, and particle sizes after during the grinding method, where we have sieve with the different measurement 0.5 mm, 1 and 2 mm. And also the time, extraction time manipulated from 10, 20, 40, and 60. And also the solvent temperature range from 15 to 45 degrees Celsius. And plant to solvent ratio also was actually optimized. And also the frequency of extraction, where we played with the number of extraction cycles using different solvents, which is hexane and DCM, like 1 time, 2 times, and 3 times, something like that. Okay. So next, once it's the parameters has been narrowed down, then the response surface methodology has been employed by using box Benken design, because we want to see the interaction and also to minimize the number of experiments to be conducted. So we want to see the interaction between different parameters, how it works, and which will yield the optimum extract with optimum cytotoxic effects. So these are the tables which will be generated through the RSM software with independent variables, solid to liquid ratio, time and temperature with high and low levels, which will be determined by the software. Okay, so the analysis of response based on the yield and also the cytotoxic activity, which is the breast cancer cell lines, MCF7 has been used here, and LDH-SC. So the results of the single factor experiment, so I've summarized here. Okay, for the freeze dry will be the best method based on the comparison of the yield and cytotoxic activity. And particle sizes is 1 mm would be the best. And next will be the plant to solvent ratio is 1 to 10, and frequency of extraction, where one time extraction with hexane and DCM itself will give the best yield and cytotoxic activity. So the single factor experiments should narrow down the parameters because one variable can be tested at one time. So we cannot see the interaction between the different parameters or variables because it's single factor experiment. So what happened is we narrowed down and we used response surface methodology to see the interaction between these three different variables, three different parameters, which is solid to liquid, time and temperature. And the model has been generated by the software which has 15 runs with the actual and predicted cytotoxicity activity as well. So this has been employed in our lab, and we found that there are significant, which parameters is significant is that the interaction between time and temperature gave us a significance, and time is the main actually showing significance in yielding the optimum bioactivity. Okay, so the model is actually significant, which actually showed this model is fit and adequate for the study, and lack of fit is not significant, which refers to the validity, which showed that this model has high validity for testing these parameters. Okay, so this actually generated by this RSM software. Okay, so this A figure is A figure showed the effects of time and solid to liquid ratio of the cytotoxicity of asbestos, and the next will be the interaction between temperature and solid to liquid ratio, and C is effects of time and temperature on the cytotoxicity of asbestos. So the based on the figure C, sorry if it's not very clear because I have to squeeze in into this PowerPoint. So from the software, if we move around, then it shows the time and temperature has the most significant effect towards the optimum cytotoxicity activity. So here is what we have summarized. Okay, so the cytotoxicity activity could attain a peak at 25-degree temperature and 40 minutes of the ultrasonification time. And the response rate can be seen rising from 40 to 60% at lower extraction temperature, but then at only 10 ml of solid to liquid ratio particularly. So there is no, we don't see any decrease or increase at the solid to liquid ratio when we modify it. So, but when we actually play it with the temperature and time, then we can see some increase in the cytotoxicity effects. So this is how we have actually manipulated, and we got the best optimum ultrasonic conditions, which is that ultrasonic time with the 38 minutes, and the temperature is 27 degrees Celsius with the solid to liquid ratio of 10 ml, and it was executed in the actual experiments in triplicates. So under the optimal extraction conditions, we have obtained the experimental value where the cytotoxic activity which we have obtained is about 66%, which corresponds well with the predicted value, okay, from the box Franken design, which is about 68%. So this good correlation observed between the experimental and predicted confirmed this respective model is accurate in predicting the cytotoxic response of these leaves. and using this ultrasound assisted extraction.
Excuse me, Ms. Sudha, you have another 2 minutes to conclude.
Okay, yeah, sure. Okay, and when we subjected for cytotoxic activity, and from the extract we have produced a fraction by isolation, and this fraction actually surprisingly showed like around 80% inhibition of the MCF7 cells at 24 and 48 hours. So from here, I've actually quantified the compounds that I've isolated, okay, in different batches to ensure the reproducibility of this fraction for further analysis. So the main compound is lutein, followed by this rare compound, which is the chlorophyll derivatives, and also these sterols. So this is done using this HPPLC quantification, and this is the fingerprint that we have obtained. And this is the conclusion that, you know, F3, which is fraction 3, is it has the optimum activity with the optimum conditions, ultrasonic conditions, and this actually the application is potential to be developed as an effective nutraceutical agent or supplement, and also it has been tested in vivo experiments with excellent inhibition of the MCF7 cells. Thank you for the opportunity given.
Thank you, Ms. Sudha Devraj, for your presentation. Over to the session chairs. Any questions?
Thank you, Ms. Sudha. You've done your name is Ms. Sudha, Dr. Sudha.
Yes. Yes.
I think it's a good work, and but I would like to ask you one question here.
Yes. Sure.
Cytotoxicity you have done on cell lines, right?
Yes, 7.
Yeah, then usually you must present in IC50 value, right? And how do you present in percentages?
Because we didn't do IC50 as this was actually a collaboration work. Okay, so the cytotoxic effect is done by my team member, and this was the results presented because we just did the basic percentage level. So yeah, I understand that IC50 should have been done, but because it was presented in the
comparing with you're comparing with tamoxifen 15 micromolar in IC50 value.
Yes.
And moreover, how do you assess the predicted value? How do you assess what is the benchmark for predicted value?
Predicted value of what, sir?
You said that you compared with predicted value.
Yes,
no?
yes.
Right?
The predicted value is from the software where I used box Penton design, and it gives me the predicted value of the cytotoxic activity is about 68%. So which I have to obtain in the experimental value as well. So it I found that there is a correlation, a good correlation between the experimental and predicted.
I think it would be better if you mention in IC50 value.
Yeah.
Because that is the standard format of presenting cytotoxicity in general.
Okay, sir. Okay.
So most of the manuscripts, if you see that cytotoxicity is mentioned in IC50 value, and how much IC50 value compared to the control. Control here you can use as a protocol tamoxifen, right? Positive control.
Yes.
So you just compare and say that how much fold your activity you have, cytotoxicity activity you have. Like for example, 2-fold, 1-fold, equal, similar activity, maybe 10-fold, 1-fold, 2-fold, like that. You can mention that. Then that makes more sense of work, isn't it?
Yeah, actually, I have 2-fold. The activity was 2-fold.
Yeah, that's
better
what. This is what the world scientific community understands.
Okay.
Rather than if you say percentage of cytotoxicity means I didn't understand first of all. Then I go through them. When you show this slide to me, 15 micromolar tamoxifen, they understood. I think you did on cell lines. When you did on cell lines, usually IC50 value will be mentioned, isn't it?
Yes.
Yeah, good.
So I just want to clarify here that it's 2-fold activity. Yeah. Sorry that I didn't mention.
You didn't mention that, is it, in your presentation?
Yeah.
So that makes sense. That's making added value. Otherwise, the work will be, there is no hypothesis, there is no problem statement.
Okay, doctor.
Good work.
Okay.
Thank you.
Yeah, thank you very much.
Okay, hello.
Yeah.
Yes, Professor Nahla, your comments, please. Yes. Can you please unmute, Dr. Nahla?
Am I audible?
Yes, you're audible.
Yeah. Is the presenter still there?
Yes, I'm here.
Okay. No. Okay. It's a good work whereby you optimize the extraction so that you can look into the cytotoxic activity. My question will.
Sorry, Prof, you're muted. I can't hear you.
Can you please unmute, Dr. Nahla?
Yes. In terms of the cytotoxic activity, what will be the active entity that actually produce that cytotoxic activity?
Oh, sorry, Prof. I didn't do MTTSA here. I just did LDHSA.
So meaning you don't know what compound that.
What compound? Because I didn't test on the compound. When I tested the cytotoxic activity on all the compounds that I've isolated, they showed around 40%, 30 to 40% inhibition compared to the fraction 3. So we found that fraction 3 has twofold higher activity compared to single components. So that's why we remain as the fraction.
No. My question is what?
Yes. I'm sorry that you are muted again.
You should have known what will be the content.
Yeah, that's what the content was actually lutein. The main component was lutein and also some chlorophylls and steroids. Based on what
okay, and that one you check the content from using what?
I actually isolated the compounds and then I checked using HPPLC and also HPLC method. So the HPLC couldn't identify the steroids. So that's why we have to switch to HPPLC because we want to put the fingerprint of all the components in the extract, in the fraction.
Okay, all right. So meaning you're not using any NMR to confirm, because the one that you presented is like, for me, it is more on the as a screening thing. It's a part of the screening exercise. It's just to screen and then optimize the extraction method, and then you show the different cytotoxic activities. Whether increase twofold, fourfold, so on and so forth. But my question will be like, because this study is very good. It's just that what will be the things that is actually a gift that produce the cytotoxic activity? At least if you can show us that, you have looked into maybe using an NMR to show that, okay, like by doing this, these are the things that is actually the one that you get, and then these are actually give the cytotoxic activity that you're looking for,
Oh, okay, Prof. I understand what you meant, but actually I didn't include the isolation part here because I presented somewhere else. So that's why I presented this optimization part here. So, but actually when I isolated, that is a different chapter of my thesis, which is about characterization of the isolated compounds. But then we can't, but actually for your question, I would like to clarify here that there is no specific component that contribute to the cytotoxic activity. When we tested on cytotoxic activity on each compound, it gave us the similar results actually. That's why we have concluded that fraction is when the compounds have synergistic effect together. So when they are in a fraction form, the inhibition activity was better, twofold better compared to when we tested as a single component. So that's what I want to clarify here. So more work actually should be done, more components should be actually isolated, but the isolation process from this class is actually very difficult.
All right, I think that's all from me.
Oh, thank you very much.
Okay, thank you, Dr. Sudha Devraj, for your presentation. Yes. So our next presenter is Dr. Akshaya Sridhar from Sri Ramachandra Institute of Higher Education and Research from India, who will be discussing on documentation in clinical research.
Good afternoon, ma'am. Am I audible?
Yes, you're audible.
Okay. A warm greeting to everyone. I am Dr. Akshaya Sridhar from Sri Ramachandra Institute of Higher Education and Research, Chennai, India. I am currently pursuing my final year master's in clinical research. Today, I am here to give a brief narrative presentation about the most important aspect of clinical research, that is the documentation process. To begin with, I would like to tell you the generic reason for documentation in a healthcare sector. Documentation is commonly referred to as paperwork, which is deemed to be really hectic and a tedious process. However, the documentation process in healthcare is a very crucial one, considering that it involves patient services and that we need the access of the data whenever it has happened. So it can be boldly stated that the cost of a good documentation process is definitely going to be lesser than that of the rectification cost, which is going to be caused by poor documentation. Pertaining to clinical research documentation, why should it be performed? Clinical research involves people from multiple backgrounds, ranging from doctors, nurses, trial managers to statisticians, who possess different qualifications, experiences. Furthermore, the research projects are often multicentric in nature, and the possibility of adverse event is a frequent happening in clinical research when pertaining with the real world scenario. Therefore, documentation in clinical research helps improving the validity and credibility of data and ensures an organized, smooth workflow process. Moving on, we'll now hear about the two means of documentation, the physical or manual documentation and the e-documentation, electronic method. The physical documentation is wherein you actually record stuff on a paper and then store it in files in a physical storage space. Whereas electronic documentation is when you start recording the patient data through your mobile phones and electronic devices like laptop, etc. So it can be safely said that the manual method of recording data actually requires more physical storage space, which require proper maintenance. So we can always prefer electronic mode of documentation in the current scenario. Moving forward to one of the most important element of data documentation, it should have the following 6 essential features, that is the ALCOAC checklist. A stands for attributable, that is the raw data should be traceable to an authorized person. Whoever is recording the data should be known to us. L stands for legible, the raw data should be easier to read. That is the written data should be legible, and if it is entered in an electronic data system, it should have a readable and identifiable format of data. C stands for contemporaneous. The raw data which is recorded at the time of observation only must be ensured. It should not be recorded at some other later date. O stands for original data, wherein the data is the information obtained from first-hand observation and it is not a copy. A stands for accurate, that is the data is unaltered and the correct reading of whatever has been observed from the study. So C means complete, that is the data is inclusive of all the data from the study. According to the stage of the clinical trial, the essential documents can be categorized as follows. The before study documents, during the conduct of the trial, the documents that are required, and post study and after the completion or termination of the trial. So I am now going to enlist all of the documents and talk in brief about some of the significant documents in each stage. So let us first start with the pre-study documents. The pre-study documents include the investigator's brochure, which is the collection of all clinical, non-clinical data pertaining to a new investigational product, that is the animal data and everything. Protocol, case report form, informed consent form. These are all the essential requisites before the study starts. It ensures that the study goes in a particular direction. And any other written information that is going to be provided to the subject in their exact wording and content should be there. And if there is any advertisement used for subject recruitment processes, it should be appropriate. So that advertisement is also one of the pre-study documents. And the insurance statement, wherever applicable, should be documented to ensure the compensation that will be provided to the subjects. Most importantly, the approval of institutional ethics committee. That is one of the important pre-study documents. Moving on to the second portion of the pre-study documents, we should also get to know the composition of the ethics committee which has approved the study, and all the regulatory approvals must be obtained, and the curriculum vitae of all the investigators and the personnel, just to check if they are really qualified and eligible for the conduct of the trial should be there. And then financial aspects of the trial and any laboratory recordings or values, if it is pertinent to a trial, that normal range has to be provided. If it is a new investigational product, then the sample of the label, how is it going to be labeled, and instructions for handling and storage conditions should be mentioned very properly. Moving on to the last portion of the pre-study documents, This may seem as a very little factor, but shipping records is one of the essential requisites because that only proves that the investigational product has been intact throughout it. And in case of randomized control trials, decoding procedures and a monitoring report before the trial has been started. So now I'm moving on to the documents required during the study. So I can just tell you that whatever documents we have seen in pre-study, any updates or amendments to those documents can come under during the study documents. When we do a study only, we will get to know the practical aspects, and there might be some amendments or changes that has to be made. So all that will come under during the study documents. And coming to the core of during the study documents, it mainly involves the monitoring visits that is organized by the sponsor and informed consent forms from the patients. If it is appropriately signed and everything, source documents, that is to match the relevant documents that the patient is original record. So completed case report form. So whatever is related to the patients will also come under during the study documents. So, and any notification by the investigator and sponsor in case of any adverse events in the trial can also be said during the study documents. And whatever related to the patient, that is the subject screening log, identification log, all that also comes under this subject enrollment log. So finally, the signature sheet is very important because that will serve as a record for all the personnel who are involved in the trial. So a sample signature of all the people who are involved in the trial should be maintained. And how are we going to utilize the retained body fluids? So now I'll move on to the subject study completion documents, the documentation of the investigational product and its accountability at site. So therefore, we have to maintain proper records of the investigational product during the study and the subject identification code list, and we have to destroy the investigational product by the sponsor or at the trial site itself, and that has to be documented. So audit is nothing but an external body coming and visiting and monitoring your site. So if it is available, an audit certificate. And the final trial closeout monitoring report, which is written by the investigator and sponsor. Now, the last set of documents, that is the post-study documents, the investigator will write a report to the institutional review board or ethics committee and to the regulatory authorities, and the clinical study report, which encapsulates all the study results and interpretation from it. So now I would like to talk a few things about good documentation practice. It is one of the crucial components. So I have listed out so many documents. So it should follow the good documentation practice. So it is commonly referred to as a good record keeping practice, and this is going to be really helpful during regulatory inspection, non-conformance, or device investigation.
Ms. Akshaya, you have another 2 minutes to conclude.
Okay, ma'am.
Yeah.
Okay. So these practices are going to be mandatory, that the documentation and the products is going to meet the real industry standards. That is, it stands out in the pharmaceutical sector. So good documentation practice is one of the most important aspects that need to be followed by people who are involved in the trial documentation. So every time that when a data is recorded and you document it, there has to be 4 questions. Is it true and is it accurate? That is the correct representation. Is it timely and is it legible? When these 4 questions are answered and the data is recorded, that means it is compliant under good documentation practices. Here are a few do's and don'ts. I'm going to mention about do's and don'ts of clinical trial documentation. So overall, I would like to say the do's should really reflect your professional capabilities. So we must make sure that we do everything properly, because without documentation, there is no proof. So these are some of the don'ts of the clinical trial documentation. So we are not supposed to anticipate. We should be unassuming in nature and go with an open mind and record what has exactly happened, and not be in a hurry to change anything or like alter the subject record, because that will all fall under research misconduct. So hence now we know that documentation is really one of the important aspects of clinical trial. It can be stated that if it isn't documented, it did not happen. This is the thumb rule of documentation. So conversely, now you can understand, just because something is documented doesn't mean it has taken place. We have to always make sure and ensure that the document, whatever data we have, is credible. So these documents should serve to demonstrate the compliance of the investigator, sponsor, and monitor with the standard good clinical practice guidelines and all other applicable clinical trial regulatory requirements. Therefore, timely filling of these essential documents will ensure the successful management of the trial. Actually, I would like to thank everyone. This is my first international presentation. I am a dentist by nature, and I'm pursuing my master's in clinical research. So I am really excited about this presentation, and thank you very much. Thank you, sir, thank you, ma'am and sir, everyone. Thank you.
Thank you, Akshaya, for your presentation. Session chairs, any question for Akshaya?
Maybe I think we can give a chance to Prof. Nala. Prof. Nala, good afternoon.
Hello, Dr. Akshaya Sridhar.
Yes, ma'am.
Okay, it's a good presentation. I believe this is the summary of what you would like us to know about the documentation in clinical research, right?
Yes, ma'am.
Okay, I mean, there is no other methods that been done, meaning, you know, like some kind of at least systematic review analysis.
I'm trying to do a systematic review on this, ma'am. Like I compiled all this data, like pre-study documents, what are the essential things, and make.
Okay, I mean, the one that you're doing is basically it is sort of like a review on the documentation in clinical research, right?
Yes.
Okay. Yeah, because if we mention it about the systematic, then the presentation will be in a different form, whereby you will show, like, you know, in the table of, you know, of all those you know, papers that you have went through. You understand? You understand?
Yes, ma'am.
You know, it's okay. It's okay. I mean, I think this presentation will help you later. As you mentioned, you are pursuing study for the clinical. Okay, and of course, the documentation is very important for anyone that would like to do clinical research, mainly those that are involving patients in a large number. Okay, so I wish you all the best for your future study.
Ma'am?
I wish you all the best for your future study.
Thank you so much, ma'am.
Okay.
Yeah, I think Prof has made a very valuable comments on that. Actually, it's a general review, like review type, you know. And if you have included, you know, how to validate the questionnaire, clinical data questionnaire, if you include some of the what do you call, you know, the review process, you know, and validation process, and it would have been added value to your also review, you know, because this is something less, you know, actually, and evidence-based data, you know, how the data been validated, you know, and approved, you know, and documented. So since you mentioned about various documentation processes, you know, and also include one method, at least one method you could have been, you know, suggested or something would have been added value to your thesis, isn't it?
Yes,
yeah.
I'll consider that, sir. Sure.
Thank you. Thank you very much.
Thank you, sir.
Otherwise, fine.
Thank you, sir.
Thank you, Akshaya, for your presentation and participation at the event. So in combination with prokinetic agent in early developing zebrafish. Samrat Bose.
Yes, am I audible, ma'am?
Yes, yes, you're audible. You may begin your presentation.
Yeah. Okay. So first of all, I'd like to say thanks for providing me this opportunity to present my topic in this great forum.
Excuse me, Samrat, your voice is echoing, and please try to put this slide on slide mode.
Yeah. Now is it okay?
Yeah, the voice is fine now.
Okay. So myself, Samrat Bose, currently I'm doing my research work under the guidance of Professor Dr. Himanshu Maji in JS University, which is situated at Kolkata, India. So this is a part work of my research project. Initial screening, I actually continue my initial screening of a few targeted molecules. Initially, I have taken two non-essential amino acids along with one prokinetic agent. Later on, I'll go for some other molecules, their derivatives and their metabolites. So initially, this is my work. So I'd like to present over here. So these are the contents: introduction, objective, material, methods, result, discussion, conclusion, and references. So before entering into the topic, I just want to mention that why I select this thing. During my literature review, I have found very significant information which is published in different renowned journals, where it is stated that, which is stated by the WHO actually, that most of the developing countries, they are dependent on mostly the traditional medicines along with the food supplements, considering their treatment procedures. So when we are talking about the food supplement is not only the vitamins or minerals, but food supplements also contains the amino acid supplements, and including the dietary supplements, where amino acid is one of the main component. And now we all know that amino acids are basically two types: one is essential and non-essential. These are very common, but still I just find out that considering the both essential and non-essential, both are having certain adverse effect if you consume it. Considering the lysine, it will limit the protein synthesis, acceleration and growth in fish, and metabolic effect of dietary lysines imbalance the protein synthesis, may lead to potential change in physiology. Considering the non-essential amino acids, which is we may not need to consume, but still, if we consider glycine, it is reported that it may produce some toxicity in later stage of development in postnatal neurological impairment, where glycine converted extracellularly the methyl glycoxyl, that is toxic aldehyde, which may lead to diabetes also. Another important fact which I found on a paper that considering the leucine, which is an essential amino acid which we must consume from outside, where different recommendations were there in different countries. Like in Brazil, the recommended limit is 5.6 gram per day, whereas Germany it is 5 gram per day, in Switzerland it is 2.4, in Thailand it is 0.7 gram per day, and so on. So that's clearly indicate the variation in concept of safety of amino acids and allowable limit. There is no specific allowable limit for every country, and considering a specific, if I consider specific research also, there is also a kind of variation. So lack of clinical safety information of amino acid used in general population, as well as an absence of the regulatory tools, overseas dosing of the amino acids in dietary supplements, or protect food products, the current reality. So moving into my topic, that why I select my two initial non-essential amino acid, one is cysteine and L-arginine. Considering the cysteine, it's if I consider its structure, it is a sulfide gel containing amino acid, which is similarly the structure is more similar with the homocysteine, and it is reported in clinical published paper that homocysteine is an independent risk factor for cardiovascular disease. And arginine, it's a non-essential amino acid, and no adverse effect reported, but lack of clinical safety information is there. And it is also sometimes ameliorates many common cardiovascular disorder, but what is its exact effect, that we need to find out. I also select another prokinetic agent because there is a common tendency of these sorts of prokinetic agent to consume it concomitantly with the consumptions of other food supplements. So that is why I consider the domperidone, which is a very common prokinetic agent, which has reported hazardous cardiovascular effect. Because it is reported that it can prolong the QT intervals, and considering to involve the blockade of IKR. So however, it is unclear the effect of the cardiac system fully, therefore I consider this one. So moving into my next slide, as we can see, this is the pathway of arginine metabolism. So here I have selected the main molecule arginine, but as you can see, the source is different. But if I consider only the molecule, then I can screen that whether it is having any effect or not, and later on I'll move on to their metabolites and considering other molecules also. So the main objective of my study is very simple. The study will examine the acute toxicity and cardiac effect of the non-essential amino acids prokinetic agent at different concentration in zebrafish embryo, starting at the blood period. that is 4-hour post-fertilization up to 120-hour post-fertilization. Now the question comes, why zebrafish? Why not other animal model? Because of the genetic similarity of zebrafish with human is huge, as it is reported that 84% of the disease-causing gene, which is absolutely similar with human, as well as the 70% of the zebrafish gene is also similar. And moreover, there is another advantage of zebrafish is that their embryos are transparent. So easily we can observe the development of initial for a few hours. We can easily observe it, document it, also record it, and that can be an evidence of its morphological, considering its morphological study. So the material and methods handling of zebrafish, it is procured from the breeder, and the maintenance has been done at a specific temperature, and also we have to maintain the dark life cycle at 14 to 10 hours. And we have to maintain a specific pH for its breeding purpose. And during breeding, we have to maintain a ratio, specific ratio that is 3:1, and where we'll get the eggs. And the method of observing toxicity and cardiac development on embryos under light microscope, where LC50 study has been performed as per OECD 236 guideline. And under that guideline also will continue the study considering its morbidity and mortality study. And will observe the different phases of development, and which is easily observed under the microscope and can be documented. We all know that this
Mr. Samrat, I think you will have to fasten. You have another 3 minutes left.
Okay. So the gestulation begins at 6-hour post-fertilization, and embryogenesis can be completed within 96 hours. So our main target is to observe the developmental phase from hour 6 to 120 hours. And so let's move on to my next slide. So these are the stages of embryonic development of Daniel radio, which is recorded in my lab. As you can see, all of you, these are the phases, very initial phases from the zygote period, which is recorded at 0 hour. This is a single cell. Then at cleavage period, which is recorded at 0.75 hours. Just 2 cells, as you can see. This is the division or 4-cell stage, as you can see. So these are major stages. Generally, we have to consider this 48-hour stage, that is the hatching period, where we can easily observe their heart rate, and we can record it at images softwares by utilizing the light microscopes and all. So initially, I will go for the LC50 study of arginine along with the LC50 study of cysteine and domperidone. Also, I just mentioned over here the study of arginine, where I performed it at a different concentration. At different concentration, I initially select for the study, and the mortality was recorded at the end of the treatment, and dead embryos were identified in the absence of the heartbeat under the observation of compound light microscope. And several deformities have been identified, as you can see in this slide. That is tail bending, tail body bending, and fully ruptured, and other indications are also there. So this is the result part, where as you can see in the first in the figure 6, that is where percentage mortality study of domperidone has been recorded, and LC50 values are there, which is the values were 3.14 milligram per liter, and it is recorded at different intervals at 24 hours, 48, 72, 96, and 120 hours. And similarly, in case of arginine also, the same percentage of mortality study has been recorded, and LC50 values at different intervals, that is at 24, 48, 72, 96, and 120 hours, it is around 0.38 milligram per ml, and so on. As you can see, as the hours goes on, the value is reducing. If you compare the value with the cysteine is less potent in comparison to arginine, because as you can see, the LC50 values of cysteine is much higher, which is 109.39 gram per ml. Up to, if we consider 120 hour, it is 61.518 gram per ml for cysteine. Now, how I just observe and monitor it and document it and recorded it. As you can see, this is a control heartbeat considering the arginine study. And after applying the drug application, as you can see, it is much lower. So easily we can record it and monitor it and count it by image softwares and other additional softwares backup. So this is the before and after heart rate of the given drug domperidone in zebrafish, and Considering the application of domperidone, these are the observation at hour 1, hour 2, hour 3, hour 4, and hour 24. As you can see, as we are increasing the time frame, the effectivity of the drug is much higher. At 0 hour, only few doses are effective and showing the significant effect. Whereas if you compare it with the 24 hours effect, the heart rate is very less in comparison with the control. Similarly, the same study has been performed on the 6 different doses of arginine, where we also see the effect of arginine is much potent initially from the 1 hour to up to the 24 hours, because in everywhere the dose is showing the significant reduction in heart rate. But in case of the application of cysteine, as you can see, initially the heart rate, the initial dose at 1 hour, few doses are only significant, as you can see, but later on specifically at 3 hour, all the doses are showing very higher heart rate by the application of cysteine. And when I observe at 24 hours, that is quite low, but still the effect is significant. Now, as a whole, if I see that the effectivity of three drugs, that is domperidone, as you can see in comparison to the control, the effectivity is much higher in comparison to the initial few hours, that is 30 minutes or 1 hour, whereas 24 hours it is reducing the heart rate. For arginine, it is in comparison in here also, the heart rate is at hour 3, it is much lower than in comparison to the control. But if you compare at 24 hours, the heart rate it is regaining its heart rate to the normal level. At cysteine also, the thing is exactly opposite. By the application of cysteine, I observed that the heart rates are much higher at hour 3, whereas it's continuously going down and closely to the nearest to the 24 hours, but still the doses are significant. Now, just I want to conclude this presentation by saying that the treatment with different concentration of the non-essential amino acid at different concentration, like 0.25 milligram per ml, 0.30 milligram per ml, and of L-arginine, and considering the prokinetic agent, considering domperidone, that is 0.6 milligram per liter and 1 milligram per liter, causes a dose-dependent decrease in the heartbeat. But the treatment with cysteine at specifically 0.60 gram per liter causes a dose-dependent increase in heartbeat and certain developmental toxicity found, like in domperidone at 3 milligram per liter, at 3 milligram per liter concentration, and 3.4 milligram per liter, embryos of the species have destroyed. At 4 milligram per liter, the tail end bending of the larvae has been observed. At 5 milligram per liter, the notochord bending of the zebrafish larvae has been observed. So after the above study, it can be concluded that the consumption of food supplement containing non-essential amino acids along with the prokinetic agent may create severe cardiac adverse effect, as they are showing the significant cardiac effect on Daniel area, which is 84% genetically similar with human. Okay. So this is my presentation. These are the references. Thank you. Hello, am I audible? Yeah.
Okay, thank you, Bose, for your nice presentation. I would like to see in one slide you mentioned some gram per ml, or is it milligram per ml? May I know?
Yeah.
Somewhere you mentioned in your slide. Go up. Yeah. Come down. Somewhere you mentioned gram. Actually, I think that is supposed to be milligram.
Yeah, sir. In here only, sir. As you can see, this is gram per ml, and these two are milligram per ml.
Down you put gram per ml. Actually, gram per ml is correct or wrong?
It's correct, sir. It's correct, sir.
109.396 gram per ml, you mean?
Yeah, sir. It's correct. Because the sensitivity of domperidone and arginine is much more higher, so that is why we can consider the low amount, but considering the potency of cysteine is much lesser. So that is why I select this dosing form. I repeat this study multiple number of times, and then I concluded that this dosing is okay for this morbidity and mortality study.
Okay, and And can you show the evidence that genetically you have any evidence any in your references?
Actually, there is a paper which is reported in Nature. I have the reference, but if you give me some 5 minutes time, I can show you. At this moment, I don't have that, but I have that in my document. I have that reference.
Mr. or Dr. Bose, Dr. Bose, I think that reference is very, very important, and you must have included in the references, right? First.
Yes, sir. I had that.
Because your whole work is rotating on that particular fact,
Exactly, sir.
That is the main fact. Actually, you are emphasizing here, saying that
right,
the genetically what do you call 84% matching with the so the cardiac effect of arginine maybe and food supplements, must be careful to taking the arginine, probably something like that.
Right, sir.
Yeah. Okay. Any questions from Prof. Nala? Prof, any questions, Prof?
No, I'm okay.
Okay.
Okay. Thank you so much. Thank you so much, session chairs and Samrat, for your presentation. So next we'll be moving on to lunch break. So as we are running short of time, we'll be reducing the lunch break time to 30 minutes. So requesting all the participants to be back after 30 minutes. So that will be the by time 1:30, and the Indian participants, it will be 3:30. You have to be back for your presentation by 3:30. So kindly make a note of it. So please be back after your lunch break. Thank you.
Jazlia, sorry.
Yes, sir.
Yeah, how many presentations still present? Today's one, session one?
Yes, sir. There are more presentations left.
Oh, is it? Okay. Because I'm simultaneously preparing the evaluation report. Probably.
Okay.
I can attach here itself, right? I can attach here also, right?
It will be better if you can email to Shravani, sir.
Okay, I'll email to her probably.
Yeah, because we have two session chairs, and accordingly
we are already 5:30 here actually.
Okay, sir.
So probably you can see that how many presentations. Based on that I can plan accordingly. I think we have
couple of presentations still left, sir. Oh. We have completed only the first 7 presentations yet.
Okay. But
eight presentations.
Here mentioned in the document there are about 46 are there, right?
Yes.
All 46 under pharmacology and toxicology, you mean?
Yes, sir. They're coming under this session.
Okay.
Sir, any difficulty?
No, it's okay. I mean, it's quite tiresome actually, because 46 presentations now actually.
Yeah, quite tiring.
Quite tiresome because actually I had my.
Okay, sir.
I don't know how about Prof. Anas, she's okay with because. Because we see that probably it will be disseminated to a few of the sessions here. No problem, but I can whatever we can do.
We'll manage.
We can do then.
Okay, sure. No problem. Thank you. Hello everyone, welcome back after the lunch break. So requesting all the participants to get ready to continue the sessions without any delay.
Good afternoon, ma'am.
Good afternoon. Session chairs. So once the session chairs are back after the lunch break, we'll begin.
Hello. Hello, ma'am.
Yes.
Mine is presentation as 220.
Can I know your name, please?
Bhupendra Prajapati.
Which session?
All B.
Can I know which session?
Pharmaceutical chemistry and pharmacotherapeutics. Session 2.
Pharmaceutical session 2, the hall B. I'll just allocate you to hall B, sir. This is not the room. This is for pharmacology and toxicology. I'll just move you there. Yeah.
Thank you.
Hello, ma'am.
Yes, Sirisha.
Ma'am, me belongs to same department, pharmaceutical chemistry and therapeutics. Where I have to move?
I'll just move you to that particular room.
Yeah, ma'am. Thank you.
We'll continue with the sessions. Meanwhile, our session chairs will be joining us.
Yeah, I'm here already.
Okay, sir. Thank you so much. So we'll continue, sir.
Shall we?
So our next presentation is by Dr. Kandashri from Hassan Institute of Medical Sciences from India. We'll be discussing about a prospective randomized study of propofol and etomidate on recovery profile.
Hello?
Yes.
Am I audible?
Yes, audible now?
Hope my screen is visible.
Yes, it's visible. Please continue.
Okay. A very good afternoon to all the dignitaries present in this international conference. So my topic on which I have done my study is a prospective randomized study of propofol and etomidate on recovery profile and adverse effects in electroconvulsive therapy. So it is very well known that electroconvulsive therapy requires pre-anesthetic medication before the therapy is started. So starting off with the introduction of my study. So ECT or electroconvulsive therapy involves the induction of a generalized therapeutic seizure that serves to treat and improve psychiatric patients symptomatically. Anesthetic agents are used in the currently practiced, which is known as modified ECT regimen. The essential features of a good anesthetic agent includes the rapid loss of consciousness, effective attenuation of the hemodynamic response. So what happens after ECT is there may be an increase or decrease in blood pressure as well as disturbances in the rhythm of the heart. So an anesthetic agent should be able to stabilize these. Minimal interference with seizure activity. As we know, electroconvulsive therapy involves the use of a generalized therapeutic seizure. So that seizure should be at an optimal level. So the drugs, the anesthetic agent shouldn't interfere with the optimal seizure duration. A successful ECT treatment, apart from providing the adequate seizure, should also ensure smooth recovery from the induction period and have minimal adverse effects for better patient satisfaction and compliance. So the objectives of this study was to compare the two anesthetic agents, propofol and etomidate, in terms of recovery profile. That is, how fast is the patient able to recover from the anesthesia? That is, how like the patient is, how ambulatory the patient is has become, how well the patient can, or how quickly the patient can recover from the state of the sedation to the state of proper consciousness and able to carry on the activities, and also to compare the two agents in terms of adverse drug reactions. So this study was done after an institutional ethics committee approval. So totally around 80 patients were involved in the study after taking informed consent at Krishna Rajendra Hospital in Mysore from October 2017 to May 2019, and mostly psychiatric patients requiring severe psychiatric diseases requiring ECT were enrolled into the study. The patients were randomized into Group A and Group B. So Group A received propofol of 2 mg per kg and Group B received intravenous etomidate of 0.3 mg per kg as induction agents. After administering the induction agents, the blood pressure cuff was inflated to about 50 mm above the systolic pressure in order to isolate the circulation of lower limb and ensure accurate measurement of motor seizure. That's how we measured the motor seizure. So coming to the results of my study. So if you can see that the blue stands for propofol group and the orange stands for etomidate group. And what we found out is that in the age distribution pattern, most of them belong to the age group of around 19 to 25. And on an average, even the older adults of 26 to 35 were also involved in the study in both the propofol and etomidate group. So mostly it was the younger and the middle-aged adults involved in the study. So the older aged adults post 40 and 50 were fewer in number. Coming to the gender distribution. So what we saw was there was a very strikingly high percentage of males present in the study. So in both the groups, around 90 to 92% were mostly males. And females were very less in the study in both the groups. So coming to the diseases that required ECT. So overall, the distribution pattern of mental health disorders were. So as we know, the gray and the blue of 21% each were standing out. These were bipolar affective disorder and schizophrenia, which were the major disorders requiring ECT, and they stood out. The other disorders included substance-induced psychosis and major depressive disorder. So this is I measured the recovery profile by using a score called as modified Aldrich score. So the modified aldrich score had several parameters, including the recovery from the time of consciousness, and also had parameters such as recovery from the time of consciousness. And what we found out was both propofol and etomidate almost had similar timing of similar recovery profiles. But however, there was a statistical significance, which I'll show in the next table. So the p-value was around 0.001. So what we found out was etomidate propofol had a better recovery over the etomidate group. So coming to the distribution of adverse events in both the groups. So blue stands for propofol group. In the propofol group, hypotension and tachycardia or increased heart rate and bradycardia were commonly observed. And the other minor adverse effects included excitement. Whereas in the etomidate group, what we found out was muscle pain was one of the, or the pain at the injection site. Bradycardia was one of the, bradycardia was, and muscle pain was some of the other adverse effects seen in etomidate group. So coming to the discussion, what was observed in the discussion, what we have seen in the discussion is that, just to start off with the introduction, as I had mentioned earlier, so basically it is used to treat severe psychiatric disorders, and there is application of a generalized therapeutic seizure. So an ideal induction agent should be able to allow the patient to allow sufficient duration of the seizure to take place, so that therapeutically the seizure acts with the help of the electroconvulsive therapy. Apart from that, early recovery from the, because what happens when we give the anesthetic agent, the patient may not be able to be ambulatory, and it hinders the recovery. But an anesthetic agent will help, which helps in early recovery, early motor activity, and early recovery from consciousness is always good. So age distribution, as mentioned earlier, 19 to 25 and 26 to 35 years were the maximum age groups that was represented in my study. And the current findings were coherent in a study conducted by Tan et al. As well as Manjula BP et al. In the gender distribution, what we found out were 73 subjects were male and just 7 were female. And what we found out is that a male predominance was observed in alcohol use disorders, and female predominance was observed in depressive disorders and neurotic disorders. Similar findings were seen in a study conducted by Zahavil et al. And Balaji Dontu et al. So majority of the subjects who received this ECT belong to the schizophrenia group and bipolar affective disorder group. And the rest were depressive disorders, Parkinson's disease, and substance-induced psychosis. These findings were also recorded in a study conducted by Ozakanovic et al. So the modified aldrich score, as I mentioned earlier, was used to assess the recovery profile between the two anesthetic agents. The components of the score assessed were activity, breathing, circulation, and saturation. These were the components in the modified aldrich score. The score was assessed over a period of 15 minutes. So what we found out was there was a high statistical significance with p-value being 0.001. What we found out was propofol had a better recovery over etomidate group. So in the present study, coming to the adverse effects.
You have another 2 minutes to conclude.
Yeah. I'll conclude fast. Okay. So what we found out was that hemodynamic stability was found to be better preserved with etomidate. There was higher incidence of hypotension and bradycardia with propofol. So hemodynamic instability is more common with propofol, but etomidate had a very good hemodynamic stability. In the etomidate group, the adverse effects were pain at the injection or muscle pain or myalgia was also seen, and the recovery also was very late. So conclusion, propofol is assessed to have a better recovery than etomidate. Rapid recovery and restoration of normal cardiorespiratory functions after ECT is highly desirable, and it will put a less strain on the working staff. It also helps early oral feeding and thus reduces the requirement of intravenous fluids. Based on this parameter, propofol in the present study seems to be better alternative to etomidate as induction agent in modified ECT. The use of induction agent etomidate, on the other hand, has relatively less adverse effects compared to propofol, Hence, etomidate may be an alternative option for use in modified ECP. Thank you.
Thank you for your presentation. Session chairs, any question?
Okay, I would like to ask you one question here. Actually, if you compare propofol and etomidate,
Yes, sir.
propofol has the half-life of 2 to 24 hours, you know.
Okay, sir.
And how could you get in your findings that propofol is the better recovery than etomidate?
Sir, propofol.
Compared
Sir,
to
propofol is preferred in daycare as a daycare anesthesia, sir. Like in case of any daycare procedures, because
that's true. But actually, if you compare with the propofol and etomidate, both are actually short-acting general anesthesia.
yes, sir, short acting there. Yes,
and their
sir.
recovery is very faster, actually, in minutes, I can say.
Yes, sir. Sir, I'm not saying that etomidate is highly like, you know, it has similar recovery times as that of propofol.
Correct, but propofol is protein binding, highly protein binding, and it will be metabolized by the liver enzymes, actually.
Yes, sir.
So if you see that case, how could propofol will be having better recovery than etomidate in your study?
Sir, in my study, sir, it was found that, sir, despite the pharmacokinetic characteristics and the properties of propofol.
No, because the pharmacokinetic characteristics have been already developed, you know. If you see propofol
Yes, sir.
and etomidate, they're already miscellaneous general anesthetic agents, which are
Sir, that's what I just want to make it clear, sir. It's not like etomidate has a bad recovery. The recovery time periods were very similar, sir. In fact, I'll show the table, sir.
Yeah, you're right. Because when you mention the significance, no, because
yes, sir. Only in the last minute, if you can see, sir, there is some change. The rest, it's similar, sir.
Actually,
it is
9.17 and 9.1, there is no difference, actually.
Yes, sir. There is no difference, but
How could you say significant then?
sir, but still, according to the statistical analysis, this was considered to be significant, sir. But the time periods of recovery were very similar.
Can't just check on that, actually, because, you know, when we should not misinterpret.
Yes, sir, I do
the
understand. Yes,
yeah, when
sir,
we have the data with us, you know, we should not misinterpret the data, actually. You know, you can see that significant is something which is having a difference of, let us say, if your time of recovery is in minutes, you see, then
yes,
the significant of difference will be actually plus or minus minutes. For example, 2.67 plus or minus 1 minute, definitely a significant difference.
Yes,
if in
sir.
hours, if minutes is no significant, you see, because, you know, when we compare 1 hour is 60 minutes, so if you say 2 or 3 minutes plus or minus, then we not significant, something like that. So kindly see that, you know, the interpretation. Otherwise, it's fine. Probably.
Yes, sir.
That's a question, actually.
Okay.
Probably rather doubt, actually, you know, this is question.
Sir, I will recheck with it, but when the statistical computation was done, this was the result that...
See, statistical computations will be done by software and all. Actually, whatever the data you input into the software, that will be done. But we need to interpret the data properly, correctly.
Yes, sir, I agree.
Relevant to the existing pharmacokinetics data, because propofol and etomidate are established existing data. One is
yes,
the protein, both are protein binding, but
yes,
propofol is more protein binding than the etomidate. So when it is protein binding, the recoveries will be, see, both are short acting.
Yes,
so
sir.
The elimination will be a bit later than the etomidate. So in such cases, no, and etomidate got another problem because it is a stereochemical molecule. So that has got another issue also with R and S isomer, you know, combination of R and S isomer and all those things. So
yes,
chemically, if you think about that, you know, anyway, it's okay. I mean, it's a presentation wise, it's fine. It's okay, fine.
Thank you, sir.
Okay, thank you, sir. Thank you for your valuable comments.
Hello, doctor.
Hello, ma'am. Good afternoon.
Can you just put your last slide, which is the conclusion slide?
Yes, ma'am. Ma'am, is my screen visible?
No.
Oh, sorry. Can you just share your screen again?
Yeah. 1 minute. Yeah, I'll just put the conclusion slide, is it?
Yes, only the conclusion.
Yes, ma'am. Yes, ma'am. Just a minute.
Okay. It's actually it's just catch my okay.
Yes.
Yeah, it's just catch my attention. Okay, I do understand that you are looking into the comparison between the propofol and the intermediate. Okay, but when I went through, because the conclusion is like the whole thing about your findings, which is you're answering your objective, and then this is what you are going to suggest, maybe for your future practice, I assume. But here it looks like there is no how to say you by reading this conclusion, it looks like either you use the propofol or
Ma'am, I got it. There is an ambiguity, ma'am. I understand. It's not very clear.
Yeah, it is not clear because you know, like I'm telling you, let's say the second last sentence and the last sentence, it looks like it is crossing or contradict each other.
Yes, ma'am. Ma'am, yeah, I would just like to answer this, ma'am. So, ma'am, regularly the agents used as a pre-anesthetic medication in ECT are thiopentone sodium and propofol, ma'am. So on etomidate, not many studies have been done, and etomidate is known to be a cardioprotective agent. And in case of if patient has any coexisting cardiac morbidities, and as I mentioned earlier, ECT also causes unstable hemodynamic changes like tachycardia and things like that, which etomidate will make sure it will stabilize and it will not cause. So that's why etomidate was compared with propofol. So what I can say is it depends again on the discretion of the treating psychiatrist, ma'am, based on what the patient's general condition is. In case if the psychiatrist feels that the patient is unfit for propofol, like he may have some cardiac comorbidities, or if the patient is not hemodynamically stable, then he can go for etomidate even. And what I can say is etomidate does not have a bad recovery. It has similar recovery as such as compared to propofol. But that's how, with respect to the cardiac characteristics, if you take cardio-stabilizing properties, etomidate is emerging and it has a very good value in this with respect to this area. So it can be used. That's what I want to put it across. It's an alternative agent.
Yeah, maybe the arts of writing a conclusion.
Yes, ma'am.
I mean, it can be in a way that.
Ma'am, propofol is favorable in one way, and etomidate is favorable in another way.
Yes, so, I mean, I do understand because when we are reading this, it looks like it's actually both okay. You understand because it's already across each other. So maybe for future reference, sometimes the way on how we write a conclusion, it should be clearer. For example, I mean, this is just my suggestion.
Yes, ma'am, definitely I'll take it.
Let's say if you want to say that for etomidate, it is let's say it has more cardioprotective
yes, ma'am,
compared to propofol. So maybe you can say that I mean by showing, for example, by showing a cardioprotective effects of etomidate, you understand?
Yes,
so
ma'am. Yes,
an alternative option. For
yes,
example,
ma'am. And ma'am, even if you had seen in the adverse effects section, etomidate has very fewer adverse effects compared to propofol.
Yes, correct. But only the way on how the conclusion written, you understand?
Yes, ma'am. Yes.
If people, because sometimes when people want to look into very fast,
got it, ma'am.
Certain things, like.
Yes, ma'am. Got it. I should be very crisp and clear, ma'am, with what I convey.
We just look into the objective, then immediately we look into the method.
Conclusion. Yes, ma'am.
Whether method is like corresponding to the objective, and
yes,
then doing this, and then we look into the results, and then results we will relate with the conclusion which is answering back your objective.
Yes,
so I'm just curious on this part. It's just a little bit on us on how to write
yes,
the so that people may not, are thinking that, oh, it's actually.
Yes, ma'am. Got it.
You understand?
Yes, ma'am.
So meaning now in the current practice, so in the psychiatric ward for the ECT, so it is more into propofol or started to use the etomidate?
Ma'am, conventionally, propofol and thiopentone sodium are only used, ma'am.
So
As pre-anesthetic induction agents, they are used. So but etomidate, ma'am, they have not many studies are there of use of etomidate as an induction agent before ECT. Okay, etomidate, so, what we found is that it has a very good cardioprotective and cardiostabilizing.
Sorry to interrupt.
Yes,
and we are running short of time.
Ma'am, so etomidate can be used as an alternative, ma'am. That's what my study tries to convey.
Okay. All right then. Okay.
Ma'am, I hope I made sense. Yes, ma'am.
Okay,
thank you. Thank you.
Yes, our next presenter, let me know if you're available for the QA because we have received a video presentation from Golnaz Hemati. Is Golnaz available here? Okay, so we'll be moving on to our next presenter, Dr. Manjula MJ. Dr. Manjula MJ. Requesting everyone to be on time because we will not be able to give you another chance to present. Dr. R.A.M. Jain Nachia.
Yes, madam. I am present here. My name is Dr. R.A.M. Jain Nachia.
Yes.
I'm here.
Okay.
Yeah. Can I?
Yes. Dr. R.A.M. Jainaf is from B.S. Abdur Rahman Crescent Institute of Science and Technology from India, and she'll be presenting on the study on drug therapy problems in hypertensive patients with CVD cardiovascular diseases at multi-specialty teaching care hospital from South India, a prospective study. Please begin, ma'am.
Yes, ma'am. So, my name is Dr. R.A.M. Jainaf Nachia. I am from India, and my university B.S. Abdur Rahman Crescent Science and Technology, Vandalur, Chennai. Can I audible, ma'am? Can I audible?
Yes, you're audible.
Yeah, it's my top title is a study on drug therapy problems in hypertensive patients with cardiovascular disease at multi-specialty teaching care hospital, South India. This study is a prospective study. Hypertension is an increasingly important medical and public health challenge because of its high frequency of concomitant diseases like cardiovascular, cerebrovascular, kidney disease, and risk factors. According to WHO, is estimating that hypertension causes 1 in every 8 deaths. It's making hypertension the third leading killer in the world. Globally, there are 1 billion hypertensive and 4 million people die annually as a direct result of hypertension. This present study got approval, ethical committee, ethical clearance from Annamalai University, Rajamuthaya Medical College, Chidambaram, Tamil Nadu. And this present study aim is to analyze the drug therapy problems in hypertensive patients with cardiovascular diseases at multi-specialty teaching care hospital. Main objective is to identify and categorize the drug therapy problems of hospitalized hypertensive patients according to PZNE2, that is the Pharmaceutical Care Network Europe, and to identify the risk factors and causes of DRP. DRP is a drug-related problems as well as we can say drug therapy problems also. And it is a drug therapy problem. It is an event or circumstance involved in drug therapy that actually or potentially interferes with desired health outcomes. This present study, plan of the study, first we have collected the designing of present study and patient selection and identifications for the study based on inclusion exclusion criteria. And the study is seeking approval from ethical considerations. And there is a group of the patients, only this study belongs from the observation study. And then we have done a pilot study. Then after we collected the data and final analysis and report we have generated. And the research, coming to the research methodology, the study setting is Raja Muthiah Medical College Multi-Speciality Hospital, Annamalai University. The study period is 12 months. The study design is prospective and observational study. Total sample size 75 hypertensive patients. By using this sample size calculation we have used for this present study, and total 678 patients were visited in this study site with the diagnosis of hypertension with various concomitant diseases. In this 475 patients not eligible in this study, 203 number of the patients not eligible for inclusion criteria. In the month of first month, they were dropped out. 188 patients they were dropped out in the first month. In the second month, 212 patients were dropped out, and finally, the number of 75 patients were completed the study successfully. This inclusion criteria, this present study inclusion criteria, minimum 6 months of the hypertension affected with hypertension, and the patient prescribed 1 or more hypertensive agents, and 35 years and age and above the patients included in the study. Both genders were included, and these are the concomitant diseases patients were selected, like hyperlipidemia, hypothyroidism, hypertensive heart disease, hypertensive retinopathy, hypertensive nephropathy, diabetic foot ulcer, bronchial asthma, myocardial infarction, and coronary artery disease. They were included in this present study. And particularly this study, patients were willing to participate and they were provided informed consent, and patients able to understand English and Tamil language only included in this study. And the exclusion criteria, patients who are pregnant and lactating women were excluded, and the patients having psychotic illness not included, and the patients having chronically ill-looking patients not included, patients unable to give proper information of data collection form not included, patient not willing to provide consent form not included, patients who are discontinued from the treatment and not taking medicines, patients not included in this study. And coming to the results part, there is a gender-wise distribution in the present study. Male has a more predominance in this study, but at the side by side I am discussing with the others articles also. When I have discussed with the others articles, means others study, they were the same study. It is related with one Indian study also. Male have more the gender. Male is involved more in this study, but female was very less. Part of the females were included in this study, but few were the same study. They were considered the other side. Female were more. Present study, when compared with female, males are very higher. Patients were included. When age-wise, non-elderly, 73.3% of non-elderly patients were higher. And coming to the education-wise, in that uneducated, 46% of the patients were uneducated. And when coming to the polypharmacy wise, 70% of the patients were received 5 or more medications, and 58% of the patients were hospitalized more than 10 days. Social habit wise distributions, 58% of the patients were addicted to alcohol and smoking. And history wise, 45% of the patients, they have the family history of the hypertension. And the monthly income wise distribution, 82 patients have the less than 1,500 monthly income. And the residency wise, the rural patients, they were coming from the rural part. 84% of the patients were coming from the rural part. And economical status wise, 70% of the patients, they were very low economical status. And 54% has been coming to the duration of hypertension. They taking the treatment. 54% of the patients, they were 10 years, they have taken the treatment for hypertension. And hypertension along with the CVD means the coronary vascular disease complications. There is a stroke. 29% of the patients were have the stroke, and 16% of the patients were MI, and 12% of the patients were CHF, and 9.3% of the patients were cardiac arrhythmias. When coming to the comorbidity wise, there is no complication. That means only hypertension will be there, but there is no complication. Only 25% of the patients were no complications, and very highest concomitant diseases in the rural part, in the Chidambaram, that is Tamil Nadu. Hypertension with type 2 diabetes mellitus patients, they have 26% of the patients, they have more concomitant diseases like hypertension with type 2 diabetic mellitus. And when that in this present study assessed on categorization of drug interactions of drug-related problems, when prescribed with aspirin with clopidogrel, one patient got a severe abdominal pain. When prescribed with furosemide and metformin, the one patient had a severe abdominal pain and involuntary urination. And two patients were suffered with when prescribed aspirin and enalapril maleate. 2 cases were suffered with constipation and blood in stools. When prescribed aspirin and atorvastatin drug, 2 cases
Ms.
they were
Jainab, sorry to interrupt. Ma'am, you have another 2 minutes to conclude.
Okay, madam. Okay, madam. And when that medicines prescribed, these all the number of the patients when the medications prescribed, they have they had a drug interactions, and means assessed about categorization of the drug interactions, and they had a body itching when prescribed enalapril and furosemide. They had a renal impairment. They had when prescribed of aspirin drug, they had a renal impairment and creatinine clearance of less than 30 ml per minute. And identified and categorization of there is that technical drug related problems and clinical drug related problems we have identified and categorized. And adverse drug reactions, they had means when prescribed ACE inhibitor, they had adverse drug reactions of dry cough. 3 patients had suffered. And prescribed prednisone and ferrous supplement metformin, they had constipation, body itching, and involuntary urination. The causes for DRP also assessed and categorized. There is that in this present study, 89% of the patients, there is they were not awareness of the medicines to take caring of themselves, and also they are most of the patients, they are forgetting to take their medications. This is the main causes we are observed. The reduction of causes for DRP. And study limitations. This study very small size, and tablets, only we have means concluded for drug related problems. We are unable to check that injectable preparations. Why? Because many of the patients, they have re-hospitalized also. We did not take their data. And coming to the conclusion part, in the present study, it has seen that for essential hypertension treatment seem to be inconsistent with the international hypertension guidelines like GNC8, and diuretic happens to be cost effective. It has been pleased to use monotherapy drug. And the number of drug-related problems among patients with hypertension is relatively high. These DRP were associated with poor blood pressure control. The study showed that 97.7% of the patients have DTP, which is too high. Mainly technical problem, we have observed that there is a ORAC symbol missing, physician signature is missing, but many prescriptions, they were written like continue the same medicines. Like in this sentence only it has written but there is no medicines are prescribed, not written. They have written only continue the same medicines, the previous page. Like this one sentence only they have written. The study shows that 21% of patients have CDRP, which is very low. They have that drug interaction, area, non-adherence, drug without indication, inappropriate dosage were identified, and significant association were found between the elderly and the occurrence of adverse reactions, between renal impairment, the occurrence of drug choice problems and overdosage found. The patient counseling in time was detected, prevented, and well managed in post intervention study and reduced the ADR occurrence. The Indian Tertiary Care Teaching Hospital is one of the most powerful health professionals, and they are needs to sensitize and train. The monitoring of ADR and their effective management is a reasonable possibility in Tertiary Care Teaching Hospital, and this could improve the quality of life of hypertensive patients. The cost of hypertension care are mainly dependent on the expenditure on blood pressure lowering drugs. The treatment of hypertension with diuretics or beta blockers was more cost effective than treatment with other hypertensive agents like ACEI and calcium channel blockers and adrenal receptor blockers. And this study, I wanted to thankful to UGC BSR Fellowship for given opportunity to complete this study successfully. And thank you, madam. I have completed my work.
Yes. Thank you, Dr. Jainab. Session chairs, any questions?
Doctor, yeah, good presentation. Yeah, I would like to ask you one thing. You got, can you show me your ethical certificate when you got the approval? Can you show the certificate that you have shown, right? You got the, to conduct the study, you got the ethical certificate in medical college, right?
Yes, sir. We have got this certificate. Actually, this is my research study, PhD study. I have started my PhD 2013. I have completed my studies 2017. And one of my co-author, Dr. S. Paramila sir.
No, please. I have not asked any.
This is a certificate, sir.
Yeah, you got ethical approval in 2013, but you conducted the study, it looks like 2020-21, you mentioned the date.
Actually, sir, one few part of the study recently we have conducted. My co-author he has conducted, and we both involved in this study.
Yeah, because I'm not sure how the ethical certificate is valid, because the protocols keep changing, every time. From 2013, the protocol of hypertension treatment is different from the protocol of treatment of hypertension maybe now, because there are new drugs are added up with safety and efficacy. So that is one question, question number one. Question number two, I wanted to ask you on the population, because out of the population of 75, you took major patients which are having smoking and drinking habit. So may know any significance? You wanted to know a reason? You choose deliberately, or actually it has come the outcome on serendipity? Do you understand my question?
Yes, sir. Yes, sir. I got your questions. That means you are asking that 75 patients they have a completed study, but their social habit is very more. It seems you are asking that.
It's not a social habit. How can the low income people having a social habit? Because that actually they're addicted. It looks like smoking and drinking. Hypertensive patients usually they're aggravated patients there, And how significant your study then in that perspective? And moreover, you conducted in a span of many years. It looks like some data will be earlier, 2013. Now the current data. So probably I think you may need to relook into adjust into the protocol, and probably may do something.
Okay, sir.
Because your conclusions, makes more sense. Your conclusions make more sense than to fit into current scenario, current protocols, and current therapeutic drugs which are available more or less, for the treatment of hypertension, and already patients who are suffering, having such habits, with hypertension.
Yes, sir.
Thank you.
Yes, sir.
Okay. Hello, doctor.
Yes, sir.
Yes. Should I call you Dr. Nachia or?
Yes, ma'am. Zainab.
Dr. Zainab.
Yes, ma'am.
Okay, I mean, it is a good study. I mean, it is good effort, and then this area is looking into the drug therapy or drug related problems is one of the things that we used to look, especially when those work as a pharmacist in any wards. Okay, my suggestion for future reference. I mean, since like the title for your today is like you're looking into the drug therapy problems, right? You're looking into the drug therapy problems for hypertensive patients. Okay. The way on the presentation should just focus on the drug therapy problems. I do understand you wanted to show the analysis of the statistic for your socio-demographic medical data to show that the distribution is significant or not significant. But for this kind of study, the socio-demographic, you can just present it as a descriptive, and then zooming directly into the drug therapy problems among the hypertensive patients. Okay.
Okay, ma'am.
So if you do that, then you will catch more attention, and then people are more focused.
Pardon, ma'am, I'm not getting your point. What, ma'am?
Ma'am, am I audible?
Yes, ma'am. It's audible, ma'am.
No, I mean, can you hear me just now?
Yeah,
Yes, ma'am. Yes, ma'am. We can hear you.
Yeah, can hear, ma'am.
Okay. So I mean, this is just my suggestion for future, because I have a feeling that the one that you presented is actually a part of your studies.
Okay, ma'am.
And then you put some of the cost and everything. No need is actually, because your study is just looking at the DTP. So just focus on the DTP. Okay. I mean,
okay,
and this is what we wanted to look into. Okay, so by showing that, then we will have more, the benefit will be later in future. So for those healthcare professionals, okay, mainly the doctors and the pharmacists, when they want to prescribe, so we can look into, to prevent any future drug-related problems. Okay.
Okay, ma'am. Okay, ma'am.
Okay.
Okay, thank
you so much. Thank you so much,
Thank you so
session
much,
chairs.
ma'am.
Thank you, Jenna, for your presentation. So we have our next presenter who have joined here. So I would like to welcome Dr. Manjula MJ from Hassan Institute of Medical Sciences from India, who will be discussing on comparative study between the safety and efficacy of phenylephrine and methyltramine in the management of hypotension induced by spinal anesthesia. Dr. Manjula?
Can I share my screen, ma'am?
Yes, please.
Yes. Can you hear me now? Is it audible? Yes.
Ma'am, are you able to see my screen, ma'am?
Yes, your screen is visible, but your voice is breaking.
Okay, am I audible now?
You may begin, but I don't know how far we'll be able to continue the few. I mean, can you please talk and see?
No, ma'am.
Can you begin and see? Yes.
Okay, shall I
better.
begin?
Yes.
Okay, today I'll be presenting my study on comparative study between the safety and efficacy of phenylephrine and methyltramine in the management of hypotension induced by spinal anesthesia. To introduce with the incidence of hypotension following spinal anesthesia due to the preganglionic sympathetic blockade, which will lead to vasodilatation and peripheral pooling of the blood in the lower limbs, which will reduce the cardiac preload and also the output. And bradycardia produced by the blockade of cardio-axillary nerve fibers of upper thoracic spinal cord segments, that is from T1 to T3, can treated with vagolytic drugs. For example, atropine, but often the response is erratic and undesirable. And also the volume expansion can be done with crystalloids or colloids infusion in order to manage this hypotension. Various vasopressor agents have been used in prevention of this spinal anesthesia-induced hypotension. For example, ephedrine and norepinephrine was the first agent to be used successfully to treat this purpose. However, the prophylactic administration of ephedrine in spinal has no longer been advocated due to the adverse effects of this. And mephentermine has been used at our institution. And this current study had been taken to compare the two vasopressors, that is phenylephrine and mephentermine. Phenylephrine, being an alpha-1 blocker, it acts by constriction of vessels, whereas mephentermine, being an indirectly acting sympathomimetic, it will enhance the release of these catecholamines and thereby the actions. Aims of my study was to know the safety and efficacy of mephentermine versus phenylephrine in spinal anesthesia-induced hypotension, and also to know the adverse effect of mephentermine and phenylephrine. The materials for the patients who were posted for lower abdominal surgery at Sri Chamarajendra Hospital, which is attached to our Hassan Institute of Medical Sciences, Hassan. The inclusion criteria, we included the patients aged between 18 to 60 years who were belong to ASA 1 and 2, and the patients posted for infraumbilical and lower limb surgeries, and the patients who were ready to give the written informed consent. And the patients we excluded were the patients with known case of ischemic heart disease, the patients under ASA 3 and 4 grading, patients who were allergic to either of the drug, and the patients who were not willing to give the informed consent. This study was a longitudinal interventional open label study. The allocation was simple randomization, but it was an interventional study, and the purpose of this study was the treatment purpose, and the endpoints of classification were safety and efficacy. Here we randomly allocated the patient into two groups. Group A with the IV bolus phenylephrine 100 microgram, and it was repeated for five bolus doses, and we did not give beyond the 500 microgram. And IV bolus of mephentermine was 6 milligram. Fall in blood pressure less than systolic blood pressure less than 20%, or an absolute value of less than 100 mm of Hg was defined as a hypotension, and the number of bolus doses given in either group and the time taken for recovery of this hypotension was measured. Then the blood pressure, mean arterial pressure, heart rate, ECG, oxygen saturation were recorded as a baseline before giving the spinal block, and at every 2 minutes after the administration of spinal anesthesia for 20 minutes, then thereafter the procedure for 5 minutes till the end of the surgery. Statistical analysis was done by using SPSS software. The p-value less than 0.05 was considered as a significant value. Here are the demographic details of our patients. Out of 100 patients I recruited, 17 were from Department of Surgery, 17 from the orthopedics, and 16 from the gynecology. We did not include any pregnant females who were posted for cesarean section. The mean age of our patients was in phenylephrine, it was 44.78 with the standard deviation 9, and mephentermine 45.08 with the standard deviation of 9. Here we did not have any significant difference between the basic basal parameters, either hemodynamic or the demographic details of the patients. Here are the distribution of comorbidities between the two groups. Here in the hypertension group, we had 14 patients in phenylephrine group, whereas 12 in the mefentremine, and diabetic patients 2 in the phenylephrine and 1 in the mefentremine group, and hypertension and both hypertension and diabetic patients were 6 in the mefentremine, whereas 5 in the phenylephrine group. And here the distribution of duration of the procedure between both the groups. Less than 40%, 40 minutes was the duration of surgery in phenylephrine group 2 patients and mefentremine 1. Between 40 to 50 minutes of the surgery were distributed 25 in the phenylephrine and 26 in the mefentremine group, whereas 15 group in the phenylephrine and 16 in the mefentremine group were done the procedure between 51 to 60 minutes, whereas more than 60 minutes of surgery conducted over 8 patients in the phenylephrine, whereas 7 patients in the mefentremine. Again, we did not have any difference between these two groups in with respect to the duration of procedure. The mean with standard deviation of the duration was 53 minutes, 53.5 minutes in phenylephrine, whereas 52.2 in the mefentremine. And the distribution of number of bolus doses required for the recovery of the blood pressure was here 82% of the phenylephrine group had required only 1 dose, whereas mefentremine group we had 66% of the patients, whereas in the doses, bolus doses required were 2 among 8 of the phenylephrine patients, whereas 13 of the mefentremine group had required 2 bolus doses. And when we observed, the 4 patients in the mefentremine group were required 3 bolus doses of mefentremine, whereas only 1 patient had required 3 doses of mefentremine for recovering the blood pressure. And the mean recovery time in the phenylephrine group was 6.8 with a standard deviation of 3.5 minutes, whereas mefentremine it was 7.42 with a standard deviation of 4. Here we observed the significant difference between the number of bolus doses required and the recovery time between these two groups. And when we observe the comparison of fall time and the recovery time between these two groups, the fall time was 27.8 and 27.7 between those two groups, whereas the recovery time was 6.08 in phenylephrine and the mefentremine it was 7.42. So here we observed the
Dr. Manjula.
Yes, ma'am.
Dr. Manjula, excuse me, you have another 1 minute left.
Sure, ma'am. Here we observe the significant difference between recovery time of the patient, whereas we did not observe any statistically significant difference between intraoperative variation of heart rate, the systolic blood pressure or the diastolic blood pressure. Then here we also observe a significant difference between fall in time between the ASA group 1 and group 2. To discuss with I'll just come to the conclusion of my study. Here we observed the efficacy of phenylephrine and mefentremine in controlling the hypotension due to spinal anesthesia was almost same, but the recovery time required by the phenylephrine is comparatively lesser than the mefentremine, and number of bolus doses required by the phenylephrine was comparatively lesser than the mefentremine, and phenylephrine is comparatively faster acting, and change in the heart rate followed by the mefentremine bolus dose is more compared to phenylephrine. Hence, the phenylephrine is better drug in the patients who are prone to develop increased heart rate, and also in the patients with ASA2, preferably phenylephrine had showed better outcome than the mefentremine. Thank you.
Thank you, Dr. Manjula, for your presentation. Yes, session chairs, any questions for Dr. Manjula? If not, we'll move on next. Any questions?
Yeah, I think no
Yes.
questions. Yeah, I think.
Okay. Professor Nahla, shall we move further?
Just a very quick question.
Yeah, sure.
So Dr. Manjula, I mean, could I just know that in your current practice, I mean, which of those vasopressor that you use?
Pardon, ma'am?
In current practice, which of those that currently being in use?
Usually in our institution, we use mephenteramine, ma'am. Commonly used is mephenteramine. And when I went through the scientific evidences, I found phenylephrine used as a intramuscular or intravenous IV infusion, ma'am. Very less studies were I found of this bolus dose. That's why this study had taken, ma'am.
Okay, I mean, do you think that your study may change your current practice?
Yes, ma'am, because as we observed, the elderly patients were more prone to have the spinal anesthesia induced hypotension and bradycardia, ma'am. And whereas this mephenteramine, it has caused both the pharmacological effect on hypertension and also the heart rate. Whereas phenylephrine had comparatively better cardiac effect compared to the mephenteramine, ma'am. So in the patients who are having these cardiac diseases and also in elderly patients, better to use phenylephrine over the mephenteramine and other drugs, ma'am.
Okay, so meaning currently your hospital is using phenylephrine, is it?
No, ma'am, mephenteramine they were using. Now we had started, ma'am, after conducting this study.
I mean, no, I just want to get it right. So now the current practice is using the mephentermine.
Yes, ma'am. Only in selective patients, ma'am.
Okay, and then the outcome of the study may change the practice whereby the phenylephrine will be used more.
Yes, ma'am.
Okay, all right then. And this one, the patients when they've been, been put, after the spinal anesthesia, they have been informed that you are conducting the study and
before itself, ma'am. Before giving the spinal itself, we had explained this mephenteramine. We had explained them that usually, if you develop this decreased blood pressure after giving the spinal anesthesia, we used to give this drug mephenteramine. But as a part of study, we are comparing two drugs. So you might be administered with the newer drugs. And we had also explained them what will be the outcome, what will be the adverse effect. Only after taking informed consent, we had done the study, ma'am.
Okay, all right then. Okay.
Okay,
Priscilla madam.
Yes.
Priscilla madam, I am Dr. Kumudavalli. Still, I was waiting in the room C. Actually, that invitation, it was in C only. Now only they have changed it here.
Ma'am, your presentation hasn't come yet, so you can wait for it. Okay, we are. Thank you, Dr. Manjula, for your presentation. So next, I would like to invite Sunil K. Prajapati from AIMST University from Malaysia, who will be discussing on the knowledge, attitude, and practice of needle stick injuries among dental and nursing students. Welcome, Mr. Sunil Kumar.
Thank you, madam. My slide is visible.
Yes, it's visible. You may begin.
Okay. So very good afternoon to all of you. Thank you for giving the opportunity for presenting on this renowned platform. My name is, as already pronounced, my full name is Sunil Kumar Prajapati, and I am the supervisor of this research work. And my co-investigator means my student. It is actually undergraduate research project. So my student name is Miss Tae Jing Jin. Actually, she's working, so she asked me to present. So I'm presenting this. So the title of this research work is Knowledge, Attitude, and Practice of Needle Stick Injury among the dental and nursing student of the same university where it is conducted. That is AIMS University, Malaysia, right? And we need to firstly understand what actually mean by needle stick injury. So any injury which is done by syringe, which is accidentally inserted or penetrated in the skin of any individual is called needle stick injury. I will use NSI as a short form rather than speaking this long word. So why it is matter of concern? It is matter of concern because once it is used, it is having may have the pathogen. So if the pathogens are there and inserted to the healthy individual, then it may cause the infection. And as per WHO, the report saying 20 different blood-borne pathogen can be found in the needle stick injury or NSI. That could be hepatitis B, C, HIV, and list is long. So we aimed to find out or to assess the knowledge, attitude, practice of this injury on NSI among dental and nursing students of the same medical institution, Williams University, so that we can find out that how prone they are for the infection, and these dental and nursing students are encountering most of time during the administration or care of the patient. So we set the objective, that is to find out the demography of the participant, to assess the knowledge, attitude, and preventive practice of NSI, to identify whether NSI is reported or not, and to find out what is the causative factor of NSI among the students. Right? Why we are conducting this all? So justification is mentioned because a very few data is mentioned for this NSI, and this study was conducted in other countries. At that time, it was not very much published in the Malaysia. So we wanted to extract the data in at least that particular reason, so that we can find out the attitude and practice of the dental and nursing student. So let's talk about the methodology firstly. So the methodology having many aspects, out of them the study design, which is cross-sectional, and questionnaire was designed ethically, of course, permitted, and study site was the same, Ames University, and sample size was 310. It was calculated by online software, that is TrioSoft. We having the sampling method also means we actually conducted this as a stratified convenience. So we prepared 210 printouts, and we also print out for the informed consent. We got the informed consent also. Study duration was 2 semesters, nearly 2 semesters. It is actually practically it was 8 months, and it was conducted in 2018 to 2019. As I told, it was of 2 semester study as a part of their undergraduate research program. So if participants are participating to fill this questionnaire, they are taking just 15 to 20 minutes. Data source was Ames University itself, their students who participated in that study. As the part of the instrumentation, we are requiring the questionnaire, that is print out, we requiring the A4 size paper, of course, and the laptop. And statistical analysis we did on the Excel sheet, on the SPSS software. We applied the Cronbach alpha for the reliability. We did the descriptive analysis. We applied the Spearman rank correlation. We find out the data on the basis of chi-square method also. So to design the questionnaire for the knowledge part, we designed 13 questions where we conducted the descriptive analysis. There was a question of yes, no, I don't know type, maybe multiple response type, maybe multiple choice type. For attitude also, we designed the questionnaire, which was of 5-point Likert scale, means strongly agree is giving score 5, and strongly disagree was giving score 1. And there was a criteria set which was calculated as a poor, moderate, or good attitude. Accordingly, we also designed for the practice same criteria: poor, moderate, and good attitude for the needlistic injury. Study population, as I told, that the sample size 310 was the sample size, and this was taken from the dental surgery, means dental student and nursing student. Their further segregation is also mentioned. So inclusion criteria is just that much, that the student should belong to dental and nursing of the same university, and the inclusion criteria is dental and nursing student of the same university. The exclusion criteria is if they are filling the incomplete form, if they are belonging to other faculty, if they are not willing to give the informed consent, then they will not be included in the they was not included in the study. Validation we have done. We did the construct validation, means reliability we got 0.791. We also conducted the content validation, phase validation with the help of different experts for that questionnaire. So this is our layout for the research study. If we talk about the result, then in this conference, I am presenting a very few data for this. The whole study, it is very hard to present in 10 minutes. So you see, we decided for 310 sample size, but due to dropout and other parameters, we finally got the total 285 number of the participant, that is 92%. So if we talk about the demographic characteristic, then you can see very easily it is mentioned here. Gender, the females was higher in the study population, right? The males was less, the number is 66, the percentage is 23.2. If we talk about the ethnicity, then mostly student was of Chinese category, then the Indian, then Malay, then other category student was there in that Ames University of that particular year. If we talk about the course, then dental student participated more in comparison to the nursing science students. Okay, I'm sorry. Let me silent this. Sorry for this interruption. Okay, so if we can study about the academic year where they are studying, means in which year they are studying. So you see the first year student and pre-final and final year student were participated a lot in this study, while third year was least, and the second least one is second year student. So if we talk about the knowledge part, then how we actually, let me make it, how we collected the data regarding the knowledge part. So awareness, if we compare among these two nursing and dental students, they both having nearly similar awareness. Means they have good awareness regarding the NSI. The 98% it was. If we talk about the blood-borne diseases, we checked their knowledge. We found that they are well aware about the HIV, that it is transmitted with the help of needle-stick injury or NSI. The awareness regarding the malaria, it was very least. So if we talk about the attitude part, then attitude regarding the NSI was very good for nearly all. Only one person, one student was having moderate, not poor. Still, he or she was having the moderate attitude regarding the NSI. So if we want to further investigate that from where this moderate NSI attitude was there, so it was from dental student. If we find out who were participated and how much participated in this, that total number is 150 here in the dental and remaining one from the nursing. So if we find out the relation, means association between the attitude and the gender. So there was a relation between the gender and other demographic variables, but it was not significant. Means p value is not less than 0.05 in any case. If we talk about the practice, then their practice is good nearly in all the students. Only 7 students having moderate practice regarding the NSI, and no one having the poor practice regarding the NSI. So if we want to further investigate what is the preventive practice and how much number of the student from dental and from the nursing they are having, so moderate is more in the dental, but overall their preventive practice is good for NSI. If we want to associate the demographic variable with the practice, then again there is a difference, but there is no significant association is found in the demographic variable with the practice. So if we talk about the final discussion, means what exactly we got in this study. So as we already have discussed that 92% was the response rate. And regarding the awareness, if altogether we calculate 97.9%, they are having a good awareness regarding the NSI. This is the calculation of average of these two, dental and nursing. They averaged out here and found this result 97.9. Regarding the reporting of NSI, this is the important finding we got in that year. That is 43.5% of the student did not report their injury to the medical officer. Though there are dental student, that is 58%, and nursing student 54%, they are reporting about these all. But if we subtract from 100 and find out the average, then it is 43.5% not reporting. That was the main finding in that research. Okay, so student of the higher academic year, of course, they are having greater awareness in the NSI. Right. So if we also investigated that what is the reason for not reporting the incidence of NSI. So firstly, and the mostly they are reporting that fear of stigmatization and discrimination. If they will report, people will talk about this. They feel same also. Sometimes the percentage is mentioned here, 36%. Lack of knowledge of.
Sorry to interrupt, Mr. Sunil. We are running short of time. Can you conclude?
Yes, madam, it is finished. I think it is second last, right? So consumption of low possibility of infection is also the reason for not reporting the NSI. So if we find the common causes, why it has happened, due to carelessness, maybe during the suturing or surgery, maybe recapping the needle, maybe drawing the blood. So as per conclusion, if we talk about, then they all have the good knowledge, attitude and practice regarding the NSI. The only thing is that the reporting was only moderate. 43.5% was not reporting, as I already mentioned. Thank you very much. This is the limitation, which is we know in the CAP studies. This is the, actually I just put here because one query was there. So the certificate is also mentioned. Thank you very much for your kind patience.
Yes, thank you so much, Mr. Sunil.
Yes,
Session chairs, do we have any questions for him?
Hello, Mr. Sunil.
Yes, madam.
All right. Good effort in presenting on the behalf of your student. Congratulations to your student as well.
Thank you very much, madam.
I'm just curious in terms of the sampling procedure. I mean, if you would just like show us the slide.
Okay, madam.
It was stated there by stratified sampling and then randomly. Yeah, I mean, I'm just wondering on how the stratified convenience sampling method been done.
Madam, actually, we firstly, it is convenience. You can understand that it is done in the university, so it is convenience. And stratified means we actually not distributed to everyone, means going to the class and giving to everyone. The student have applied. There is some calculation is there. That time the student have shown me that the alternate way they will give to the student only. So that was the way that time we opted to distribute the questionnaire.
Yeah, I mean, that's why I'm just wondering, because the number of the students for dental and nursing will not be that many to conduct the stratified, because the word stratified gives us some idea. From your explanation, it looks like it's more on the convenience sampling method.
Convenience, because it is.
And then when it's distributed randomly, I mean, I'm not sure how far your student really do meaning randomly, you know, because sometimes when some certain researcher, they just have a misconcept when they use the word randomly, meaning, you understand?
Yeah, I
It's not
understand.
giving random,
The students sometimes do something else and show something else.
But it's just that because the way on how it is being distributed can also be shown in the demographic data that that you have presented just now. So that's why I'm just curious.
Thank you
because
for
we
your
know that
suggestion,
because
madam. What I will do, I will put the appropriate word. If I will go for further publication on these all, then I will put the exact word which is done there in in distribution of the questionnaire, so that it would not be further questioned.
Also, I mean, it will be good if we can see the item for this questionnaire, because we don't have the idea, what are the things, the items that have been put under the section for
Madam,
knowledge, for attitude, and also the preventive practice. You understand? So
I do understand.
I couldn't give like much more comment on this basically.
Actually, thing is that 10 minutes is really very less to present the whole study of a path. So I thought that if I will present, I will read all those, it will consume the time. So I didn't mention here. If you want, madam, I will send you on your personal email ID, or maybe I will forward here itself.
Yeah, because in the section also you did mention that you used the Chromback Alpha, but it wasn't shown here. So I'm just like curious in terms of the reliability and also the validation of the instrument of the study basically.
So madam, if you need, I can share you. Actually, they have submitted a thesis to me. The soft copy is with me only. So the SPSS sheet is also with me. So I verified on my level. It's a very simple in the SPSS. If you just run the method, that will give you very easily the value. So you can verify from there also, madam.
If you can show the Chromback Alpha before and after, I believe the instrument should have been tested in the pilot study first before proceed to the main study.
Pilot study was done 20. I mentioned here also. I think somewhere it was mentioned. Where I mentioned the Cronbach, the same place. Yes, 20 questionnaire were distributed for pilot study to determine the construct validity. But thing is that in SPSS we can extract only the data. If we are going to put the same table, and so many tables are there, so putting all the tables, it's very
yeah. Can I just know that what are the tests that you use to determine the construct validity?
Reliable, this Cronbach Alpha, madam.
No, the Cronbach Alpha is meant for reliability.
Yes, madam, reliability, and there is an option available on the SPSS.
Yeah. So what are the things that you use for the construct validity?
Method, madam, I just given command in SPSS sheet. Once you
so meaning there is no construct validity. It's just internal reliability, which is the internal consistency that you're using the Cronbach Alpha, right?
Okay, you mean to say in that way? Okay. So what we did, madam, the face validity and content validity was observed by already expert in the medical department. We have MBBS also and dental department also, since the study was. So these both are done by that. And regarding the validity, we directly calculated with the help of SPSS sheet.
So meaning there is no construct validity being done.
I think this word should be more appropriate.
For pilot study, if you're using only 20 head, meaning only 20 respondents, and then you would like to determine the construct validity, it will be difficult. So that's why I'm asking, what is the name? What was the name of the test that you used to determine the construct validity?
Okay.
The one that been shown here is just the reliability, the internal consistency.
This word should be more appropriate, madam. Good.
This one should be reliability.
Correct. Thank you, madam. I will take care of this also. Once I will go for the good publication, I will take care of this also.
All right. Thank you.
Thank you.
Great,
Thank you very much, madam, for guiding
Thank you,
us.
ma'am, and thank you, Sunil, for your presentation. So I would like to invite our next presenter, requesting all the presenters to kindly limit your presentations to 10 minutes and a discussion of 2 to 3 minutes. So our next presenter is Dr. Shoaibul Haq from King George Medical University from India, and he'll be discussing on a systemic review on novel antifungal 1,3-beta-D glucan inhibitor role in resistant vulvovaginitis. Mr. Shoaibul, you're not audible to us. Can you please begin?
Am I audible to everyone?
Yes, yes, please.
Okay, very good afternoon. Let's start with my presentation. This is that is a systematic review on novel antifungal, that is a 1,3-beta-D glucan inhibitor role in the resistant vulvovaginitis. So as we know that the fungal infection are very common nowadays among the low socioeconomic people, and this is vulvovaginal candidiasis, the most common infectious disease that markedly impact the quality of life of the young females, and that may lead to the depression into the females. And the drugs till nowadays we are using are mainly belongs to the azoles groups, and the problem is most of the azole group drugs are now resistant to these infections, which are caused by the different species of the candida. And some other groups which are we using in is the echinocandins for these resistant infection. What is the problem of the with these groups, that they are giving, we have to give those groups intravenously, like the echinocandins, like mica fungin or the casfofungin or any dulafungin. So this may take a long duration, and patient having some post injectable side effects also. So this new drug ibrexafungerp is already approved recently by the FDA. This is a glucan synthase inhibitor that interferes with the formation of the fungal cell wall, and they inhibit the growth of the resistant candida species like the candida auris or the candida glabrata by acting on the alternate catalytic sites. So what is the focus points of this review paper? That this review paper is to assess the role of the ibrexafungerp in the vulvovaginitis in compared to the existent antifungal groups, which also act on the same enzymes. And what is the main focus point? That patient has to take this drug only a single course, and low toxicity and better activity in the tissue, and the better penetration. And the search methods is we followed the, of course, the Prisma guidelines. The data was searched on the different databases like the PubMed, Google Scholar, and the Scopus by using the some specific words like the ibrexafungerp, oral antifungal, glucan synthase inhibitor, and the vulvovaginal candidiasis. So we also followed some inclusion and the exclusion criteria. We included the studies that are mostly talk about the ibrexafungerp, their activity in vitro, their safety and the bioavailability, and we excluded the studies which doesn't include the full text, and also the studies with not so much talk about the ibrexafungerp activity and their safety. So in the chart, in the identification, we find out the total 120 articles from the databases, and there is no additional records from the other sources. And during the screening, we after removal of the duplicates, there we find out the total 55 articles, and we also include some articles with which are the review articles and which having only abstract. And after that we got the total 23 articles, and full text articles excluded, also some with other reasons. So we include the only 7 articles for our study. So what is the mechanism of the most of the fungal antifungal drugs? That the main component of the fungal cell wall is the ergosterol. So we have to break this fungal cell wall, and this ergosterol is formed from the squalene, which is converted into the squalene epoxide by the mediation of the enzyme 14 alpha demethylase, which is converted again into the lenosterol by the squalene epoxidase enzyme, and which converted into the ergosterol, which is the main component of the fungal cell wall. And this is inhibited by the majority of the azole groups of drugs like the triazoles or imidazoles. Nowadays we are using the like clotrimazole and other azoles drug. So other mechanism of action is the by making the pores into the fungal cell wall, like we are using the amphotericin B. So nowadays most of these drugs are resistant to the antifungal drugs. Some other groups we are using in the resistant cases like the echinocandins, which act on the beta glucan synthase enzyme, which involved in the formation of the beta glucan in the formation of the fungal cell wall. If we inhibit this enzyme, so there is the stop the formation of the beta glucan and the inhibition of the fungal cell growth. So we have to inhibit this enzyme. And the echinocandins, mostly the problem with the echinocandins like the anidula fungin or the mica fungin, caspofungin, they are available only into the intravenous form. And recently we find out in the after post-COVID cases, in the cases of the mucormycosis, what the patient affect with the intravenous antifungal drugs is the inhibition of the kidney function, and there is the swelling of the body tissue. What is the mechanism of the ibraxafungerp? It causes the inhibition of the fungal cell wall by the decrease in the formation of 1,3-beta D-glucan polymer. And this is a structure distinct from the echinocandin and interferes with differently with the target enzyme on the other catalytic site, and it's partially overlap the binding site of the echinocandins. Like in this figure, we show echinocandins work on the FKS1 and the FKS2, and the ibraxafungerp GDP binding regulatory subunit acts on the alternating catalytic site. So what we find out in our study, we collected the data from all 7 studies, that in the according to the first study, there is no cross resistant developed and the potent in vitro activity, and we find out the broad spectrum of the activity including the candidiasis or the aspergillosis. In the second study also, there is no cross resistant high fungicidal activity and good clinical response in the phase 2 and phase 3 trials. They are even in comparison safe to other azoles. And in the third study, we find out the broad in vitro activity including the azoles resistant candida species, and they also active in the acidic pH at the 4.5. So in the fourth study, we find out there is no serious adverse effect like the azoles, or in less severe adverse effect in comparison to other antifungal drugs. And so we find out in fifth study, there is no cross-resistant developed and in vitro activity against all clades of the Candida auris, and outcome is promising novel antifungal agent against the Candida auris. In sixth, there is also no cross-resistant and high in vitro activity even in the minimum inhibitory concentration from 0.03 milligram per liter to 0.05 milligram per liter against the isolates, and the potent agent for the treatment of the vulvovaginitis. So we find out that the ibraxafungerp is more potent than other azole group of drugs. So in results, we find out that that ibraxafungerp is having the significant antifungal activity in vulvovaginitis, even caused by the resistant species like the Candida glabrata. So all the 7 studies verified that the ibraxafungerp is an optimistic antifungal agent for the treatment of the vulvovaginal candidiasis. And the present systematic review shows the favorable characteristics of the ibraxafungerps, like low toxicity, better activity at the low pH, better penetration activity. As a future perspective, this is a single day dose therapy. Patient has to take only 600 milligram in divided doses in the morning and the evening, and it appears to be safe without much interaction with the other agents. So patient with the like immunocompromised patient, or the patient which already taking their drugs for other diseases, it can safely use this drug for the treatment. And the different mechanism of action offers a potential novel method, of course, for suppressing the resistant fungal infection in our society. In conclusion, we find out that the ibrexafungerp is a promising agent for the treatment of vulvovaginitis with good in vitro activity against the resistant candida species. This is a single day dosing therapy, so patient don't have to take for the long period of time like the other drugs that patient has to take oazos for the weeks to months and up to years. This is just a single day dose therapy. So, however, of course, if the patient take some drugs orally, so there is no use of the intravenous setup, and the cost is also the low. So, a low cost and the availability of the drug in the market of the developing countries is the task of the pharma companies. These are the references of the studies. Thank you very much.
Yes, thank you, Dr. Shoaib Haq, for your presentations. Session chairs, any questions? Okay.
No questions here.
Yeah, thank you. Thank you so much for your presentations.
Thank you.
Before we move on to the next presenter, I would like to request all the poster presenters to kindly move to Hall C. As we are running late and short of time, requesting all poster presenters to move to Hall C. If you have any questions, please put in the chat box. So we will move on to our next presenter. Next, I would like to invite Dr. Kumudavalli Suresh Kumar from Vinayaka Missions College of Pharmacy from India.
Yes, madam.
Yeah, ma'am, you may begin. Yes,
Yeah.
Kumudavalli will be presenting on the evaluation of toxicological studies of Cardiospermum calibacum lin.
Yeah, myself, Dr. Kumudavalli. I am professor in Department of Pharmaceutical Analysis in Vinayaka Missions College of Pharmacy, Tamil Nadu, Salem. And my present topic is, a part of research topic is evaluation of toxicological studies on Cardiospermum helicobacter. And the content is introduction, aim, objective, plan of work, novelty of the proposed research, plan profile, pharmacological activity, conclusion. So nowadays, here my aim of the present work is, and my main work is to find out the toxicological studies of that particular plant in the zebrafish model. So most of the toxicity studies are conducted to assess the degree to which the substance are toxic for humans or animals, to investigate the mechanism of toxic chemicals, or to develop new or improve tests for specific types of chemically induced effects. In toxicological studies, it includes the acute toxic studies, whereas the examination of adverse effects and that exposure to single dose of substance, genotoxicity, and studies that seeks to assess the potential of substances to interact with genetic materials, and the repeated dose toxicity studies that are undertaken to find out whether cancer may be developed as a result of exposure to certain chemicals, and studies to ensure the safety of medicine. The main benefit of using zebrafish related to their size, husbandry, and is early morphology. Instead of other fish species, zebrafish adults are only approximately 1 to 1.5 inches long. Also, zebrafish have been utilized as a laboratory species for quite some time, so the optimum breeding and maintenance conditions have been well determined. So the fish embrace toxicity test has been proposed as an alternative for the acute toxic study for the Cardiospermum helicobacter L. It belongs to the Sapindaceae family, and it has a different active constituents like phenol, asparagine, protocatalonic acid, protocataldehyde, casein, rutin, and quercetin, etc. The medical use of whole plant of Cardiospermum helicobacter is diaphoretic, diuretic, emetic, laxative, refringent, rubefacient, and stomachic. So it is mainly used in the treatment of rheumatism, nervous disease, stiffness to the limbs, earthquake, and snake bite. The leaves are rubefacient. They are applied as a poultice in the treatment of rheumatism. Fish embryotoxicity assay was performed as per OECD guidance 236.
The test method described in this test is intended to determine the acute or lethal toxicity of the chemicals, compounds, or embryonic stage of fish. Toxicities includes the principle is based on the SOPs on and NIH guidelines should be followed thoroughly for these studies, and it should be performed by well-trained and qualified staff. These should comply with norms of good GLP with proper characterized and standardized. Animal production studies should be designed so that the maximum amount of information is obtained from the smallest number of animals to avoid causing excessive pain or tissue damage in the animals. Pharmaceuticals with irritant or corrosive characteristics should not be administered in concentrations. Aim and objective of my research work is to assess the toxicity activity in zebrafish from isolated biomolecules from the whole plant of ethanolic extract of this Cordyceps helicabum. The objective of this research work the objective of the acute toxicity of isolated biomolecules from the whole plant of ethanolic extract of Cordyceps by using zebrafish embryo assay method. That plan of work is as followed: collection and authentication, extraction, phytochemical studies, standardization, and pharmacological assay, this embryo toxicity assay. Novelty of the proposed method is here we are using the zebrafish is a powerful model for translational studies. In particular, zebrafish embryo is widely used model to study developmental histogenesis. Zebrafish scale represents a very helpful model due to its accessibility, easy handling, quantity, and similarity to human bone. Zebrafish GIOP model can be used to test the effectiveness of natural molecules to counteract the onset of PN-dependent effects. So this is the plant profile. This plant, and this is called as this is one of the rheumatoid drug. Method of preparation procedure for the acute toxicity zebrafish embryos. So they were maintained in tanks in reverse osmosis and activated carbon filtered water. They were raised in a facility with the photoperiod cycle of 12 to 12 hours, light and dark. The water parameters were strictly controlled. The temperature was maintained at 27 plus or minus 1 degree centigrade. Conductivity is 740 plus or minus 100 microsmere, and 7 or plus or minus and are dissolved in oxygen 95% saturation. Zebrafish embryos were collected immediately after natural matting, rinsed in water, and checked under the stereo microscope. The unfertilized eggs and those showing cleavage irregularities or injuries were discarded. These embryo toxicity tests were based on the OECD guidelines, Protocol 236. The zebrafish embryos were grouped into 20 embryos, each in 0.1% volume by volume methylene blue, treated with fresh water to prevent fungal outbreak. A stock solution of 1 mg per ml of ethanol fraction of Cordyceps were used, and the 0.1% methylene volume ethanol were used as the solvent for the extract. And the embryos were exposed into the fraction of 2 HPF. The exposure was continued till 96 HPF hours. Okay. Then the embryos were examined for delayed hatching, yolk sac edema, pericardial edema, craniofacial abnormalities, curved body axis, notochord curvature, loroiosis, sclerosis, neosis, tail restoration, growth retardation, hemorrhage, tail malformation, crooked spine. The mortality was recorded as number divided by the total number of exposed embryos. The stages of development and the heartbeat were captured using a high resolution camera and compared with group 1 normal embryonic development. So these were grouped in 5 groups, and the 5 groups were given control and with 1% to different concentration of ethanolic extract of this Cordyceps helicabum isolated molecule. Then this is the different stages development of the embryos. Embryonic development of the control group. This is group 1, and this is the group 2. In this group 2, the various hours were selected, and we have studied about the what is the embryonic growth will be happen by using this 0.1% ethanol. This is group 3, and this extract is 3.125 microgram per liter were given, and this is the group extra dose 6.25 microgram liter of the biomolecule were given administered to the egg yolk, and this is 12.5 milligram, and this is 25 milligram per liter, and this is 50 milligram per liter, and this is 100 milligram per liter. So these are the results. The embryos were exposed to test fraction 2.52, and the exposure was continued till 96 HF at various time intervals of 6 hours, 12 hours, 24 hours, 36 hours, 48 hours, 72 hours, and 96 hours. And the mortality rate of zebrafish embryos exposed to the ethanol extraction of cardiosphere were very less. Okay, so there is no mortality rate, and all the 6 hours, all the 96 hours. So acute toxicity of ethanol extraction, there is so that we can find out that the cardiosperm and helicobacter isolated molecule will not be having any toxicological effect, so that we can use the further studies. So acute toxicity of ethanol extraction of this tested in zebrafish embryos as per guidelines. Normal developmental process was noted in group exposed to graded, and there was no mortality till the dose level of 100 milligram per liter. From the result, it is evident that the fraction is safe up to a dose of 100 milligram per liter in fish embryo toxicity assay. So this was selected for toxicity state to provide future herbal active biomolecule drug formulation to cure disease. Okay, so this is the as my conclusion is, this is a novel method of in vivo activity in bone protection. For this reason, this ethanol extraction of this cardiosphere lymph represents an excellent candidate to develop a new natural approach against human osteoporosis. Our result will contribute to encourage the in vivo studies and the use of molecules of natural origin for the treatment of human bone diseases. So this work was supported by Vinayakam Research Foundation, Deemed University, Salem, Tamil Nadu as a seed money project proposal. This is all the few references. Thank you.
Thank you, Kumundavalli ma'am, for your presentation. Sorry for the delay and the trouble that have caused you. Session chairs, any questions for Ms. Kumundavalli?
Can you show me your reference, please?
Yeah, wait, sir. This is one few of the reference only, sir. We are not having much references for this work. So from the review article and some of the articles we have selected, and this work is still going on. The molecules were isolated, and the still work is going on as osteoporosis activity in embryo fish model.
Okay, I would like to ask you one question. What is the rationale to choose zebrafish for this particular one? Because previously there was a presentation that 84% of zebrafish matching with the cardiac, human cardiac related, but what is the rationale to choose zebrafish for human osteoporosis? How do you correlate? Is there any publication related to that?
Yeah, only one review article is there, sir. Based on that only we have selected in this one.
Did you mention that review article here? In the reference?
No. I didn't mention that review article here.
That is the most important one, right? Which is related to your work. Novelty. You're saying novelty that you're using a zebrafish as a model.
Okay, sir. Okay, next time I'll, because still work is going on here. Okay, this is actually on the funding project. So that only part of the work, only toxicity studies were completed now, still now. So that,
okay. May I know what are the constituents probably if in your literature for the this particular specimen? Cardiospermum helicarbium.
This is already we are here in India. We are using here for rheumatoid arthritis. This drug, this plant is used as the rheumatoid arthritis. So that for this rationale only I have selected this plant. Why can't we use some for this plant? Some of them have isolated volatile oil also in this one.
Yeah, okay, that's fine. That's fine. You selected because it is used in the for human osteoporosis or something. But may I know what are the constituents probably?
Constituents is mainly flavonoids, sir. We are having flavonoids in that one.
How do you justify flavonoids will be useful in human osteoporosis? Because the histology of human osteoporosis, right? Yeah, probably. Okay. Any prof maybe any questions?
Okay, thank you, sir.
Yeah, I mean, I do agree with you because the the conclusion part is a bit confusing. It's okay that sometimes it can be typo error. I believe your study is a big study, and you're presenting a part of the study.
Yeah, actually, I mean, I have some straightforward comment. Actually, So far presentations were, I gone through the presentations were, because I am evaluating the presentations, I have small confusion here. Actually, frankly speaking, most of them are presenting, actually, their presentation is very bulky slides are there. The slides are actually very bulky and, reading and that indicate actually, they're like, for namesake you are presenting means that's not nice actually, because we are an international conference. So probably your presentation slides will be very minimum and you can explain more and within the 10 minutes, and if you give me a presentation for 10 minutes, I will give you more information in 10 minutes with less number of slides and less number of wording there. So that will not be stressful for the evaluator. At the same time, it's good for the presenter as well, because if I give you if I had to give you marks, but most of the presentations for very less marks I've given. Because they're very bulky. Some are very bulky, some are unable to express, some are reading. Try to maintain the quality, in the conference actually, the presentations. I trust it would be great, if you all improve the presentation skills. Thank you very much.
Thank you, sir. Thank you, Professor, for your comments, and thank you, Kumdavali. So quickly moving on to our next presenter, Camelia Roy.
Hello.
From Sri.
Hello.
Yes.
Ma'am, I just want to ask a question to Kumudamali, ma'am.
Okay.
Yeah. Ma'am, I just need to know, to work with zebrafish, what are the ethical, permission is required? Because we know that for other animals it is required. So what is the current ethical guideline or permission?
We have to follow the OECD guidelines 236, sir. For the fish embryo studies, no need to get any ethical clearance.
Maybe, ma'am, you both can exchange your email addresses in the chat box so that it will be convenient to discuss further and share more information.
Yeah, sure, ma'am.
Yeah, thank you, ma'am.
You can do that. Thank you so much. So next I'll invite Camelia Roy from Sri Ramachandra Institute of Higher Education and Research from India, who will be discussing on the therapeutic model through traditional medicine on transplacental genotoxicity caused by modern medicines in pregnant women.
Good evening, everyone. Good afternoon to everyone, and warm regards to all my esteemed speakers and session chair. Myself, Camelia Roy from India, pursuing my PhD from Sri Ramachandra University from the Department of Clinical Research and from the Faculty of Center of Toxicology. I'm here to present a part of my PhD work titled Therapeutic Model Through Traditional Medicine on Transplacental Genotoxicity Caused by Modern Medicines in Pregnant Women. So here I will proceed with two beautiful quotes. Hopefully my screen is in it's sharing mode. It's visible and I'm audible.
Yes, you're audible.
Okay, ma'am. Thank you. So I'll start my talk with these two beautiful quotes. Like as per my topic, that medicine is there. So the person who takes medicine must recover twice, one from the disease and one from the medicine. It's very indeed and it's very important who are taking or consuming any kind of medicine and overcoming any diseases. And second, as my talk is a part of toxicology, so I'll go with a famous quote by Paracelsus, like all substances are poisons, there is not which is not a poison. The right dose differentiates a poison. So here I will begin the talk, and this is the outline of my talk with a brief introduction and my area of research with the prevalence of the various drugs and its mechanisms, and I'll be explaining a little bit of genotoxicity and the purpose of my work and the prophylactic management to mitigate the damage which will be causing by the drugs and a scope of my research. So here with a pictorial presentation, I'm here to describe or give an overview of my work that more than half of the pregnant women use prescription or the OTC, over-the-counter medications, and the prevalence of using it is rising very rapidly. So therefore, their absolute necessary medications should only be used during the pregnancy, like that. There are so many medications that that have been prescribed to pregnant women and that have an effect on the fetuses, like about 2 to 3% of birth defects have been reported in literatures. So according to the new PLLR rule, that is the pregnancy and lactation labeling rule that has been that came into effect in the around 30th June of 2015. So according to prior PLLR, there was three classifications were there, like pregnancy, labor and delivery, nursing mothers. And in the new PLLR labeling, this labor and delivery has been shifted to the pregnancy classification, and the nursing mothers have been shifted to the lactation. And a new clause has been added to this PLR labeling, that is the females and males reproductive potential. So,
excuse me, Camelia, your slides are not moving.
Yeah, ma'am, I'm just explaining this current slide.
Yeah, fine, sure.
So in this new clause, it's been like it has been explained like it will be included with informations about the need for pregnancy, recommendations for the contraceptions for various like who are like expecting or already conceived. So coming to the next slide, like as per my talk, that medications that are being prescribed to pregnant women reaches the fetus through placenta and serves the placenta, which serves as a functional link between fetal and maternal birth. So there is a higher possibility and chances that the drugs which are being prescribed to the pregnant women with who are suffering with several complications reaches the placenta. So as per the NIH reported that there are some common complications that are being reported during pregnancy, like hypertension, allergies, gestational diabetes, preeclampsia, preterm labor, depression, anxiety, and many more. So there is a particular state like during the organogenesis where the drugs reaches the embryo and can probably cause subtle or permanent functional or metabolic abnormalities. So there are two more, like as per my slide, it has been said, is there any side effects have been causing during the pregnancy, or any genetic material is been getting damaged due to those medications which are being prescribed. This is the concern of my area of research, and the prophylactic, then how to lessen the effect or the damage. So coming to the next slide, I will briefly explain the area of my research, which I'm going to do in my, like I've already started my PhD work. So that I will be looking into the common complications. There are so many complications, but I'm only looking onto like two, this hypertension and the allergies, which is the most common, and sometimes it can be a little bit fatal. And then I will be like the drugs which are being prescribed during the pregnancy that for treating these two complications. And then I will be evaluating the transplacental genotoxicity with certain methods to identify if any damage is being caused by the prescribed medicines. And then if any damage is being caused in the genetic level, then how I will be treating it with the prophylactic management with traditional approach, like traditional medicine, using the traditional medicines. So coming to the next area, like what is the prevalence of the transplacental, like crossing the drugs, how it is causing. And so as we know that during the third trimester, transplacental drug transfer increases.
Excuse me, Camelia, your slides are still not moving.
Yeah, ma'am, I'm explaining this slide. Is it moving, ma'am?
No, it's not. You are still in the second slide, quotes.
Now, ma'am?
Okay.
Yeah, thank you. And as we know that the during the third trimester, transplacental drug transfer increases due to the increased maternal and placental blood flow. And as the placental thickness decreases, and the surface the surface area of the placenta increases. So there are 4 parameters where a drug can cross the placenta and reach the fetus, like the protein binding, pH difference, lipid solubility, and the molecular weight of the drug. So as we know that with these different, this is the prevalence and chart which I have tried to show that during pregnancy, these are the complications are like induced during this gestational period. So in the present, like in my study, I'm trying to focus as I have told in the prior slide, that I will be focusing on the 2 complications, that is the hypertension and the allergies. So as per the my study to focus and the area, like I will be the 2 prescribed drugs which are being prescribed by the practitioners to treat allergies and hypertension are loratadine, it is an antihistamine, and the labetalol, which is an antihypertensive. So according to the reports, both drugs have a slight chance of crossing the placenta. Slight chance of crossing the placenta, and are also been reported, it has been found in the breast milk, because these 2 drugs are having the low molecular weight, that is the less than 500 gram per mole. So there is a higher chances, and it has also been stated in literatures that it has caused DNA damage, and there are like certain aspect which is causing effect to the fetus. So in other words, like these effects may not be very severe that can be visible to our naked eye, but it may cause minor defect in the gene level, where it can cause certain kind of complications later at the later stage of that fetus or the newborn child. So as coming to this loratadine treatment, which is like a 2nd generation antihistamine, according to FDA pregnancy category, it falls into the category of B, with a molecular weight of the 382.9 gram per mole, which is less than 500 gram per mole. So there is a higher chances to...
Excuse me, Camelia, you have another 2 minutes to conclude.
Sure, ma'am. Sure, ma'am. So there is these 2 drugs are being crossing the placenta, because the mechanism of drug, we know that the when the blood vessels in the fetus are there is a villi-like projections, hair-like projections are there. So when the mother blood flows through the space, and if the molecular weight of the drug is less, so it is growing through the circulation and it easily crosses the placenta. So coming to the topic, as I am in the how the drug is passing the placenta. So during the placenta, maternal plasma albumin decreases while the fetal albumin increases. So coming to the prophylactic management, this is the genotoxicity. Genotoxicology is a study of agents or substances that can damage cells, DNA, and chromosomes. So this is the main part of my study, which I will be seeing by using some assays like the micronucleus test and the COMET assays to see identify any kind of damage is happening, though it has been already reported that these 2 drugs are causing damage. So according to the lacunae, I want to fill the lacunae that through the use of traditional medicine to have which will be having a major impact in the society by attempting to lessen the effect. So here are the certain traditional medicines according to World Health Organizations, this cinnamon, ginger, and the chamomile tea, garlic, sitobaldi, and shankapushpi. These are the certain drugs which are being these have the effects to these other traditional medicines which can be used during the pregnancy, because during pregnancy we cannot use every drug every herbs. There are certain complications linked to it. So therefore, keeping in considerations and the dosage volume and dosage concentration and everything, we will be using these prophylactic management. We're using these traditional medicines to treat the damages that is being caused by those allopathic medicines. But in my talk, I'm not totally against the saying that I'm against the allopathy drugs or the Western medicine. I'm also supporting them, because in emergency we need those medicines to suppress or take the emergency quick decisions. So these traditional medicines we can suppress or mitigate the effect that is being causing and which is causing the damage for long term. So side by parallelly, we can use these traditional medicines as a hope to just to lessen the effect. So here I will conclude my talk that this transplacental genotoxicity is it will have a greater impact. So to fill the gap between this, using the Western medicine, it is causing disease, and then using a traditional medicine to mitigate. So we are trying to link and make a major impact in this society, because this transplacental genotoxicity is a very rare route which are being tested, tested in this in our healthcare system. So here I will conclude my talk with awareness and knowledge are the key to stay healthy, to keep and bring the next generation healthy. Thank you.
Thank you, Camelia, for your presentation session. Chase, do we have any question?
No questions.
Okay. Thank you. So we'll be moving on to the next presenter. Yes, next I would like to invite Dr. Aisha Naik from Veer Surendra Sai Institute of Medical Sciences and Research from India. We'll be discussing on the effect of enoxaparin on D-dimer level in hospitalized COVID-19 patients and observational study. Dr. Aisha Naik. Okay. We'll be moving on to the next presenter. Please make sure, presenters, that we will not be giving another chance. Please make sure you present during your allocated time. So our next presenter is Niharika Modi from JSS College of Pharmacy. Is Ms. Niharika Modi available? Ms. Niharika, can you hear us? Yes, Niharika. Niharika, you're not audible to us. Yes, Niharika Modi's topic is carbon nanotubes, treatment and applications in neurodegenerative and Alzheimer's disease. Niharika, you're not audible. We cannot hear you. Niharika, can you please check your mic? Niharika, maybe you can reconnect. Can you please rejoin Zoom with your audio? You're not audible to us. Niharika, we request you to rejoin the Zoom link. Next, I would like to invite Anju Kumar Singh from JSS College of Pharmacy.
Yes, ma'am. Am I audible?
Yes, Anuj. Yes. So Anuj will be discussing today on social media's impact on pharmacovigilance. Mr. Anuj, you may begin your presentation.
Some slides are visible.
No, it's not visible. Your folder hasn't opened. We can just see your USB drive.
Yes, sir.
Can I share again? Give me 1 second.
Yes, it's visible now.
Okay. Yeah. Good afternoon, everyone. I'm Anuj Kumar Singh from JSS College of Pharmacy. I'm currently pursuing the M.Pharm from Pharmaceutical Biotechnology. So my topic is social media's impact on pharmacovigilance. So now let me explain the pharmacovigilance. Pharmacovigilance is the science and activity related to the detection, assessment, and understanding and prevention of adverse event or any other medicine related problems. So why are choosing this topic? Consideration for using social media in pharmacovigilance is social media present a new channels and method that can enable companies to move away from traditional pharmacovigilance system and safety data. These channels have a capability to allow swift and open communication between companies and the consumers. So now social media is monitoring will become a standard practice in pharmacovigilance and definitely also in the future. However, before the careful evolution will be taken. So pharmacovigilance deliver some of the important capabilities to pharmaceutical companies like adverse event case management, including expanded reporting, aggregate reporting, signal intelligence, and risk management. So as you all are aware of social media, so social media doesn't need any explanation for it. So now I can come to the pharmacovigilance and its sources. So these are the this diagram showing data mining from algorithm, and these are the preclinical trials, post-clinical trial surveillance, and these are the PSUs, periodic safety update reports, individual case study reports, and the most importantly, advanced technologies like other IT sources, apps, and most importantly, social medias. So this is the data mining from social media, how we mine a data of related to pharmacovigilance from social media. For ADR detection from social media, we use like a Medline, Embasys, Scopus, Web of Science database, and the Google Scholar. So these search engines are generally used to extract all the information from social media. So this is how this diagram showing the process. Social media, firstly, we did collect the data collection, then we have a raw data, then we have a classified the different things, then we filtered that data, then we ADR, adverse drug reaction, extract, then we have a drug adverse reaction pairs with specific drugs, and then we have a statistical analysis, and if we found any of the flaws and any advance of the adverse drug reaction, then we have a flagged drug. So this is how I take one of the example. Like we have to search in a Medline PubMed database. We say like I taking the 1,374 article identified. From these 1,374 articles screening in first round. Then we exclude irrelevant article like for example 1,188. So 186 articles are remain for second round only. From that 101 articles are covering the just border of pharmacovigilance. So we also exclude these also. So 85 articles are selected from 1,374 articles. So 15 articles are selected on the basis of screening references. So totally we take like, for example, 100 fully article assessed. So this is how the process of data mining from social media for pharmacovigilance. So this is how showing a bridging gap. As we saw, this is the time of COVID-19 and we people are just locked in our houses. So in this time, the social media can be a biggest boon for pharmacovigilance during for the COVID-19 drug, orphan drug, new medicines, and LMICS. So this is how showing the social media and artificial intelligence helping the. This is the way how we classified the various data for social media, the specialized healthcare social network for have solicited data and unsolicited data. So unsolicited data, we take just spontaneous reports, and solicited data are derived from data collection system. These are like generally social networking sites, solicited data, unsolicited data also again, internet search logs. So don't we have all the boons? We have some of the challenges also. The use of social media for pharmaceuticals represent the knowledge discovery process. This present a study, examine its challenges. So on the 3 dimensions, we have to challenge like conceptual challenge that related to the purpose and value of the social media used in the pharmacovigilance. Like, is it valuable? The knowledge is valuable or not? Shall we check if we can't relevant tool directly? Next is the technical challenges that related to the feasibility of the process. Like this is how a process is, this is too hard, or this is not easy to extract the specific reports for pharmacovigilance from social media. And next is the environment challenges that compliance is concerned affect from acceptability of new pharmacovigilance, like a proposed data, like data privacy, regulatory framework. Next, we have aesthetics, how the role of statics showing the comparison with the other things with social media. So this graphical shows those 3 dimensions differ across the social media, clinical trials, spontaneous reports, and the electrical health records. So we have taken the 3 dimensions of patient report outcome, and second one is seriousness, third one is completeness. So patient report outcome, the social medias have highest number, have a highest graph in this given diagram. Social media can exclude that patient experience most frequently. That's why. And traditional pharmacovigilance focus on most serious of event, maybe less central in social media. So social media post may be less complete, conversely contain less sensitive identify information than electronic health record. This is how showing the statics, how our social media is most doing well in comparison to these things. Now come to the conclusion point. As we all know, this is the time of internet time, or we are just fighting this COVID-19 period. So now we need such type of applications in for future, like how in order to realize the potential that social media has to offer number of challenges remain to be resolved, but we need to be consider for the future use. For example, I'm taking in 2014, ICR is funded the BEV radar project to conclude the scientific research in the use of social media network to develop a dedicated application apps for reporting the adverse drug reaction to regional competent authorities in Europe. The BEV radar consortium comprises patient advocate, regulator, academia, and member of the pharmaceutical industry working in the collaboration. The rapid development of technology bring it with the opportunity to harvest a vast amount of data, both from the social media and emerging biomedical technologies, to allow the earlier signal detection, develop early warning system, and enhance the pharmacovigilance and patient safety. So this is all how showing our pharmacovigilance and the role of social medias. So how important the social media is nowadays for pharmacovigilance. So that's all. Thank you so much for your attention.
Thank you, Mr. Anuj Kumar, for your very quick presentations. Session chairs, any questions for Mr. Anuj Kumar?
Yeah, I think
hello, Mr. Anuj Kumar.
Yes,
okay, it's like from your review,
yes,
on how are they going to extract the ADR that been reported in the social media? I mean, to be included, for the pharmacovigilance.
When we have a specific Medline, Scopus, and Google Scholars, so by using the specific terminologies and so we extract the articles. So from that articles we read and all that things. So found out the relevant article, and then after we will do this. Hello.
Professor Nahla?
Also, basically, you are doing a review.
Yeah.
So you are like reviewing articles that use social media as the platform when they are wanted to collect the data of the ADR for the pharmacovigilance. Is it?
Yes, ma'am.
Okay, no, I was thinking that the presentation is heading somewhere. It's like because from the way on how you put at the end, it looks like you have a solution for it.
What, ma'am? Sorry, ma'am, I'm not getting.
No, I mean the way on how you put at the end, it looks like you are telling us a solution to it. It's like I was hoping that you will mention that there's a like an apps or whatever it is that can capture the wordings in the social media that you understand or not. So the one that you presented to us is a review on how getting the ADR for the pharmacovigilance study.
Yeah, ma'am, it's a review that how this social media is nowadays act as a boon for pharmacovigilance, even in the time of everyone is locked in their houses. So that's why I'm showing this review. So that when people can easily post it their reviews and all these related to the drug, so they can easily post it their whatever they get is the side effects, so they can post it on the social media, and we extract from that and we found out this drug is relevant or not, this drug is okay, or we have to continue out with this drug or not. For example, when like our government in Indian government gives a toll-free number for this COVID-19 vaccine. So after taking a vaccine, you have to inform on that toll-free call number. So what are the adverse effects or what are the side effects you get after this? Like someone gets fever and all this. So this is some sort of call. So I just saw this how this social media like Facebook, Twitter, and all these things we all are using in every day.
Okay, thank you.
Thank you. Thank you so much, ma'am.
Thank you, ma'am. And thank you, Anuj Kumar, for your presentation.
Thank you, ma'am. Thank you, Jelia.
Yes, is Niharika Modi ready? Niharika, can you check your mic now? Niharika, you're not audible to us now also. Can you please check?
Please.
Niharika, I'm very sorry, but we cannot hear you without the audio. It will be difficult for you to continue the presentation. So I'll be moving on to our next presenter, Sayantan Pal. Is Sayantan Pal ready?
Yes, ma'am.
From JSS College of Pharmacy, yes.
Ma'am, I am audible.
Yes, you're audible. So Sayantan will be discussing on opening the treasure chest of the non-coding RNA.
Yes, ma'am. Thank you, ma'am. 1 second, ma'am. Good evening to one and all present here. I am Santan Pal, and today my topic is about opening the treasure chest of non-coding RNA. But before that, I am going to introduce what is RNA. RNA is unlike DNA, is a single standard molecule made up of backbone of ribose sugar and phosphate bond. RNA are basically of two types, coding RNA and non-coding RNA. Coding RNA are those types of RNA that code for fundamental proteins, while non-coding RNA are those types of RNA which doesn't code for any fundamental protein. For example, the non-coding RNA examples are tRNA, rRNA, some small RNAs like microRNA that consist of 22 nucleotides. From beginning to nowadays, we have find some functions of this types of non-coding RNA. The functions are heterochromatic formation and some histone modification. Some non-coding RNA can do DNA methylation and some can do gene silencing. Now, these are some functions that nowadays we can determine with the help of some tools. Some diseases that associated with non-coding RNA are here. They are cancer, Prader-Willi syndrome, autism, cartilage hair hypoplasia, and Alzheimer disease. With taking the example of Alzheimer disease, now I am going to show how an non-coding RNA have function in it. Alzheimer disease pathogenicity consists of a non-coding RNA named as BACE1. It increased the amount of beta amyloid and which further increased the senile plaque that cause neurodegeneration, and hence this help in causing of Alzheimer disease. As I say in previous slide, that its function is gene silencing. If we can find some non-coding RNA which code for some protein that silence the gene BACE1, then we can cure the Alzheimer disease. There's a hope that we can cure Alzheimer disease. Here is the basic methodology of how we can determine the function of non-coding RNA. The methodology consists of first, step 1, procurement of genes from database. Step 2, identification of non-coding RNA in the genes. Step 3, obtainment of already known ncRNA. Step 4, creation of an artificial intelligence. Step 5, training set with already known ncRNA. Step 6, non-coding RNA motif identification. Step 7, function of non-coding RNA prediction. Here is the first step, procurement of gene from database. The genes are taken from NCBI or we can say National Center of Biotechnological Information. These genes are taken from NCBI, and the non-coding RNA are identified from the gene script that is taken from NCBI. Then we have to prepare our own ideal gene script, and we have to compare with the gene script that is taken from the NCBI side. Then we have to check our accuracy. Now, for second step, identification of ncRNA in the genes. From our own RNA library that we have created with our gene analysis script, we check that how many RNA are there, non-coding RNA, and how many RNA are for coding RNA. We check by translation and check that if it codes for any fundamental protein. If it codes for any fundamental protein, then we discard it. And if it doesn't code for any fundamental protein, then we take that non-coding RNA as our sample RNA. Now, as third step, obtainment of already known non-coding RNA. As already known non-coding RNA, we have taken from NCBI. With this one, we made a motif library. Motif library is type of library that contain our ideas. And now with this motif library, and we have our gene library, we can do a further comparative study. For the fourth step, there is creation of an artificial intelligence. We create our own artificial intelligence, and we set all the function of known ncRNA with sample, and set the gene library with an AI so that AI can score the further similarities. Now, as fifth step, we have training set with already known ncRNA. Now we train our AI with the help of TensorFlow application to check the similarities between our sample RNA and our known RNA, known non-coding RNAs. With the help of the TensorFlow, we can score as it gets if our sample RNA is more similar to the known RNA, then it will show high points, and if it has less similarities, then it will show low points. Then in seventh step, we have non-coding RNA motif identification. In non-coding RNA motif identification, we set another application, that is random forest. By the help of the random forest, we check all the amino acid, and we arrange the different sequences of amino acid with the help of random forest, and check that our motifs are identified and scored, and again scored with our known set of RNA. Function of RNA prediction. Here is our next step, and by the help of the all the gene libraries and data and scored chart sheet, the coding the sample RNA. If the sample RNA have highest score, the sample RNA that have highest score will be taken, because the sample that have highest score.
Sorry to interrupt, Sayantan. You have another 3 minutes left.
Yes, ma'am.
Yeah.
If the score is high, that means our sample RNA is more similar to the known function of the RNA, and the function will be same. By the help of that, we can determine a function of the sample RNA. With this, there is the conclusion that this process takes a lots of time and a better half of our fortune and supercomputers to conduct this type of process, and it takes many years of research and trained person. But if we able to do this types of research, we can cure some types of uncurable diseases. With this, thank you for hearing me. Thanks.
Thank you so much. Over to session chairs.
Yeah, actually, I'm sorry how to say. Actually, you presented something like, actually like a demonstration. See, actually, we need some slides which are actually software, how the software works, and show the non-coding RNA and coding RNA, something like that would have been much better presentation.
Yes, sir.
And you just only something, you were reading the slide and mentioning something. Looks interesting, but sounds interesting, but if you show it, these kind of presentations are involving demonstration, like for example, if you put some few snapshot or screenshots of the software, how does it work, and maybe probably could demo some animation video included in this slide, would have been much better presentation. Otherwise, it looks interesting. Thank you.
Yes, sir. Sir, this is just an idea behind the basic fundamental methodology, sir. I apologize if my presentation is not that good, but the technical team will try to maintain and update this presentation. Thank you so much, sir.
No. I mean, see, there is nothing wrong with the presentation. There is. Don't misunderstand me. What I mean to say, the slides you are mentioning, actually, you are mentioning something like, demonstration, because this requires something. You are talking about genetic code and all, and you use some software, right?
Yes, sir.
Yeah, that's what I'm talking. I'm interested only in the software, how you use. So if you put some few screenshots and showing that how to differentiate between the non-coding RNA and RNA, coding RNA, would have been much better presentation, I mean to say.
Yes, sir. Yes, sir. Thank you, sir. Sir, I will try to show it next time.
Thank you, Sayantan, for your presentation.
Thank you, ma'am.
Yeah. So we will move on to our next presenter. Mr. Kaustav Mahapatra.
Yes, ma'am. Am I audible?
Yes, Kaustav. So welcome, Kaustav. Kaustav is from JSS College of Pharmacy from India, who will be presenting a comparative study of anti-inflammatory and anthelmintic activities of T. orientalis by in vitro method.
Yes, ma'am.
Yes.
Let me just share the screen once.
Okay.
Ma'am, am I sharing the screen right now?
No, you're not. You have not started sharing the screen yet.
I think my screen is getting shared.
Yes.
Thank you, ma'am. Sorry for the interruption. A very good evening to everyone. So my topic for today is comparative study of anti-inflammatory and anthelmintic activities of tremor orientalis by in vitro method. So before I start, let me give you a brief fact. About 1.5 billion people all over the world, that is approximately 24% of the world's population, get infected by helminth infections and some sort of inflammatory diseases every year. So it's more prevalent in the north part of America, that is especially in the US. And now owing to the this pandemic situation, it has created a lot of stress in the economic conditions of the industries. So a research in these kind of fields is really necessary. So moving on, I've chosen Trema orientalis lin as my plant product. Obviously, it's a natural product, and let me give you a brief about the morphology. It is approximately 18 meters in height and 1.5 centimeters in width. It belongs to the family Ulmaceae, and the common Indian name is Indian charcoal. It can be found in central parts of Africa like Ghana, Ethiopia, and central parts of Asia like India, Malaysia, Australia, etc. The ethnomedicinal uses includes leaves, stems, roots, and whose decoctions can be used to treat dysentery, diarrhea, pneumonia, bronchitis kind of diseases. The major phytochemical constituents are flavonoids, tannins, saponins. The major pharmacological activities include anthelmintic activity, laxative activity, hypoglycemic activity, diuretic activity, etc. So now comes to the part of inflammation. So inflammation is basically a defensive response by the cells when they are introduced to any pathogens like microbes or any chemical agents. This can be indicated by redness, pain, swelling, etc. There can be acute inflammation. There can be chronic inflammation. Many anti-inflammatory drugs are used, over-the-counter NSAIDs are used to treat inflammation. One of which classic drug is aspirin. So aspirin works by inhibiting the prostaglandins by targeting the COX-1 and COX-2. So what is helminth infection? Helminthiasis is nothing but worm infections where parasitic worms cause infections in the stomach and intestines. So anthelmintics are used. One such anthelmintic is albendazole. It's a drug of choice in the treatment of pinworm, hookworms, roundworms, etc. So albendazole acts by joining in the beta tubulin of the parasites, and it inhibits the polymerization. And it also depletes the parasites' glycogen stores by blocking the glucose uptake. So now the main part is why we needed this study. So there is a term known as helminthic drug resistance, which is due to a prolonged use of anthelmintics. The helminths develop some kind of resistance, which can further deteriorate the conditions. Plus, Trema orientalis is a natural product, so it has less side effects and it's less toxic than the synthetic drugs. And there has been no conclusive comparative study on Trema in orientalis regarding the inflammatory and anthelmintic activities. So I've segregated the materials and methods into two different sections. One is the anti-inflammatory activity, and other is the anthelmintic activity. For the anti-inflammatory activity, I have used inhibition of albumin denaturation, heat-induced and hypotonicity-induced hemolysis. And for anthelmintic activity, I've used DIP method. So coming to the first method, that is inhibition of albumin denaturation. So the first thing is, why do we need to check for albumin denaturation inhibition? Proteins usually lose their secondary, quaternary, and tertiary structures when heat is applied. So it causes denaturation of protein, and due to inflammation, heat is applied, and so inflammation has a direct relation with protein denaturation. So in this method, I'm comparing the test sample with the standard drug, that is aspirin, and I'm checking for the percentage of inhibition of albumin denaturation. So coming to the methods, several concentrations of reaction mixture are mixed with 1% bovine albumin, and the pH is adjusted with 0.1 in HCl, and all the extracts are incubated for 20 minutes at 37 degree, and then they're heated and again for another 20 minutes. The samples are left for cooling, and the turbidity is measured at 660 nanometers of UV spectrometer. And owing to the turbidity, we can conclude that the test and the standard drugs can be compared. Coming to the next method, heat-induced hemolysis. So hemolysis is nothing but rupture of RBC. So RBCs can be ruptured by the application of heat. Again, we are comparing the test drug with the standard, that is aspirin, and we are calculating the percentage inhibition of hemolysis. So for the methods, the reaction consists of, in one test tube, we take 2 ml solution. In 1 ml, we take the different test samples of the test sample of different concentrations. And another ml, we take 10% RBC suspension. And in a different test tube, saline is taken as control. So all these test tubes are, all these centrifuge tubes are incubated at 56 degrees for 30 minutes. And then they are heated in a water bath, and then they are cooled under the running water. And then they're again re-centrifuged in 3000 RPM for 5 minutes. And the absorbance of the supernatant liquid is measured at 560 nanometers. Now, the third method is hypotonicity-induced hemolysis. Now, when we put the RBC cells into hypotonic solutions, the cells usually swell up and causes destruction of the cells. That is hemolysis. This is lysis. So we put hyposaline solutions and phosphate buffer. Phosphate buffer usually maintains the physiological conditions. So to check, and again, we compare the test with the standard to check the percentage inhibition. So different concentrations of extract were mixed separately with 1 ml phosphate buffer, 2 ml hyposaline solution, and 0.5 ml HRC suspension. And aspirin of the concentration of 100 microgram per ml was used as standard. All the mixtures were incubated at 37 degrees centigrade, and then they were centrifuged for 3000 RPM for 5 minutes. The supernatant liquid was separated, and the hemoglobin content was measured by checking under 560 nanometers spectrophotometer. So now coming to the second phase of the study, the assessment of anthelmintic activity, we use dip test. So in dip test, we take Haemonchus contortus, that is the earthworm, Indian earthworm, and we group them into 6 groups. We name them as A, B, C, D, E, and F. And for each group, we add albendazole as the standard drug. The concentration of albendazole is 20 microgram per ml, and so different concentrations of chloroform extract of the test drug is taken. The concentrations are 10, 25, 50, and 100 microgram per ml. Each group is dipped into different concentrations of test drug, and the paralysis time and death time are noted, which is compared with the standard drug. So here are the results. The first is the result for the inhibition of albumin denaturation. Here we can see that different concentrations of the test drug, 100, 200, 300, and 400 microgram per ml were taken, and the absorbance was measured at 660 nanometers, and was compared with the 100 microgram per ml of standard aspirin. It was found that 400 microgram per ml concentration, the test drug showed 41% of inhibition of protein denaturation, in compared with 100 microgram per ml concentration of aspirin, which showed 56% of inhibition of protein denaturation. Coming to heat-induced hemolysis, again, it showed a linearity. When we were taking 400 microgram per ml of concentration of test drug, it showed 38% of inhibition, compared to the 100 microgram per ml concentration of aspirin, which showed 52% of inhibition. In the hypotonicity also, it showed 44% of inhibition in 400 microgram per ml of concentration of test drug, and 54% of inhibition in 100 microgram per ml of concentration of aspirin. It showed death of the sites. In albendazole, 2.5 minutes approximately, 2 minutes approximately of test time. Whereas in 100 microgram per ml concentration of this drug, the chloroform extract, it showed 3 minutes of paralysis time, and death time was 10 minutes. So in conclusion, we can say that the anti-inflammatory activity and anthelmintic activity both were present in the chloroform extracts of Trema orientalis lin. However, Trema orientalis had a greater extent of anthelmintic activity than the anti-inflammatory activity because in the anti-inflammatory activity, it showed 400 microgram per ml concentration showed approximately around 44% of inhibition compared with 100 microgram per ml of test drug, which showed 56%. Whereas in anthelmintic activity, the 100% death of the parasites were noted at 100 microgram per ml of concentration of test drug in comparison with 20 microgram per ml of the standard albendazole. Thank you.
Thank you, Mr. Costa, for your presentation. Session chairs, any questions?
I'm having, madam, one question.
Okay, that's Lakshmikant. Yes.
So why you are using chloroform extract for the anthelmintic activity?
Sir, your voice is not clear, sir.
So you are using chloroform extract for anthelmintic activity, yeah?
Yes, sir.
So why you are going for chloroform extract? Why you are not using that is the hydroalcoholic or ethanolic extract?
Sir, actually, I reviewed some articles and it, like I got into the conclusion of using the chloroform extracts.
So because the chloroform extract is also toxic, na?
Yes, yes.
So better we have to use that is the hydroalcoholic extract or ethanolic extract.
Yes, sir. So actually, I checked for the effective dose and lethal dose of chloroform extract, and after that only I found out that up to 100 microgram per ml can be used for comparative studies with albendazole.
Okay.
Yes, session chairs, if we don't have any questions, shall we move further? Okay, Kaustav, thank you so much for your present. Okay, sir. Yeah.
Yeah, I mean, similar question actually, but okay. I mean, since you have considered the lethal dose of chloroform, and actually it's a nice presentation. You know, after a lot of presentations I have seen, I think you have done extensive work. I appreciate you first of all. But of course, you should have considered the other solvents as well, like one of the presenter is told. And otherwise, it's a good amount of work you have done. And may I ask you why you choose aspirin as a control? Why didn't you choose other anti-inflammatory drug?
Sorry, can you just repeat the question once again? Yes, sir.
You use, what is the control drug you used for anti-inflammatory?
Sir, aspirin I used.
Aspirin. May I know why you choose aspirin? What is the rationale behind choosing aspirin than other anti-inflammatory drugs? Is it a protocol one? I mean,
protocol. I followed.
Voice is breaking it.
Hello.
Pardon me?
Hello.
Yeah.
Yeah, sir. Actually, I followed a protocol. Like I reviewed an article in, it was Leela Prakash et al. 2008, and which showed that aspirin had excellent like anti-inflammatory activity, and which had like less amount of toxicity.
Less amount of toxicity. Aspirin. Okay. Never mind. Okay. No point. It's okay.
Thank you, sir. Thank you, Costa.
Thank you.
So next, I would like to invite Parkavi M. Is Ms. Parkavi or any co-author available? Parkavi? Okay. So next we have Dr. Kumar Sogata and team, that is Shivalanka Sri Chandana and Shreya Nityaja, who are independent dental practitioners from India, and their topic is contraceptive pills, a dental nightmare. Welcome, Dr. Kumar Sogata.
Hi, ma'am. Good evening. Can you hear me?
Yes, I can hear you.
Yeah, okay. I'll just share my screen. Give me a second.
Sure.
Is my screen visible?
Yes.
Yeah. So, okay, ma'am, have you unmuted Dr. Nitya and Dr. Shreya?
Yes, yes, they can unmute by themselves.
Yeah. Okay. So I'll start. Good evening. My name is Dr. Kumar Sogata, and I'm an independent practicing dentist from India. And I'm here with my fellow colleagues, Dr. Shreya Shivalanka. She's joining us from Hyderabad.
Good evening, everybody.
And Dr. Nitya Jha, she's joining us from Bombay.
Good evening, everyone.
And we are here to try and bridge the gap between dentistry and pharmacology through our review paper, how oral contraceptive pills have actually created a dental havoc within the dental healthcare system. Well, it was the year 1960 and oral contraceptive pills came into the face of reality when the United States of America finally approved it. With the coming in of oral contraceptive pills, now as I talk in this presentation, 100 million people across the globe are using it at one point in time. So this has become the second most utilized amount of contraceptions after female sterilization. However, the journey of the pill, along with its history, was not as simple as we quoted today. In the year 1937, it was for the very first time when progesterone and its action on ovulation was discovered. Slowly, as the decades unfolded, it became clear on how the synthesis of biomimicking chemical compositions of the already present estrogen and progesterone in the female healthcare system could help in progress in this particular field. Carl Desai, as we see over here, is nicknamed as the father of the pill. He was the first one to introduce it to the world. With the coming in of the pill, there was a huge boom across the world. The journalistic media like Time made it their cover and called it as the pill of the century, whereas the conservative healthcare people and the Pope himself defended the ban on the birth control. It gave right-wingers to associate it with a lot of movements called as the reproductive justice movement, the doomsday movement, etc. However, we are here to talk about the chemistry and its dental consequence. The chemistry of oral contraceptives biomimics somewhat the presence of estrogen and progesterone in our body. They work on three triad bases, that is maintaining consistent hormonic levels in the body, suppressing the release of other hormones, and it also prevents ovulation. Together, these triad of these three things help to trick the system and the pituitary gland to understand that the body is not ready for pregnancy. In the year 1978, for the very first time, International Journal of Oral Health published a report in which 273 oral contraceptive pill users were examined. And there was a direct relation that was established between the age, the duration of use, and how was it impacting the various fields of dentistry, including periodontal ligaments, the gums, the gingiva, the periodontium, and the bone itself. There were subsequent studies over the decades. In 1978, Kolkhoff talked about it and said how gingival inflammatory index scores increased with OCP users. Harrikin and Adricani's study talks about how oral contraceptives meant more pocket and attachment loss in our gingiva. Dominguez in 2011 reported how increasing probing depth and sulcular depth was being affected by OCP. Now, you must be wondering that how come a drug that was meant to reduce pregnancy rates ended up causing a dental havoc? The answer is the inflammatory response activation system, which is present in our gums or the gingiva. So what happens is that the elevated hormonals, the ovarian hormones predispose women, resulting in formation of gingival inflammation and periodontal destruction symptoms. As a result of which, three important things happen: exudation, edema, and inflammation. The triad of which are the perfect recipes to cause gingival enlargement in healthy females, as a result of which a female may also have problems such as gingivitis or periodontitis. But the story doesn't end here, because oral contraceptions have pronounced effect on gingival microvasculature. It has been shown that hormonal dosages of these oral contraceptive pills influence the impact of progesterone and estrogen receptors on the gingiva, resulting in the release of pro-inflammatory cytokinins and prostaglandins, resulting in loss of attachment. Now, we dentists quite know that once your periodontium has weakened and once your gingiva has weakened, you're bound to lose the tooth. estrogen and progesterone collectively work to maintain the health. And that is the reason why menopausal problems and pregnancy-related problems such as pyogenic granuloma is quite evident in gingival overgrowths within the women who have that. So this is a basic structure of how clinical health deteriorates. The dental impact does not only happen in the gums or the periodontium. It has been seen that a number of antibiotics prescribed by the oral physicians do not work when a woman is under oral contraception. Your TMJ itself, the only opening guiding your mouth, fails to action properly in women who do not have proper estrogen level, because bone itself is a concomitant factor of estrogen. Dry socket is another common problem we dentists have to observe in patients who are using oral contraceptive pills and then come for extractions or impaction problems. The second half of the presentation will be taken care of by my co-presenter, Dr. Shreya Shivalanka.
Thank you, Dr. Saugata. We shall now proceed with the aim of the study, that is to access the curious relationship of birth control pill and dental health. Our research question is, if there is a need to redefine our thought process before using oral birth control contraceptions. We have done a systemic review of the same involving the following steps of literature review, systematic methods to collect the data, synthesize the findings both qualitatively and quantitatively. In this process, 27 articles were collected, out of which 14 of them were finally chosen with Cochrane collaboration taken as reference, and other scientific research data such as Medline and Medno. As you can see on the screen, this was our inclusion and exclusion criteria for our presentation today. From these finally selected 14 articles taken into consideration, we have vehemently stated, yes, oral contraceptive pills and dentistry do share a common denominator. However, more case studies need to be done intricately to prove the hypothesis scientifically and further our research into developing an alternative. So what does literature has to tell about this? In the past 50 years, there have been a dramatic increase in perceptions, especially concerning to sex steroid hormones in health and disease. In general, women with pre-existing gingival conditions or even susceptibility to them, they may experience an exacerbated response to bacterial plaque. With oral contraceptives, the increase in gingival inflammation is related to the duration of use and the results. Women using contraceptives had 16-fold higher levels of bacteroid species and experienced a 2-fold to 3-fold increase in the incidence of localized osteitis following extraction of mandibular third molars. And now, the last part of the presentation would be taken over by my co-presenter, Dr. Nitya.
Thank you, Dr. Shreya and Dr. Saugata. From what was presented by my co-presenters, it is very evident how oral contraceptives take a toll on a woman's oral health. However, we strongly believe that further studies and researches should be conducted on the same, as there have been new revelations in each study that has been conducted so far. Even though the dental issues that are developed due to the intake of the pill are treatable, not all of them are curable, especially the conditions that are associated with the gingiva. There's a general misconception that dentistry is all about oral health and hygiene. Topics like these are a testament to the fact that dentistry has gone beyond the horizons of just a single tooth. I would like to take this opportunity to thank my fellow co-presenters, without whose aid this review wouldn't be a success. And lastly, I would like to thank BioLeaks for giving us the platform to throw light on such an eye-awakening topic, which is not much spoken about. And these were our sources. Thank you.
Thank you.
Thank you, Dr. Saugata and team, for your amazing presentation. So, session chairs, any questions?
Yeah, it is good presentation overall, and I think Can you, what are your future suggestions then? Probably the team, one of the team can answer.
So actually it will depend on the kind. For example, if you're talking about gingivitis, which is the basic of problems in dental healthcare, the first thing would be to not undergo any treatment if you're using oral contraceptions, right? There are a lot of other methods of contraceptions present. So if you're already prone genetically to have gingival problems or periodontal problems, or, caries is also genetic in many patients, it has been found. So if you're prone to having these problems, do not use oral contraceptions. Go forward with other contraceptive pills. Today, there is something called as an injectable contraceptions are also there. There is the concept of mini pills, which have also come up, right? So these are the alternatives which can be applied, I guess.
Thank you. Thank you.
I'd like to add a very small point, sir. So right now there is something called as combined oral contraceptives, which claims that it doesn't affect the periodontal health, possibly related to a very lower level of progesterone and estradiol than previously used medications. But yeah, further study needs to be done on this because the available information is very limited.
Okay.
Okay. Thank you.
Thank you. Thank you, Dr. Shreya. So thank you, Dr. Sogata, Nitya, and Shreya for an amazing presentation. I mean, always you guys have been giving something or the other very different and innovative topics. So that's really nice. So thank you so much for being part of this event as well. So thank you so much, and looking forward to your amazing presentations in the upcoming years and conferences. Thank you so much.
Thank you.
Thank you.
Bye-bye. So our next presenter is Arpan Bera. Is Arpan Bera available? Arpan, can you hear me? Okay. If you're available, kindly unmute, or your co-authors, if anybody available to present. We will not be recalling your names. Okay. So next presenter, Raju Ji. Any co-authors available from Raju Ji? Okay, next we have a video presentation, sir, and the author is available live. So next we have a video presentation by Golnaz. We will be playing.
Hello. Okay.
Yeah, Golnaz, okay, you're playing the video? Okay, okay, fine.
Yes. You can see my slide.
Yes, yes, okay.
Okay. Good evening.
Good evening, you may begin.
Yes, yes. Okay. My name is Golnaz Hemmati from Department of Pharmaceutics, Faculty of Pharmacy, Ahvaz Jandishapur, University of Medical Science. The title of my presentation is The Effect of Topical Quercetin-Loaded Liposome on Pregal Ulcer Healing in Rats. Introduce of quercetin. Quercetin, a polyphenolic flavonoid, extremely hydrophobic in nature, is a component of onion and is able to interact and permeate lipid bilayer, and such capacity is very important because there is a correlation between antioxidant activity and ability to incorporate into membrane. Introduce of skin. The skin is the largest organ of the body, accounting for about 15% of the adult body's weight. Introduce of pressure ulcer. Pressure ulcer is an important cause of morbidity, mortality, and extended hospital stay among patients with spinal cord injuries or ischemia reperfusion injury. A step of wound healing. Wound healing process include 1. Rapid response, 2. Inflammatory response, 3. Proliferation, migration, and contraction recovery. Introduce of liposome. Liposomes are colloidal carriers containing phospholipid as the main composition of their bilayer structure, which resemble the lipid cell membrane of the human body. See a list of material and animal in this slide. Preparation of liposome. Liposomes were prepared by fusion method. Briefly, the lipid components was heated separately and was added up to 100% to the non-lipid components, heated melted lipids, and the mixture homogenized with a homogenizer, and it to cool down to room temperature. Characterization of liposome. Particle size of liposomes was measured in triplicate by laser scattering. Encapsulation efficacy of liposome was determined directly. In vitro drug release studies were performed using dialyzed membrane method. Stability of formulation were stored at refrigerator for 3 months, and the particle size and encapsulation efficacy of the formulations were measured and compared with those at time of presentation. Viscosity of formulation were measured by Brookfield viscometer. Differential scanning calorimetry, DSC, thermogram of soy phosphatidylcholine, cholesterol, and formulation were recorded on a differential scanning calorimetry. In vitro skin penetration and retention studies were performed using jackets France cell with a receiver medium. Animal study. Pressure cell model in rats and histopathological assessment were measured. The animals were classified into 6 groups below: 1, 2, 3, 4, 5, and 6. In brief, animal shaving the hair, the back skin of the rats was placed in the clamps for 2 hours, followed by 30 minutes release, which was repeated for 4 cycles for 6 consecutive days. At day 7, clear scars were created at the back of the animal. The animal was placed in a standard mood and photographed from the high level of the wound during different days, and scar area and wound lengths were calculated by the Quick Photo Micro software and Oxyovision Rail software, respectively, for the evaluation of the rate, progression, and wound healing. For a histopathological assessment, the process of tissue reepithelialization, angiogenesis, edema, and fibroblast proliferation were evaluated. Samples were also examined for organization of collagen fibers using Masson trichrome staining. Result of liposome. You can see the particle size and encapsulation efficacy in this table. The mean particle size of the liposomes was in range of 10 and 16 nanometers as shown in table 1. ANOVA analysis showed no significant statistical differences between all of formulation. The encapsulation efficacy of formulation was in range 2 point. You can see this table. ANOVA analysis showed no significant statistical differences among all of the formulation. In vitro drug release. At 480 minutes, the percent of release was significantly different among all 3 formulations. Stability after 3 months. T-test results showed that particle size of F2 and F3 didn't change significantly over the course of 3 months. T-test results showed that encapsulation efficacy of formulation F3 and F4 didn't significantly change during 3 months storage. Encapsulation efficacy of formulation F2 was significantly differed during 3 months. Viscosity. Analysis of variance ANOVA showed a significant difference among all 3 formulations. You can see the chart for the differential scanning calorimetry in this slide for soy phosphatidylcholine, cholesterol, and 3 formulation in this slide. In vitro skin penetration and retention. The goal of this study was to prepare a formulation to provide the highest drug accumulation in the skin layers and restrict systemic absorption in order to have the optimal effect on wound healing. This is an indication that most of the drug has been trapped in the skin layers, didn't cross. Animal study. In different days of experiment period, photographs were taken from the upper surface of the wound and evaluated to measure the length of the wound and the percentage of the affected area. Analyze variance on ANOVA regarding the comparison of the length and surface area of the wound among 6 groups didn't show a significant difference during the 28 days treatment period. The result of the repeated measure of the intra-group regarding the length and the surface area of the wound in 6 groups of treatments showed a significant difference among the mean in a different days. You can see the pitch force, elongation at break, and tensile strength in this table. The results of the tensile strength related ANOVA analysis show no significant differences among all the 6 groups. You can see animal in this slide. Number 1, the group received liposomal blank, group 1. Number 2, the group received F2 formulation, group 2. Number 3, the group received F3 formulation, group 3. Number 4, the group received F4 formulation, group 4. Number 5, the group received phenytoin 1% cream, group 5. And 6, the group received no medication, group 6. In the group receiving blank liposome, group 1, at the end of day 28, there are still lesions in the cutaneous tissue. In the group of animals that hadn't received any substance from the beginning of the treatment, group 6, at the end of 28 days, there are still lesions in the skin. With periodic reviews of photos of different days in group, it was concluded that the amelioration and the rate of recovery in the group receiving—
Sorry to interrupt, Golnaz. You have another 2 minutes to conclude.
Okay.
Yeah.
F4 and the group receiving the phenytoin 1% cream in the more than other groups. Histopathological assessment. In microscopic examination, the section prepared from the wound in the different days and groups on day 28 in group without medication. Group 6, some of the ulcers were still in the reproductive and inflammatory stage. The proliferated keratinocyte cells on the sides of the wound were not yet matched, and the wound surface was still present in the sacrum, containing cell residues and strands. In the dermis, inflammatory tissue with many inflammatory cells was obvious. In the F2 received formulation, C and D formulation group 2, some of the lesions were similar to those in the untreated group 6, reproductive and inflammatory stage, and some were also in the stage of rebuilding of injury. In the lower part of the scar, a germinous tissue containing many vessels and inflammatory cells was obvious. In the F3 treatment group, E and F, the surface of the ulcer was covered by keratinocyte cells, and the germinal tissue containing vessel and a few inflammatory cells was observed in the dermis. The keratinocytes on the surface of the wound began to produce keratin. The surface of the wound in the F4 group, G and H, was well improved, and the wound were re-established. In the group, collagen bundle were thick in the stem tissue. The number of the cell and vessel were decreased, and the similarity of the part with the adjacent healthy tissue was increased. In the group treated with phenytoin 1% cream, I and J, the surface of all ulcer was covered by keratinocyte. The amount of vascularity and the number of inflammatory cells were decreased, and the collagen bundle were thick. In the group with liposomal blank medication, K and L, keratinocyte didn't completely cover the surface of ulcer. There was also a high amount of vascularity and inflammatory cell in the tissue. A staining method was used to measure collagen level of tissue. In this method collagen fibers are shown in blue. The group with the drug therapy, A, group 6, the F2 formulation, group 2C, and the non-drug liposomal group A, B, had narrow collagen fibers. In the treatment group of F3, group 3, D, F4, group 4, E, and phenytoin 1% cream, group 5F, thick and regular collagen bundle were observed. This suggested that recovery has progressed in group with thick collagen bundle and the damaged tissue is similar to healthy tissue around. Conclusion. According to the results obtained in this study, it can be concluded that administration of F4 formulation as well as phenytoin 1% cream, which is used in the clinic for the treatment of pressure ulcer or subcutaneous ulcer, result in wound healing due to its natural structure and minimal systemic absorption and side effect. This formulation can be suitable candidate for the treatment of pressure ulcer. At the end of the study and presentation, I hope you aren't tired and thank you for attention.
Thank you, Gulnaz, for your presentation. Session chairs, any questions for Gulnaz?
Gulnaz, though you joined late, nice presentation. It was very consolidated work and a protocolized work. I would like to ask you one thing. Actually, there was a publication recently on the phenytoin 5% and 10% case series studied against neuropathic pain. So may you know what is the rationale to use 1% phenytoin?
In this drug study, the formulation of phenytoin 1% in the drug study, and I can use it.
Yeah, I understand. Yeah, you have used as a phenytoin 1% cream, but actually I have a publication proof that it says that there was a case series done, a comparison study done 5% phenytoin and 10% phenytoin against neuropathic pain, because
yes,
usually skin ulcers also includes the pain and inflammation as well, isn't it?
Okay, it's true, but I can this formulation for prepare by for my formulation. This is medication in this drug study and use in Terial.
Okay. Maybe you may have some reference for that, isn't it?
Again?
You might have some reference for that, isn't it?
Yes. I list in this my presentation. At the end of list of my presentation, I list.
I know. Yeah, because your comparison is on question, not actually on phenytoin cream, but you just used 1% phenytoin cream, isn't it?
Okay. Thank you.
Thank you.
Thank you, Gulnaz, and thank you, sir. So next, I would like to share my screen for Lakshmikant M. Purane from Shivnagar Vidya Prasarak Mandal's College of Pharmacy from India, who is presenting a review on urolithiasis, its diagnosis and their treatment.
Myself, Lakshmikant Purane. I am from India. Topic of my oral presentation.
Are you able to see Lakshmikant's presentation?
Hello.
Yes, are you able to see Lakshmikant's presentation?
No, ma'am. Actually, video is not on.
Okay, just a moment.
for your presentation. Any questions?
Hello.
Okay, the questions can be asked in the chat box.
Hello.
Sir, is it possible for you to share the screen? Your video is not playing, sir.
Can I play from video from my laptop?
Yes, yes, sure.
Start.
So you can play now. It's visible to us.
Okay, ma'am. I will play.
Sir, we cannot hear you. Can you please enable the audio from your laptop? So maybe we will get back to you. Meanwhile, we will go on with the next presenter. Niharika Modi, can you please check your audio? If that means we will have to give you a last chance to present. Niharika, can you please check your audio?
Am I audible?
Yes, you're audible now. Please continue your presentation.
My slides are visible, ma'am.
No, Niharika, your slides are not visible. We can see your screen, but the slides have not started playing. Can you please check it quickly?
Yes, ma'am. Just a second.
Now?
Is it visible, ma'am?
Yes, it's visible now. You may begin.
Okay. Good evening, everyone. This is Niharika Modi. I am currently pursuing MPharm in Pharmaceutical Biotechnology from JSS College of Pharmacy, Ooty, India. So my topic is carbon nanotubes, treatment and applications in neurodegenerative and Alzheimer's disease. So neurodegenerative and Alzheimer's disease. First, I want to give a brief introduction about both So neurodegenerative disorders are characterized by loss of neurons, and there are intracellular aggregations of proteins. There can be two conditions in neurodegenerative diseases that can be: first, conditions causing problems in movements; second, conditions affecting memory and conditions related to dementia. And Alzheimer's disease is the most common type of neurodegenerative diseases. And the mechanism involved in neurodegenerative disorders could be oxidative stress. That is when, when antioxidant levels are decreased in the body. Excitotoxicity, like when toxic actions of excitatory neurotransmitters takes place. Depletion of neurotransmitters, and mitochondrial dysfunction. Alzheimer's disease, like in Alzheimer's disease, there is degeneration of brain neurons, especially in the cerebral cortex, and presence of neurofibrillary tangles and plaques containing beta amyloid cells. Role of nanotechnology in neuroprotection. Nowadays, nanotechnology can play a very advantageous role in the neuroprotection because it is safe and has high efficacy. Drugs target to specific sites to reduce off-target toxicity, and also it has improved pharmacokinetic behavior by sustained release of drug with wide safety margin. It can be used in other fields also, like brain tissue engineering, and other advantages can be like high bioavailability, in vitro and in vivo biomedical research. Now let's talk about what are carbon nanotubes. So carbon nanotubes, they are cylinders formed by rolling or folding of a graphene sheet, that is graphite. They are constructed from hexagonal rings of carbon. These carbon nanotubes are actually called as an example of true nanotechnology because their diameter is less than 100 nanometer, and they're also very thin, like 1 or 2 nanometers. As mentioned in slides, there are mainly two types, like single-walled CNTs, in which a single layer of graphene is present, and then multi-walled, in which multiple walls of graphene is present. Now comes the mechanism. So CNTs can cross the cell membrane without causing cell death or excessive damage. That is the main actually advantage of carbon nanotubes. That's why carbon nanotubes are trending nowadays. Like how carbon nanotubes crosses blood-brain barrier. So the blood-brain barrier comprises of endothelial cells that surround the capillary walls and are sealed together by tight junctions. The capillaries are enclosed by end feet of astrocytes. The blood-brain barrier, it is like there are several mechanisms by which carbon nanotubes can cross the blood-brain barrier, like the transmembrane passive diffusion of highly lipid soluble and low molecular mass compounds. Second will be like transporter proteins, including carrier mediated transport. The third can be the receptor mediated transporter. So adsorptive mediated transporter is also a mechanism through which the molecules of drug passes the blood-brain barrier. Strategies that center on nanotube-based drug delivery system should be designed to interact with the cells in the blood-brain barrier at a molecular level without interfering with its function. Because if the carbon nanotube will interfere with the functions of the cells, then it won't be a good drug delivery system. So the nanotube should bear at a minimum surface bound functional groups for targeting and crossing the blood-brain barrier, and should prolong its half-life in blood. It should avoid the reticuloendothelial system damage. In vivo nanotube-based gene delivery vectors like DNA, RNA, and oligonucleotides, they might be safer and more effective than viral vectors. CNTs in drug delivery for neurodegenerative diseases and Alzheimer's disease. CNTs are considered as like highly promising drug delivery systems because they can easily penetrate the blood-brain barrier. Carbon nanotubes also have improved solubility in physiological solvents, and also in blood-brain barrier there is low permeability of anti-tumor drugs.
Excuse me, Niharika, you have another 3 minutes left.
Okay, ma'am. In blood-brain barrier, there is low permeability of anti-tumor drug molecules. That's why carbon nanotubes will be a good choice for anti-tumor drug delivery. That's why carbon nanotubes came into existence. CNTs for various neurological diseases, therapeutics as quality medication. The treatment through carbon nanotubes, they are indicated as a quality medication. So like from carbon nanotubes, there are two things, in vitro and in vivo. In vitro, it comes neuronal differentiation, neural stimulation, and neural interface. And in vivo, it comes neurooncology, neuroregeneration or degeneration, functional neurosurgery, stroke, drug abuse, addiction. So carbon nanotubes have a wide variety of applications in vitro and as well as in vivo. So improved cytocompatibility of carbon nanotube after functionalization. Like here examples are given. Toxicity before functionalization, the cell is damaged, but after functionalization with carbon nanotube, toxicity can be reduced inside the cell, and therefore the cell can become healthy. Second will be the agglomeration of cell, like when a collection of mass occurs on the cells, when we will functionalize it with carbon nanotube, the agglomeration of cell will be reduced and the cells can be separated easily. And carbon nanotubes also helps in cytoskeletal disruption of the cell. So what are the recent trends that are related with carbon nanotubes? It is nowadays used in gene therapy, which involves transport of the correct gene by viral or non-viral vectors to the effective area. It is a promising vector for gene encoding nucleic acids, and it is used to deliver proteins, nucleic acid, drugs, antibodies, and other therapeutic agents inside the body. And to decrease the tumor growth, carbon nanotubes are used, and it is a very good non-viral vector for gene therapy. So the conclusion is, because of its unique chemical, physical, and biological properties, actually it has a nanoneedle shape and hollow monolithic structure. That's why it has ability to get the appropriate functional groups on their outer layers. CNTs are very good viable drug carriers, and they can be functionalized to the more water soluble and serum stable. As I mentioned earlier, they are very they can be soluble with so many solvents. So with low toxicity at the cellular level, detection of CNTs does not require any type of fluorescent labeling such as quantum dots. It can be detected directly by TEM or AFM due to that electron emission spectroscopy. And nanotechnology is an emerging field where research about advancement of nanotubes is still being studied. So hope that nanotechnology, carbon nanotubes will be more, will have more advantages in future. Yeah, thank you for the attention.
Thank you. Thank you, Niharika.
Thank you, Niharika, for your presentation. Niharika, I want to ask one question. Actually, this is a review, right? You put a review there, yeah, right? Isn't
Yes,
it?
sir. Yes, sir. Yes, sir.
Can you, in your review, can you encounter any nanotube formulation commercially available?
No, sir. Actually, the papers which I read, this is actually some my B Farm level review paper. So I presented that only, yeah. So actually, and it is, sir, actually emerging. Like nowadays, carbon nanotubes are not in that much trend, but yeah, it is like people are using it for Alzheimer's disease and especially for cancer. So
Because nanotube, you just mentioned about the nanocarbon tubes, or since 1991, they were emerging in your introduction. I saw 1991.
yes, sir.
Almost 30 years. It's not recent. 30 years already, right?
Yes, sir. Yes.
According to your introduction review.
Yes.
So the emergence of nanotubes, any commercially available in the field of pharmacy, did you encounter anything like in your review? Because the review has to be extensive, you know. Whenever
Yes,
we
sir.
do review, whether it may be undergraduation or post-graduation, the review is review only. You have to do up to date.
Yes, sir. Correct.
So when you do it up to date, and that you need to encounter any, because when we support the hypothesis or the problem statement, you strongly support that yes, carbon nanotubes commercially available, few formulations which are used pharmaceutically, you know, and nanotechnology for the treatment of cancer, for the treatment of Alzheimer's disease, for the treatment of brain tumor, because there are certain diseases where, you know, the transformation of drug into brain is very typical, you know, especially in brain
Yes,
tumors.
sir.
You know?
Yes, sir.
And you need to mention in that way, you know, you need to focus on that way, you know, into one direction, you know.
Okay,
So
sir.
the review would be
Yes.
more presentable and more appropriate for acceptance of publication as well as in conference proceedings.
Thank you, sir, for your guidance. I will definitely work on it, sir. Definitely.
Yeah, in the
Thank you.
direction it would be better. Thank you.
Thank you, sir. Thank you.
Thank you, Niharika. Thank you, sir.
Thank you, ma'am.
And our next presenter is V. Purna Pushkala from Sri Sai Ram Siddha Medical College and Research Center from India. And she'll be presenting a review on the antibacterial efficacy of Siddha formulations against Helicobacter pylori.
Good evening, all.
Good
Am I
evening.
audible?
Yes, you're audible. You may begin.
Thank you. Thank you. Is my screen visible?
Yes, yes.
Okay. Thank you so much. Okay. Very good evening to all present here. I'm V. Purna Pushkala, Professor, Department of Microbiology, Sri Sai Ram Siddha Medical College and Research Center, India. So I'm going to present a review on the antibacterial efficacy of Siddha formulations against H. pylori. Starting with peptic ulcer. Peptic ulcer, as we all know, is the sore in the lining of the stomach or the intestine. And the causes for peptic ulcer are the medicines that we intake, like certain painkillers, NSAIDs, or an important cause for this peptic ulcer is H. pylori, Helicobacter pylori infection. The symptoms of peptic ulcer would be stomach pain, burning stomach pain, feeling of fullness, bloating, intolerance to some fatty foods, heartburn, nausea, and sometimes it can also give symptoms like vomiting, blood, or a dark blood in stools, trouble in breathing, and unexplained weight loss also. So the risk factors for this peptic ulcer would be our lifestyle and the food that we take. Smoking, drinking alcohol, and even untreated stress could be an important cause for this ulcer. And coming to the Helicobacter pylori bacteria, which is the main causative organism in causing ulcer. So it causes ulcer in the mucosal layer, which is protecting the underlying tissues of our stomach. So often this bacterium causes inflammation of mucosal layer in the stomach, producing an ulcer. And this bacteria can be transmitted from one person to person who are in close contact, or it can even be contracted through food and water. So the complications of peptic ulcer would be internal bleeding, perforation in the stomach wall, obstruction, or it may even lead to gastric cancer. A statistical analysis says that about 10% of world's population suffer from ulcer, and around 15,000 deaths occur every year because of this ulcer. And in the pharmacology market, 5% of market share are spent on antacid and anti-ulcer drugs. So this is a very alarming statistical survey. So in the current treatment methods given for peptic ulcers, proton pump inhibitors are given, histamine blockers are given, but though there are such treatments available, the problems that we face here in nowadays H. pylori infection are the increased drug resistance that are emerging in the bacterial population. So drug resistance, as we all know, is the resistance against the commonly used drugs, antibiotics. So this drug resistance poses a major threat to the medical field in treatment of various diseases and H. pylori infection as well. And poor patient compliance is also there, and availability of these drugs in certain areas is also poor, which all leads to the ineffective eradication of this H. pylori infection. So the future prospects will be, we can supplement these drugs with novel types of PPIs that can be identified, or importantly, we can supplement the drugs with probiotics, which can act as a natural supplement for treating H. pylori infections or ulcers. And we can also target on alternative therapeutic methods like medicinal plants or novel drugs that can be identified in drug discovery. And future perspectives should be, we should optimize the antibiotic use and also monitor the adverse reactions that are happening because of the current treatments for ulcer. And the shorter and effective treatments should be given, and increased specificity of the medicines should be there in order to eradicate this H. pylori infection. So talking about the alternative systems of medicine, Siddha, one of the ancient system of Indian traditional medicine, it constitutes a number of polyherbal formulations and herbomineral formulations. These formulations are available in the form of powders like Churanam, Chenduram, and Parpam, in the form of oil as Thailam, and Vadagam in the form of tablets. So there are many classical Siddha texts which contains a plethora of medicinal preparations which are used nowadays for a number of clinical conditions. So Siddha also has many medicinal preparations for peptic ulcer.
Excuse me, ma'am, your slides are not moving.
Is my slide moving now, ma'am?
No.
No?
You are still in the first slide.
Oh, okay. Is it not moving now, ma'am?
Yes, it's moving. Can you please?
I will. Yeah. So coming to Siddha formulations, there are many medicinal preparations for ulcer in Siddha. Is it moving now, ma'am?
Yes, it's moving.
Okay, thank you. So my main objective of this review was to have an overview on the potential anti-ulcer drugs of Siddha medicines. And forthcoming studies would be to explore the antibacterial activity of these drugs against H. pylori infection. Starting with Nilavagai Churanam and Tamarai Parpam. These are the two medicines which were which underwent an in vivo animal study. And Nilavarai Churanam and Tamarai Parpam were given for animals which were ulcer induced with pylorus ligation, aspirin, and gastric lesion induced in rats. Investigation of these drugs were done at a dosage of 200 mg per kg and 250 mg per kg body weight respectively. And it was able to find, it was found that these two medicines had significant anti-ulcer activity, and it had no toxic levels even up to 1000 mg per kg body weight. The second would be Sangaparpam and Silasathaparpam. These two are also extensively being prescribed as anti-ulcer agents in Siddha. So the in vivo animal studies were conducted on Bister albino rats for these two medicines also at a dosage of 200 mg per kg and 1000 mg per kg body weight with ranitidine as control. So ulcer models used were phenylbutazone induced and stress induced models. And the mean ulcer scores also showed that these formulations also had significant anti-ulcer activity. The third would be Tamiraparpam, which is one of the herbomineral formulation in Siddha. So the anti-ulcer efficacy of this was also studied in two animal models, aspirin and pylorus ligated and HCl ethanol induced ulcer induced models. And after the treatment of Tamiraparpam, it was reported that it had inhibition on ulcer scores, and also it changed the pH and gastric volumes. And this could be mediated by its free radical quenching property.
Excuse me, ma'am, you have another 3 minutes to conclude.
Yes, ma'am. Yeah, sure. So coming to Vellarigu Churnam, a phase 2 clinical trial of this churnam was conducted for peptic ulcer disease in 40 patients, giving 30 mg per kg body weight for 30 days. And based on the clinical improvements, it was also found that it had significant anti-ulcer activity. Agastya Reladi Churnam, this is also one of the drug prescribed for peptic ulcer in Siddha. And this was also tested in the animals with 500 mg per kg and 1000 mg per kg body weight. And the ulcer score revealed that this also had anti-ulcer activity. Yeladi Churnam, a review on the Yeladi Churnam showed that the ingredients present in Yeladi Churnam, they themselves had pharmacological activity of gastroenteritis and peptic ulcer. So this could be a potential and cost-effective therapeutic for PUD. And lastly, Panchalavana Dravagam. So this is also another herbomineral formulation prescribed for gastritis and dyspepsia. So this was also tested at 1 ml per kg and 2 ml per kg for 7 days. And it was also found that this also had significant anti-ulcer activity. So on the conclusion, there are many Siddha medicines that are yet to be explored, especially in the field of gastritis and peptic ulcer. So these possible medicines that were chosen could be a potential, could have a potential antibacterial activity against H. pylori. And hence my future studies prospects of this review would be to evaluate these antibacterial efficacy of these drugs on against H. pylori, and also to determine the MIC of these drugs to put into clinical use, and also to evaluate the phytochemical constituents that are present in these drugs. These are the references that I would like to quote. I thank you all for giving me this opportunity. Thank you.
Yes, thank you, ma'am, for your presentation.
Yes.
Session chairs, any question for Purna Pushkala?
No, actually.
Okay, thank you.
Thank you, sir.
Thank you. Yes. So next, Mr. Lakshmikant M, are you ready for the presentation?
Yeah, madam, I am ready.
Okay, okay, please continue, sir.
Can I share my screen?
Yes, please. Yes, please.
Okay. Myself, Lakshmikant Purane.
Excuse me, sir. Sir, can you put on slide mode and begin? It's very small. Can you put on slide mode?
Yeah.
Yes,
okay.
yes. Now it's better. Thank you.
Myself, Lakshmikant Purane. I am presenting from India. The topic of my presentation is a review on urolithiasis, its diagnosis and their treatment. First part is the introduction. Urinary stone constitute one of the commonest disease in the world, and pain due to kidney stone is known as worse than that of labor pain. Among all the pain, abdominal pain always drag not only patient attention, but also the curiosity. It is estimated that 12% world population experience renal stone disease with recurrence rate of 70 to 80% in men and 40 to 60% in women. Urolithiasis is a complex process that occurs due to imbalance between promoters and inhibitors in the kidneys. In the treatment of kidney stone, various synthetic drugs are available in the market. Even though surgery is the treatment of choice for urinary stones, lifestyle changes are also important, as the recurrence rate is high as 50% within 5 years after surgery without medical treatment. Herbal drugs can be easily available, and it does not produce any types of complications that like synthetic drugs to patient. The combination of herbal medicines with allopathic treatment has a great idea to get rid all the complications related to urolithiasis. Then the next part is, that is the urolithiasis is the process of forming stones in the kidney, bladder or urethra. Urolithiasis is a condition arising from the formation of renal calculi when the urine is super saturated with salt and minerals such as calcium oxalate, steroid, uric acid and cysteine. 80% of all stones contain calcium, most commonly as a calcium phosphate 20% and calcium oxalate 60%. These urinary tract calculi having different size, from small gravel-like stones to large staghorn calculi. The stone may stay in the position in which they are formed or migrate down the urinary tract, producing symptoms along the way. The other factor that leads to kidney stones or urinary tract stone production is the formations of Randall's plaques. The chances of kidney stone or urolithiasis formations are more common in men as compared to the woman. Then the next part is, so what is the mechanism for the urolithiasis, or how the urolithiasis are formed? Various factors contribute to the formation of kidney stone. The supersaturation of urine by constituents such as cysteine, calcium, or oxalate is the most widely accepted mechanism for stone formations. So in case of the mechanism of formations of urolithiasis, the first part is increase the urinary supersaturation, and this will be occurred due to the hypercalcemia, hyperuricosuria, hypocytruria, hyperoxaluria, low urine volume, and increased urine pH. Then there is a formation of, that is the crystal nucleation. This is the first steps for the urolithiasis. After crystal nucleations, the next step is a crystal growth. That is, the size of the stone is increases, and after increasing the size of the stone, there is a crystal aggregations, and after crystal aggregation, that crystals are interact with the cell and crystal retentions within the kidney or renal collecting duct, and finally there is a stone formations. Then the diagnosis of urolithiasis, there are three tests. That is, first one is the blood testing. Blood test measure calcium or uric acid in the blood. Blood test result help to monitor the health of kidneys and may lead physicians to check for other medical conditions. Second one is the urine testing. That is the 24-hour urine collections test show that the kidney excreting too many stone forming minerals or too few stone forming substances. For this test, physician may request that at least perform 2 urine collections over 2 consecutive days. And third one is, that is the imaging. Imaging test may show the availability of kidney stones in urinary tract. Other imaging options include an ultrasound and intravenous urography, which involve the injecting dye into an arm vein and taking X-ray or obtaining CT image as the dye travels through the kidney and bladder. Then the types of kidney stones. There are several locations for the stones along the urinary tract, such as kidney, ureter, urinary bladder. Kidney stones are classified as either staghorn or non-staghorn. So whenever the size of kidney stones is less than 5 mm in diameter, have a greater possibility of passing out. But so whenever the size of the kidney stone is increases up to 5 mm to 7 mm, having only 50% chances to pass out. And those stones that are having the size is more than 7 mm, they cannot pass out and always require urological interventions. The first types of this kidney stone is a steroid stones. Triplet phosphate or steroids constitute around 5 to 10% of kidney stones and formed in presence of bacterial infections and is a crystalline substances made of magnesium ammonium phosphate. Second one is calcium stone. Calcium in combination with oxalate, phosphate, and also with uric acid constitute the major proportion of kidney stones. Oxalate is naturally occurring substances, is present in various food items such as some fruit, vegetables, nuts, and chocolates, which exhibit high oxalate level. Third one is the uric acid stone. Uric acid stones are yellow, orange, and smooth stones made up of uric acid and constitute around 5 to 10% of kidney stones. Fourth one is a protease inhibitor stones. So this types of stone is commonly observed in HIV patients. Those are using the protease inhibitor drugs for the treatment of HIV. Then cystine stones. Stones are caused due to hereditary disorders such as which causes the kidney to secrete excessive amino acid, and these are rare types of stones. Last one, that is the silica stones. Silica stones are formed due to the use of different types of medicine as well as different types of herbal products such as sulfa medications, as well as certain antibiotics, that is the ciprofloxacin, as well as some certain product that containing the silica. Then the symptoms of urolithiasis. The symptoms of urolithiasis, that will be depends upon the type of stone. So whenever the stones are small, the symptoms are different. So whenever the stones are having larger size, the symptoms are different. But overall, the urolithiasis includes sharp and cramping pain in the neck and side. This feeling move to the lower abdomen. The pain starts suddenly and come in wave. Some another symptoms of this urolithiasis including a feeling intense need to urinate, urinating more often, and burning feeling during urinations. Urine color becomes dark and red due to the bloods, vomitings, nausea, and man feels pain at the tip of penis. Then the treatment of urolithiasis. In case of this small stone, the small stone does not require much treatment. They get off the body by drinking more amount of water. So whenever we take the water, that is 4 to 5 liters per day, water helps to get rid of stones throughout the body through urine. The movement of stones create pain. Such moving pain can be treated with the use of certain types of pain reliever drugs. Whenever there is a presence of large stones are not easy to remove by drinking more amount of water. These stones may harm for the body because they can damage the kidney, as well as they can damage the nephron, as well as they can cause the internal bleeding. So at that time, some certain medical treatments are necessary. Medical therapy for kidney stones, usually physician prescribes some certain types of alpha blocker drugs. So what is the role of alpha blocker? That is, the alpha blocker relax the muscle in the ureters, which helps to pass the kidney stones more quickly. The next one, that is the extra corporeal shock wave lithotripsy. In this process, by using the sound wave or shock waves to create strong vibrations to break the stone into small pieces, and these small pieces of broken stones can pass out body through the urine. The next one, that is the uteroscopy. Some stone which gets stuck in the uterus or bladder, uteroscopy procedure is used to remove the stones. Uteroscopy can examine the upper urinary tract stones. This procedure is painful and contain a small wire which are connected with a camera at the end, and the wire is inserted into the urethra and passed into the bladder for removal of
Excuse me, sir,
stones.
you have another 2 minutes to conclude.
Okay, so with a cage connected with it. Then one more, that is the lithotomy. That is, if the physicians find the large stone or near kidney, the nephrolithotomy is the one of the better choice. Then the treatment through allopathic medications, and certain types of allopathic medications are available. For example, thiazide diuretics, allopurinol, potassium citrate, as well as acetohydroxamic acids. Then herbal treatment of kidney stones. So herbal or herbal drugs product are popular in the treatment of kidney stone, because whenever we use the synthetic drug, they can cause certain types of adverse effects. So most of the time, these herbal drugs are used for the treatment of kidney stone. For example, berry root bark, fenugreek seed, shatavari root, and gravel root. And finally, that is the conclusion. Kidney stone is one of the most common problem in developing countries and rest of the world, which affect the urinary system. The chances of kidney stone or urolithiasis formations are more common in men as compared to the women. Major reason behind the kidney stone formation is the supersaturation of urine with calcium and oxalic salt, followed by the crystalline particle. High fat diet, inadequate nutrition, addition of food that contain oxalate crystal, high protein diet increases the risk of kidney stone problems. The abnormal growth of parathyroid glands, which control the metabolism of calcium, and this condition create high level of calcium in the blood and urine, which causes the kidney stones. The treatment of kidney stones, such as allopathic or herbal medications or removal of stones through surgery, is now in trend, but most of the people prefer herbal therapy for removing kidney stones. Herbal treatment needs time for their effect, but it is the safest and inexpensive treatment. So these are the certain references we have used during the literature survey. Thank you for giving me opportunity to present my work.
Thank you, sir.
Is there any questions?
Can you show the references?
Okay.
Dr. Lakshmikant.
Okay, I will show the references. So this is the first slide of references.
Can you show some latest references, 2020-21, please? If you have included though. 2019 is there.
Actually, sir, I have published one paper on urolithiasis. Actually, it is the literature survey of my PhD work.
Yeah, no mind, but could it be updated, because you are presenting in 2021? Maybe there might be some sort of other treatment options which include which will be scientific world would like to know about the other treatment options for the kidney stones, latest options. You know?
So actually, sir, there are a number of treatment for the kidney stones, but exactly there is no anyone drugs which can cure the kidney stones. So particularly most of the time, most of the people, they are prefer the herbal medicines.
You have any reference for that?
Yeah.
Which number? Which number you mentioned in that? This. Okay. Because there are no titles actually. Some of them are. Right. Which one? Which reference is saying that herbal medicines are using for kidney stone?
Can I show, sir, one more paper? Myself, one more paper?
No. It's okay. No. I'm just asking if whether you're, because whenever we climb any.
Actually, sir, my published paper I have not included in these references, sir.
You need to include all references whenever you climb to say that herbal products. Some herbal products you mentioned for the kidney stones, right? So we might be using. Anyway, no mind. Thank you very much.
Okay.
Good work. Okay, thank you.
Thank you, sir.
Thank you.
Thank you, sir. Thank you, both of you. So next, I would like to invite Raju ji from Sri Gokulam Medical College Research Foundation, Vanyaramura, Trivandrum, Kerala, from India. And his topic for presentation is effect of body weight and organs on Dactylosa hategria, suroot extract using cyclophosphamide-induced method in male albino rats.
Who is the presenter now?
The presenter has sent his video presentation, but unfortunately it's not supporting. So with this, we'll come to end of session 1, that is pharmacology and toxicology. Thank you so much to our session chairs, Professor Dr. Nehla and Dr. Bunta Venkata Subramania Lokesh. Thank you so much, both of our session chairs from Malaysia, who had been patiently evaluating all our presenters for the 2 days of our 9th World Conference on Pharmaceutical Science and Drug Manufacturing 2021. Thank you so much. So any comments that you would like to give at the end of the session? How was your experience evaluating the presenters?
Yeah, actually, I mean, first of all, I congratulate all those presenters who patiently present their slides. And a suggestion also too, actually, whenever we present in conference, the slides should be omitting the introduction part. Nobody would like to listen to the introduction part, because even though if it is a review, you just mentioned that one slide just mentioned that this is the literature review, and one slide you mentioned your discussion, and few slides you just conclude if any data is present, and that makes more sense of what you call presenting your data to the scientific community, because and also it shouldn't be bulky. And first point, and we know that there is a self-urgency to present in conference and just get the certificate and all. That intention we need to come out of it, because world conferences actually play a major role to what you call emulate, simulate the new research. People are very anxious to know what is the new research is coming up, what is the new updates are coming up. People will be sitting, like for example, I think more than me, Prof Naha also can explain about all this, because the research is actually upfront, in more advanced. So we whatever we are presenting, we need to make a sense of adaptability and into it, not just reading the slides or probably just word by word reading and all, because that makes maybe informative. We can also read. If you can send it to be a poster, that would be very good for poster actually. Such kind of more informative you want to give, actually for the reviews. So, and some are very good actually, and I try to evaluate, and I put in a brown color. So a few of the 5 or 6 presentations were very good actually, because I am seeing very new actually to hear about. For example, I can't reveal now because that will be up to the committee to decide. I just pass on the file to them now.
Sure,
and thank you also. I really appreciate Jaljilia, because she's so patient, since she's sitting like that only. I don't know when she have a supper and other things, tea, coffee and other things,
Thank you, sir. No problem at all.
and it's good to see that. And I also appreciate Prof. Dr. Naha for her absolute comments, on certain things, really, within a humble way. And thank you, Prof. Actually, nice meeting you. I'm with you, University Malay. We can meet sometime, in the University Malay and for collaborative work. So thank you all. Again, congratulations to all presenters for your excellent work and presentation.
Thank you, sir. Thank you so much. Professor Nehla, anything that you would like to convey to the presenters?
Yes, Jazila Mustafa. What a very long day for all of us.
Yes.
But I would like to congratulate all of the presenters and their team members for all the efforts, in getting into research. Doesn't matter what type of research that you are doing, starting from the review until the lab base and so on and so forth. Everything is like for us to get to know more. We learn things from each other. Only that regarding the presentation, I think my partner, Dr. Lokesh, has given a very good summary. For future references, if we are going to present oral especially, and then we have to look into the duration that given by the organizers. From there, actually we know how long, like what are the things that we're supposed to deliver within that particular stipulated period. And please be focused on the title, and I think all of you know the conclusion should be the one that answered the objective, or if you have any hypothesis for your study, okay. And if you have any statistical analysis, make sure the analysis should be the correct one. And I think some of the works that have been presented have a very good potential to be thoroughly investigated, or it can be published in a very good journals. And I wish you all the best for the remaining less than 1 month of the year 2021. We'll move to 2022, 2022. Happy New Year in advance, and those celebrating
Happy
Christmas.
New Year in advance to you as well.
And those celebrating Christmas, Merry Christmas, and wish all the best for everyone.
Thank you, ma'am. Thank you so much. And yes, advance Christmas wishes and New Year wishes to everyone who have joined us. And before we wind up, I would like to give some informations to the participants who are here. Some presentations are still going on in other rooms. If you're interested, you may join the rooms. And I have shared the certificate link in the chat box. So maybe I'll just share the screen on how you have to download your proceeding books and the certificates. So all you have to do is, just a moment, log into, yes, sir.
Ma'am, I have one problem.
Okay.
Actually, ma'am, I have registered for video presentation.
Okay.
But right now I have presented paper orally.
Okay.
So please give me a certificate of oral presentations instead of video presentations.
Yes, you may connect with the coordinator and they will do the necessary for the change of certificate.
Okay, thank you.
Yes. And the evaluation reports will be received for all the sessions by the session chairs by today, and the winners will be announced within a day or two via email. The best paper, best oral presentation, and best poster presentations for all the sessions. So kindly give us a day or two's time to evaluate and announce the winners officially through our mail. And next, I would like to share my screen and show you how to
Good
download
evening.
the certificate. Yes.
I'm Dr. Sudhamani from SSM College of Pharmacy.
Yes, ma'am.
Not at present my e-poster. So what's the next thing I have to do?
Ma'am, poster presentations have been moved to Hall C. Maybe you joined late, so you were not aware of it. All the poster presentations have been moved to Hall C long back. So maybe you can move on to Hall C. A few presentations are still going on there.
Okay, ma'am. Thank you. Thank you so much.
Thank you. I'm sorry, I'm not able to share my screen. So I'm putting the link here to download your proceeding books and the certificates. All you have to do is log into www.virtual.bioleaks.com. and you have to log in by entering your email address and name and contact details. And then you can just log into it and download your conference proceeding book and certificates. When it comes to certificates, if you are an already existing member of APR, please sign in. If you are a new member to Association of Pharmaceutical Research APR, sign up with the following details and download the certificates from the tab Certificates and Rewards. In case if you find any difficulty, please connect to our helpline numbers and our team will be responding to you and helping you in downloading the certificates. And please don't forget to give us your feedback. Your feedbacks are really valuable for us to improvise our conferences next time. And don't forget to join us at the Pharmacon Malaysia coming up in 2022 and the 10th World Conference on Pharmaceutical Science and Drug Manufacturing happening at UAE next year. So those who were unable to join us this time at Dubai, please do join us next year. And thank you so much for your amazing presentation. Thank you. Good evening. Have a nice day.
Hello, am I audible to you now?
Vaishnavi? Vaishnavi, your presentation is at Hall C. Yes, with this, we come to the end of the 9th World Conference on Pharmaceutical Science and Drug Manufacturing 2021 at Dubai. So thank you so much for all the participants and the keynote speakers who have joined us at Dubai as well as virtually on Zoom platform. Thank you for your amazing presentations and enlightening with us your recent research activities. Thank you so much for being part of this and making this event a great success. So before we wind up, I would like to give information regarding the certificate download. The certificate download link has been shared in the chat box. So if you're an APR member, please make sure to sign in. And if you're a new member to APR, you just have to click on the link and sign up with the details. The link has been shared here in the chat box. And also to download your conference proceeding books, you have to click on to www.virtual.bioleaks.com. The link has been shared in the chat box. And you may click on by entering. You have to enter your email address and contact number and your name and log in and download the proceeding books from the download section. And please make sure you give us your valuable feedback. Click on the feedback link and do share us your feedback. And let us know if there is anything that we would like to help you with regard to the 9th WCPSDM, and be part of our 10th WCPSDM next year in 2022 at Dubai. So thank you so much. If you have any questions, please do reach out to our helpline numbers. Thank you once again. Have a great evening and advance Christmas and New Year wishes to everyone. Thank you so much.